behavioral. And I did not see that our patients are shifting their behavior. We gave them those diets for heart disease, for gastrointestinal issues, for everything, you name it. And they were not changing behavior. They reverted back to their old habits, to diabetes, to liver issues, you name it. That made me dive early on in the 2000s into the field of coaching, actually, to see how I could change behavior. That took me deeper into work in nutritional genomics and the effect of our genes and expression of genes on the way we are affected in our health. And that took me to another PhD in human behavior and counseling. So I call myself a wellness and leadership expert, where I help individuals actually become the better version of themselves, where their emotions and their gut and their health are completely connected. This connection between the brain and the heart and the gut is real. And now science is proving it. And even our genetic personalities, now we can test also our personality and our genes, and we can see how this will affect our health. So it is a fascinating time to be in this world in the field of science. If I want to give you a bit about the utilitarian conferences gathering, you all know, and you've been here before, that it leads in organizing professional meetings and conferences, and especially in the healthcare and the clinical, medical sciences and pharmaceutical sectors. UCG bridges this academia industry, driving excellence and innovation in every organization. On behalf of UCG, I extend heartfelt gratitude to this organizing committee, our esteemed speakers, and all the participants whose contributions have made this event really possible. Together, we are advancing the future of pediatric adolescent healthcare worldwide. We will provide our speakers with certificates right after their presentations, and delegate certificates will be given at the end of day 2. And without further ado, I would like you to welcome our first speaker, Dr. Al-Mubarak, Dr. Muhammad Al-Mubarak, Clinical Medicine and Healthcare Leadership from Saudi Arabia. Hello, Dr. Muhammad. Stage is yours.
Thank you. Thank you for the nice introduction. Good morning, everybody. Have you slept well last night? I am used to it. When I see somebody sleeping, I wake him up. Please, yeah, I am a little bit too direct in this matter. In Japan, when they start, even in the restaurant, they put the mobile phone in they call it manner phone, manner mode. We call it silent mode. And please, if you would put your mobile phone on manner code, that we, even in the restaurant, the people, they don't like to be disturbed. I am so happy to meet you here. I have been here 3 years ago, and I enjoyed meeting a lot of people here. It is a good opportunity to share with other experts, with the attendees, different ideas different views. As you see, my name is Mohammed Al-Mubarak. I'm a consultant physician. I studied medicine in Austria and Vienna, in the University of Vienna. I stayed there about 15 years and came back with my board in medicine. The auto industry in the US teaches us you can postpone quality, but you cannot postpone its consequences. There was an auto industry crisis in the United States in the 70s because of quality failure. This quality failure that time became visible 30 years later. The Japanese got it and started earlier in this business. So I'm happy to talk about whole system quality. It is a white paper of the IHI. It is not my product. And I found it is very important to share this with everybody. Maybe you don't have the time to read it. It's about, maybe if you will break it down in slides, it will be about more than 200 slides. I summarized that in about 20 slides to make it easier for everybody to understand it. Quality is very simple, but we are complicating that. Maybe we want to sell ourselves that we know better than the other. I don't know. There was an accrediting body came to the NASA. You know the NASA for the sending people to the moon. The accrediting body entered the NASA. When they entered, they saw a cleaner doing his job. You know, this is the simplicity of the quality. If you have a mission, the people should understand the mission, not to memorize that as any citation or what, and you don't want to miss any letters, any word. No, it is make it very simple for the people, even in your organization. They started with the cleaner, say a critical body like JCI, say. They ask him, why you are cleaning? Look, his answer means that he understood the mission of the organization. He said, I am cleaning to send people to the moon. He understood the mission of the organization. They want to send people to the moon. That's why we should make it easier for our people in any healthcare organization to understand the mission, to be in contact with them. We will be having an introduction, and we'll go through this very quick issues. Let me first open my pointer. This is a very complicated pointer. First of all, I want here also, after greeting you, to thank my wife who supported me and by attending this conference, and she's there. How are you? You all know all these great people Mr. Henry Ford, with the continuous assembly line. He invented the assembly line, Mr. Ford. You know, Ford car, 1913. By doing it, he means that the parts should come to the worker, not I have to go and bring the door, bring this part, and so they come to you, you fix it and come to the other. And instead of assembling, taking the assembly time take 12 hours, it took 90 minutes. And then with Mr. Deming, with his PDCA, actually he's not, he made it maybe famous, but the actual person who invented that is Mr. Schuhart, Walter Schuhart. He is the PDCA inventor. Mr. Deming, James Womack, Lean, joined by Clark, and then here we see World War II, and we see the also industry crisis, what we talk about it, Taishi Ono, you know, the Toyota quality production system. They have to thank him, the Japanese people, and many other things. This quality business, if you don't mind, I will take off my all these things to feel free. Halas, we took already pictures. Now we can feel free. This is the innovation adoption curve, which you know already. And we have always, not only for quality, for everything, for every new idea, we have innovators, early adopters, early majority, late majority, and laggards. This is only one, this is only printing system, printing mistake. Both are 16. Where do you think will we stand? I mean, now here, people in the Middle East. You see, innovator, of course, is Japanese, number one, Germany, and USA. I can consider USA as early adopters because really it took time till they really go in the business of quality. And then the early majority, you see, Saudi Arabia, Qatar, China, Malaysia, Turkey. And there is the late majority, 34%, and the laggards, Nigeria, Afghanistan. We have level of what we are talking about quality because all what will come in this conference also depends on quality. If we are scoping people, if we are treating people, if we are whatever you do, it should be considering a quality principle. There are levels of quality. First of all, because it was defined, the quality, what is quality is conformance to specification by Rosby. But conformance to specification, is it enough? If we have an area, maybe 4 times like this. And we can build the aeroplane, a small aeroplane, conforming to the details of specification. But can we use it? We cannot use an airplane in this area. We have to consider something else. Let us see there was need for adding to the definition. This is correct, but we have to add to it. Fitness for use. This was propagated by Joran. Everybody from you know Joran, I think. It should be usable. Otherwise, it is not a quality item. But again, it has specification, it is usable, but there was something they could not sell it. The next step is customer satisfaction. Because in all this, we did not ask the customer, what do you like? You see, quality in the past overlooks customer. And this is very huge mistake, actually. Okay, you can ask the customer, every customer has his needs. That we have stated needs and non-stated needs. Stated need, when I come and tell you I want a red car with automatic gear, with air condition, these are stated needs. But there are other unstated needs, which I take for granted. If you fulfill my stated needs, that means you ask me as a customer, and you give me this car. You give me the car without brake. I will not be satisfied. Those are unstated needs. You have to understand them. The customer will not tell you everything, every detail, but you have understanding from the unstated needs of the customer to satisfy him. Say you come to the tea shop and you order, please, I need a very strong tea. This was your stated needs. Strong tea. The gentleman comes back with the tea to you and give you the tea. The tea was too cold. You weren't expecting it would be too cold. We're not talking about iced tea. We're talking about no. You will not be satisfied. Those was unstated need. We have to understand it when we are dealing with our customers, whatever the customer is. Delighting the customer, making him happy, that means you also notice unstated needs. If we reach actually this stage, that means we are good in the industry. We can sell our product very good, and it is okay. But there was also other people having more imagination, and they want to come. They want not only satisfy the customer and delighting him, they want that he really, they want to educate. As you said, also with the diet, we have a problem. This is a behavior. They want, what is this? The highest level, like chanting the customer, like a magic, نسحر and how we can enchant the customer like a magic. We have to tell him what is good for him also. That means, but the company should have a really very, very huge marketing strategies for the product, to convince the people, to understand first the people population, what they need, and to make that item which is good for them, not only for the company. Like when he wants to buy, say, refrigerator. There is a refrigerator and there is another refrigerator, a little bit more expensive. It is environmental friendly. I have to educate him. This environmental friendly. It's worth it that you pay more for that. When we come this interaction with the customer, and also put yourself. This is also your patient. He should be involved with you in every step. It is not more the old paternalism. I am the doctor here and I am alone. No, I am the doctor here, I am the nurse here, I am the technician here, we are all together. Since the Institute of Medicine to Erase Human, there a lot of progress actually in the field of medicine, particularly in the SSI, surgical site infection, hospital-acquired conditions, and all these items. But there was also something, there is a space for more improvement, actually. We are not addressing, actually, as an example, the near misses. We are really not addressing them. Are they reported or not? Many people do near miss and they don't report it because of what? Because of fear. Fear is destroying. Even among your children. Don't let them fear you. Let them love you, respect you, talk to you. If they fear you, well, of course, my father come or my mother come, okay. They will hide everything. It is the same with our employee. If we understand, if they do mistake, okay, it's good. It's the opportunity to improve this mistake. It is not an opportunity to punish people. I know I have if I have time pressure, I have already prepared one slide for the whole presentation. If you know Peter Drucker, it is a Austrian management professor. He has another definition. We used to call it external and internal customer, but he framed it different, Mr. Peter Drucker. He said primary customers and secondary customers. Primary customers include healthcare force, patient, population, community members, etc. Secondary customers, payers, partners, regulatory bodies, and accreditation agencies. I like this actually definition instead of external and internal, because you can be primary, you are external and internal. See, this is and then there's again another thing. It is the equity in healthcare. All this white paper. There is something in health equity. This is the queen of the aims by Don Berwick. You know Don Berwick, the IHI CEO and Harvard University professor, and all this knowledge, and he was also advisor to the Clinton administration. Equity is the queen of the aim. You know the aims? We have the triple aims, quadruple aims, multiple aims. I hope that you still remember it. Equity, the most important. He said the queen of the aims. It's not a question about equality. We said we are equal. We are happy. No, we are not happy. There must be equity. This is equality. This is equity. You see? Equality, okay, you gave me the same steps as everybody else, but I cannot see. And equity, that means you help everybody to reach. Now in our situation, the healthcare institution, whatever it is, it's not enough to say we are equal. No, we want equity, and this is the problem overall in the whole world, not only in our area. And then came Peter Singhi, is MIT management lecturer. He is the author of the fifth discipline. Do you know what is the fifth discipline? The fifth discipline is system thinking. System thinker, they will be always successful. You know, the COVID-19 pandemic, we have to be thankful to the COVID-19 pandemic. We learned a lot of things. You see, these leaders empowered frontline staff to make timely decisions. Gave you the authority, empowering you, not giving you authority. Empowering you really, it's removing the obstacles in front of you that you do your job. This is empowering you. And the whole system quality practices, as you saw, quality planning involves identifying customer needs, defining and designing strategies to meet them. Whole system quality, it is the approach, all the white paper is integrating quality planning, quality control, and quality improvement. There is misunderstanding of quality control, actually. We'll come to it now. This is the leader's principle, build shared sense of purpose. Let the people feel with you why you are doing this. Then they will, if you convince them, you show them why we are doing this. Now they will understand, they will cooperate with you. If we are coming, we are changing the visiting hour. Why you are changing the visiting hour? Show them data. Show them proof for that. Not only that you are the boss and you're deciding everything. Let them talk to you and give you their input and practice fifth discipline, system thinking. You have to know the interconnected components of the system to achieve your goal. Engage collective learning. As Peter Singh said, continuous learning, everyday learning. Everyday learning, that means everyday improvement, like the Japanese do, Kaizen. Everyday little improvement. A sequence approach to quality planning, engage customer to identify and prioritize needs, develop a shared vision, establish. You'll tell me how I engage customers. I engage the patient and the family. You see, mechanism to involve engaging patient, families. LR includes survey. You can make survey, focus group, concept testing as well. You can generate approaches such as customer observation, journey mapping. All all these things, co-design, even if you want to design a place in the hospital, you have to engage the patient with you and the family. That's why you see experience-based co-design. No, maybe this is a philosophy. Are we doing this, or are they doing that in the other world? Only few places are doing that. These are letting the community participate with them. We come to the quality, after quality planning, the quality control. Quality control is often unfortunately misunderstood as a synonymous with quality assurance. Quality control is internal and quality assurance is external. Quality control is every minute. Quality assurance, it comes with time lag. It comes a regulator from the ministry or come an accrediting body, JCI or a CBI or whatever it is. This is not a quality. It is very different from quality control. You see, quality control activity by stakeholder, patient and families, even environment quality performance feedback through looks like whiteboards. Give the opportunity to talk to these people in the hospital, the visitors, the community, okay, whatever, the family. Now there is no more restricted visiting hour. It should be whole day visiting hour. This is what the family need. Clinician also crucial in making control, executing evidence-based protocols, unit level, and they monitor quality department. Everybody has his part in this total quality, whole system quality. Actually, I would like that you, and then you come to the quality improvement by also engaging the same people. Take the idea from them. What should we do? Where can we improve? And they will give you idea, and you see what is possible, what is not possible. That means I cannot do everything. Yes, I cannot do it because I have a video for 2 minutes. I cannot do everything what the customer says. This is not true, actually. Maybe you remember the slogan, "Customer's king." That means we have to do everything what the customer wants. That means the customer maybe his expectation is unrealistic. Say we have a classroom education in the school, and during we have an exam, and in the same time there is a football match important. If you will ask them what do you want, they will say they will watch the football match. If they are king, should I listen to them? No. In this case, I should not listen to them. I should educate them what is good for them. Please, can somebody help me that we play to wrap up all these things that I found that it is really very valuable, very valuable video to see. With respect to the lady who made that.
What quality is, what whole system quality. The whole system quality white paper seeks to define what quality is, what whole system quality is, and how an organization can pursue a set of management practices, a set of integrated activities around quality planning, quality control, and quality improvement. And what are the ways in which to cultivate a culture that's necessary to make the most of those management practices? Whole system quality doesn't necessarily mean pushing aside all of the work that's been done till date. But it does recognize that most organizations have a quality strategy, a financial strategy, and any number of strategies in order to advance it towards its strategic goals. Unfortunately, these are all dissipated, sometimes at odds with one another, approached in a piecemeal fashion. The goal of whole system quality is to integrate all of these different strategies and focus them around something simple: customer needs, and ensure that quality is at the center of the strategic efforts and that customers aren't lost in a lot of the improvement and various initiatives undertaken by an organization. As such, there are a set of critical strategic efforts that are necessary, from first engaging the frontline staff and the customers and trying to understand and appreciate where the gaps are in meeting needs, where the needs persist, in developing a shared vision and a set of strategic priorities based on what those early conversations surface, and creating a culture that is entirely centered around that. And so that's not to say that all of the work that's been done in the past needs to be pushed aside for this, but it is recentering and refocusing of the organization towards a focused set of quality goals that emerge from the customers and customer needs.
Now, I thank you very much for listening to me, but for the last, we have something new in this event. I want to ask you. Tomorrow, I have my second presentation. I have two presentations. You have to choose one of them. This is something new. Either employee engagement and well-being, this is one, or readiness for change. Which one you will vote for tomorrow? By raising the hand. Yeah, raising the hand. Well-being, yeah, all that I see. Well-being, all for one. Okay, then we have following. Then I noticed, actually, I was happy to meet with Dr. Wan, and she drew my attention to a very important presentation tomorrow about vaccination. I cannot tell the secrets, but it's really very important. Thank you very much. Thank you very much.
Thank you, Dr. Hamad, for this beautiful, really, speaking engagement that has captured us. We have questions for Dr. Any positions, any interventions, any questions?
As you don't have any question, I have a question.
Okay.
What is the most important to you in hospital management? يعني you have
Psychological
safety. This is very important. Psychological safety. Efficiency, yes, of course, but if you don't have psychological safety, you will be not efficient. You will not speak up. Everything will be hidden, and this is very devastating in an organization. Thank you for listening to me. See you tomorrow, inshallah.
Please, Dr. Hamad, we're sending this certificate to Dr. Hamad with the Barsha for this moment of the presentation.
Yes. Yes,
Thank you. Thank you. And now on to our next presentation. But before I go into the next presentation, I would like you to meet all our keynote speakers today, from Mrs. Tahani, Azraya, and please, if I don't mention your names well, please, I apologize for that. This is Tahani, where you'll be talking about microbial biotechnology from Kuwait. I will be presenting also tomorrow on actually the work on healthcare and burnout, emotional intelligence, and behavior modification and wellness and coaching in helping physician burnout and healthcare burnout. Dr. Muhammad Aweda from Barsha University for Medical and Health Sciences. Dr. Muayyid Ilyas, also polyclinic founder and consultant gastroenterologist from King's College Hospital in London, Jeddah. And Dr. Anna Cooper, founder of the online health platform Check from the USA. And now I would like you to help me welcome Professor Ali Gasanov from Russia. And his presentation title is going to be First Experience with Endoscopy Intraluminal Gastroplasty using the Apollo over stitch shaping device. Please help me in welcoming Professor Gasanov.
Thank you very much. This is a great presentation. Good morning, dear colleagues. My name is Ali Gasanov. Thank you for the opportunity to speak at this conference. I represent two centers in Russia. First, Emergency Medical Center in Moscow. Second, Jamsi Clinic Center in Makhachkala, Republic of Dagestan. Allow me to share with you our limited experience with endoscopic sleeve gastroplastic procedure in obese patient with a body mass index over 35. I would like to express special gratitude to our partner for their assistance and helping this patient and for the opportunity to present our result for your consideration. The relevance obesity today is beyond doubt, which leads to an increased mortality rate. In Russia, 25% of the population is obese. What are the reasons for such widespread of obesity today? Many factors. First, uncontrolled eating, fast food and junks. Second, physician inactivity. For example, today everything is ready to be delivered to your doorstep, just so we don't move. Third, artificial intelligence, which can also lead to decrease human mobility. So let's move on to obesity treatment methods. Everyone is familiar with surgical treatment, including gastric resection, gastric sleeve, binding, and bypass, and balloon include. Each of these methods has both advantage and disadvantage, which I won't dwell on. With the advent of the overstitch suturing system, it became possible to treat this patient with minimally invasive methods. I learned this technique from Professor Aid Al-Qahtani from Riyadh, Saudi Arabia. Special thanks to the professor and all his team for his warm welcome, skill, and experience. The ESG technique allows for a reduction in gastric volume by 2/3, which achieved a good result comparable to this for surgical resection. However, after ESG, we don't not experience the same serious complication as with surgery. Our experience consists 10 patients who underwent ESG. The procedure is shown in the slide. The procedure was performed using an Olympus dual channel endoscope and the overstitch system for a dual channel endoscope. Where necessary, using CO2 gas through the manipulation for the insufflation, and where necessary, and over tube always place. Every necessary device shown in the slide. This slide shows our first patient, weight 192 kilograms, with BMI of 48. A weight loss dynamic after EEG are shown. Patient parameters and surgery time are shown in the graphs. First time we spent this procedure maybe 3.5 hours, but now 30 or 40 minutes. On average, our patient lost 6 to 25% of their weight in the first 3 months and up to 65% in 6 months. An important aspect is in this procedure is complication. We observed complication in the form of pneumoperitoneum in one patient on the second day after procedure. But this did not require additional intervention. The patient received antibiotic therapy for 3 days with a positive effect. So you can control the element like tannus only by gathering all the stones. Similarly, good results can be achieved with a team consists of surgeon, endoscopist, endocrinologist, psychologist, and gastroenterologist doctor. In future, we will continue to perform this procedure and gain more experience. Thank you for your attention. If you have any question, I would like to answer to.
Well, I think I see very good result, excellent result. But I'm thinking about sweet eater. Is it successful to perform such surgery in sweet eater? Sweet eater is a piece of sweet that is a lot of calories. Sweet eaters can this type of operation successful in them who prefer to eat sweets like guava, silver cake?
Thank you, Professor Muhammad. If I studying in Saudi Arabia, we have little experience. So 10 patients. If I study in Saudi Arabia, Professor Al-Qahtani had 3,000 experience, 3,000 patients. He operation not only ESG, gastros, plastic, surgeon, and bypass, and therapy, endocrinologist therapy with preparation, and ESG procedure. all good result. But we had some little bit experience ESG starting in Russia 20-25 years.
Yeah, because always sweet people, it isn't so successful in sweet people. Because they don't need to fill their stomach. It's only small pieces of sweet and then they expand. This is the problem with sweet people.
Yes.
Thank you.
I agree with Dr. Muhammad. Also, from a nutritional point of view, these patients have reduced their BMI. However, the behavior modification regarding food is very important in terms of, okay, who is educating them? I see in this, I see an endocrinologist, a gastroenterologist, an endoscopist, and a medical psychologist. However, where is dietitian involved here? Because the quality of food and the bioavailability of the food, when you do the procedure, it decreases a lot, doesn't it? And so they are going to have a lot of nutritional deficiencies. So would you account for that from a dietetics point of view? And as Dr. Khan is saying, is if we have sweet eaters, that is empty calories. That's it, with no nutritional value.
It's good. This is a very difficult question. We working this patient on the team. After ESG procedure, our procedure, patient treat gastroenterologist and endocrinologist, and give to diet, so strong diet and control.
It's a liquid diet usually for 12 weeks. Yes? Usually it's a liquid diet, isn't it?
Чаще всего с помощью жидкости, да, питаются, то есть преимущество.
Yes, the first time only liquid, and next time a little bit control. Yes. First time, 1 week after procedure, only liquid. Little bit liquid, but after normal eat, but diet.
Yeah. Thank you so much. Any more questions?
Is the procedure reversible or irreversible?
Reversible?
Reversible. Reversible or irreversible when they do the procedure.
Yes. If we're studying in Saudi Arabia,
Обратимо или нет?
we come to young girl after ESG procedure 2 years. We're cutting this tissue and no problem, gastric normal. 2 years ago.
Any more questions for doctor? Yes.
Just only a thought I would want to set from the nutrition perspective. We maybe look at not changing the BMI and more looking at the muscle mass and fat mass in the body, position of the body itself. Because according to the guidelines, we're not looking at the BMI, we're not looking at what quality of food you're eating and the status of the muscle. Again, muscle mass is the topic of the year. So maybe just changing what a successful surgery would look like instead of talking about they lost, let's say, 60, 80 kilos, maybe they improved the mass quality and the amount of muscle mass in his body and he lost the 70 kilos or maybe 10 kilos, and this is the more successful.
Yeah, that's absolutely true. Like I was saying.
You mean muscle or fat?
Yeah, as you know, looking at the results, I see here that before he was 130 kilos and then he's 80 kilos. But did he lose? Most probably he lost like, let's say between 10 to 15%, so he lost his muscle mass because you're not I don't know if you did any follow-ups,
She's talking about body composition analysis. The body composition analysis.
No, now our clinic, not this analysis. Maybe future years.
They are still using the BMI. Unfortunately, although body composition analysis using bioelectrical inclusion anthropometry is very important, but it seems that they are still using
Yes.
the BMI. Thank you so
Thank
much.
you, thank you.
Thank you, Doctor.
Thank you.
And now, moving on with another exciting session, we have Dr. Zahra.
Yes, I'm here for
Dr. Zahra Khalifa from Dubai Medical University, and her presentation title is Artificial Intelligence Across the Drug Discovery Pipeline, from Molecule to Market. Please welcome Dr. Khalifa.
Thank you. This is my pleasure. I like to remove this one. Yeah, so very good morning, everyone, and thank you for being here today. So I'll be highlighting the use of artificial intelligence in the drug discovery and development process. So, of course, we all know that the drug discovery and development journey is a very long one. It's not easy. It requires a lot of research work for so many years from multiple disciplines, so we can come up with a discovery of a new drug or a new molecule. It requires a lot of failures, disappointments, frustration, funds, manpower, so it's not an easy one. So I'll be highlighting today how artificial intelligence can convert this journey to at least a more pleasant one. We will be looking at the whole journey of drug discovery and development process, and we will see how AI can be integrated in each of those stages in the process, starting from the identification of the target genes and proteins associated with the disease, and then how we can find the ligands that can bind well to those targets, and then even like predicting their efficacy, toxicity, generating even better molecules, predicting their admit properties, and then formulating those molecules into good formulations and the process optimization during the production of those products. Even the integration of AI in modeling the in vitro and in vivo release kinetics, and even in the final stages of clinical trials. Before I go through this journey, I will just brief you about the basic concepts of artificial intelligence, which can be used in the drug discovery and development process. So why do we call it artificial intelligence? Why don't we just say intelligence? Yeah, exactly, because intelligence, it's our natural gift as humans. We are humans, we have got this, natural gift of intelligence. So when we are trying to make machines act or behave or function in a way or in another, like humans or an intelligent way, we just call them artificial. And artificial intelligence falls under two main categories, and we always hear those terms, machine learning and deep learning. To make this machine learning concept simple to understand, I will give a small example to clarify the difference between the traditional programming and the machine learning. So when I am designing a program, let's say I want to design a program to detect spam emails. So I have to give specific set of instructions. If you find the word free money, then detect it as a spam. If this is not available, detect it as safe, and so ever. So there are steps that the program will follow. In machine learning, we don't do this. We make our model learn. I will give the model a lot of spam emails. I will show him these are the spam emails and these are the safe emails. So the model will start to find the patterns. The model will start to look, okay, so the spam emails, normally they talk about free money, they talk about links, they come from a specific domain, whatever, while the safe emails, they don't have that. So the model starts to find the patterns. And now when I give the model a new email, unknown, he knows the patterns. He will look for the patterns and decides that this is a spam, this is a safe one. And I'll be using the terms he, she, him, her. I just feel that I don't feel it smooth to say it when I talk about artificial intelligence. I feel they're smarter than calling them as it. And ideally they are it's, but I don't feel this, I don't feel comfortable. So, and because they are not gender specific, so I'll just be referring, so don't bother if I say him or her or whatever. Now, these machine learning, they come, or there are different methods for machine learning: supervised, unsupervised, and reinforcement learning. In supervised learning, we have labeled data. So I tell the model, for example, this is an orange, this is an apple, this is an apple. He will learn, and then when I give him one of those fruits, he will say, okay, so this is an orange. Unsupervised learning, we don't give labeled data, so I just give him a whole bunch of mixed oranges and apples, and the model will start to look what are the features in each and classify them into groups. So put all the oranges in one group, all the apples in one group, maybe if there is a third fruit or something. Reinforcement learning, we normally refer to it like potty training. So there is an action, an interaction with the environment. So the model does something, and then we tell him that this is right, you did that progress. So he learns that this is the right thing to do. Or he does something wrong, so we tell him, no, this step was wrong. So with time, gradually, he learns what's the right thing to do and what's the wrong thing that he should be avoiding. Deep learning is a more complex model. It can work with more complex and raw data and find more complex patterns. It is containing artificial neural networks, which is a layer of units connected in a certain structure, which is actually, it is inspired by the structure of the human brain. That's why we refer to them as artificial neural networks. Now, why do we use AI in drug discovery? Definitely, the process will be faster. We can screen so many compounds in a short time. We can reduce cost and effort and manpower. And when we have a lot of data or thousands of samples, I will not say that AI can detect patterns that humans cannot, but at least they can do this in a faster and a smarter way, unfortunately. Now, in order to use AI, there are some important tools that we need to have. Now, if I want to work in the drug discovery process and I want to implement AI. So the first thing, I need to have a programming language, and Python is the most widely used, freely available, can be accessed easily, and also supported by many libraries. And libraries here, they are pre-written codes that are readily available to perform certain functions. So for example, I will import the pandas library, which is the library I can use to read data sets, or NumPy to make mathematical operations, or scikit-learn to train models. So already there are codes available that I can apply to whatever functions I want to do. And most importantly, data sets. I need data, a lot of data. Without data, the AI cannot function. So if I don't train my models using data, and the bigger the size, the more realistic and the cleaner the data, the more accurate predictions I will get. And if my data is not good, the model will not function well. They don't have any knowledge other than what the data I use to train them. So now we will go back to the drug discovery and development stages. And I'll try to, each one of those, it's a whole topic by itself, but I will try to highlight the main role of AI in each of those stages. So initially, in order to identify the targets that we want to work on, so if I have a disease of interest and I want to start doing a research on this disease, for example, let's say we want to find what are the targets to work on in patients who are having colorectal cancer, for example. So the genomic analysis can be done there are two types of genomic analysis. Mostly in the target identification, we are interested in the gene expression data. And here we are trying to check the expression level of the different genes in the body. When there are genes that are overexpressed, it means they are producing too much mRNA. It means something wrong. So what we're basically doing here, we are comparing the genes of a healthy individual with the genes of individuals having this disease, for example, the colorectal cancer patients. And we will notice that there are some genes that will be overexpressed in those patients. So most likely, those are the genes that are involved in the disease. And of course, with AI, this can be done in a very fast way. You can, of course, you're not going to depend on 5 or 10 patients. You are going to analyze data sets which are having like thousands of patients, and you find the average expression, and we can know that those are the main genes that are overexpressed in this disease. And they can be ranked, like according to the number of folds of expression. Of course, the higher they are, the more associated. Not only the expression level, but we can use also string network. String network, they like built for me network about how the interaction pathways of those, how the genes are involved in interaction pathways. And I can identify the hub genes, which are central to many of the interaction pathways. And we can use Open Target. This is a website available that I can select my targets, my candidate targets, and get to know more which targets are druggable, which targets have been previously researched for ligands, which targets that already have ligands available in the market, so I can put my priority, which targets I will select to work on. After selecting the targets, we need to find ligands that can actually bind to those targets. This is the second stage of the drug discovery process. So normally, this we call it virtual screening. I want to find ligands that can bind to the targets. Currently, it's being done by computational methods, and it can be ligand-based or structure-based. For the ligand-based, it's when I don't have enough information about the structure of the target. So for example, I want to work on target X. I don't know the exact structure of target X, but I know that compound A can work on target X. From previous experimental data, from previous research, I know that ligand A, compound A, works well or binds well with target X. And I know that compound B is very similar in structure to compound A, so most probably compound B also will bind well to the target X. This is the ligand-based concept. Structure-based is when I know exactly the structure of the target and the structure of the ligand, and this is the concept on which molecular docking is based. So it's just like the key and lock theory. How well will this ligand fit into this target? And of course, using AI, this can be done by binding data. There are data sets, publicly available data sets, like the binding DB, for example. I just have to select what is my target of interest, and I'll find a lot of experimental data. It doesn't have to be my ligand, it doesn't have to be my molecule, but there are so many molecules that have been tested on this target before, and the AI model will be able to learn the patterns. And when I give a new molecule, the AI can predict for me that this molecule is more likely to bind or not. So this is how AI works with the binding. Of course, it's not necessary that if a molecule binds well to a target, it is going to be very effective and to show high efficacy. It's not a must. Maybe it will bind well, but still the efficacy will not be so high. So we also need to predict the efficacy. We need data about the molecule, about the protein target, and we can design models using efficacy data that can tell me how likely my molecule or my new molecules will show efficacy. And the output here can be in one of different models. Most probably we are using regression or classification models. The difference here, regression model, I'm going to get a continuous value. So the model will tell me that you have an IC50 of 7 or 8 or 10. It's a value on a continuous scale to tell me how efficient my molecule will be. But when I'm talking about classification, when I'm training a classification model, it will just tell me yes or no. Will it show activity or not show activity? This will all depend on the model I will train, the data I will provide, and the expected output that I'm looking for. Similarly, we need also to check the toxicity of the molecules, and one of the commonly available data sets for toxicity, it's the TOX21. And here I have to specify exactly my toxicity endpoint. So if you look for the TOX21, if you search for it, you will find that you have to select your toxicity endpoints. Are you interested in checking the hepatotoxicity or renal toxicity, cardiotoxicity, or whatever? So you need to be selecting the endpoints. Now, after we have good molecules, okay, we have a good molecule, a good candidate that can work on my targets that I have selected, we can also generate new molecules that are better molecules. And those are like traditional approaches that are used in the de novo design of molecules. Mostly in AI, we are interested in the rule-based system. I have a structure, I have a molecule, and now I want to make it better. How to make it better? By using chemical reactions and chemical rules on how to make this, to improve this structure in a better way. So there are different methods for AI-driven molecular generation. I don't want to go into so much details because I know I have very limited time, but like, for example, we've got the variational autoencoder. How AI can support this variational autoencoder? You have the structure of the molecule. It will be encoded to a latent vector, which is a virtual latent space. So you get the molecule represented as latent vector, and you can make minor changes in those vectors, decode it back to the structure. Those changes will be reflected on the structure. Very minor changes can show reflections in the structure and can generate better molecules. Another interesting example is the generative adversarial network. And here you are designing two networks, one a generator and one a discriminator, and they play together, they compete with each other. The generator tries to generate new molecules, and the discriminator tries to check which are the real and which are the fake molecules. So with time, the generator tries to produce better and better molecules that the discriminator cannot detect, and the discriminator tries to work better in detecting which are the real molecules. So we end up with very good molecules. Now also here the structure activity relationship optimization. As I am generating new molecules and making changes in the structures, also I can integrate chat or line tools in order for the AI to explain for me what was the influence or the impact of those changes in the structure that I have done. So, for example, you will get different chat values, and you can also get very nice waterfall plots. I really wish if I can present for you some of the real graphs that we are getting from the Python, but I know I have very limited number of slides I can use. But you will get, for example, it will tell you like the changes in the aromatic ring. As you add more aromatic rings, what happened to the, how did this affect the activity of the structure? So it's a plus 15%. On the graph, you will find like a blue bar, which is towards the positive side, or maybe the nitro groups. As you add more nitro groups, you get a red bar towards the negative side. So you know which changes in the structure that actually, how did they influence the activity, whether positive or negative impact. Also, we can, once we generate those molecules, we can predict their admit properties. Input data like the molecular weights, polar surface area, H bond donors and acceptors, and the predicted properties will include, for example, what's the probability of absorption if I want to predict absorption property, what's the predicted volume of distribution, blood-brain barrier permeability, all those distribution parameters, metabolic stability if I want to predict the metabolism activity, and predicted excretion route, for example, and so on. And by the end of this stage, we can find the candidate molecule. But of course, having the candidate molecule is not enough. Definitely, we need to formulate this molecule into a drug product that can be administered safely, effectively, and practically to the patient. And of course, here comes the art of formulation. And normally in formulation development, we are using the trial and error approach. So we just design so many formulations. We go to the lab and we prepare them all. We characterize, we evaluate, and then we exclude all those having properties that we are not happy with, and we end up with 1 or 2 candidate formulas. But instead of doing all of this process, AI can make it smarter. We can, from the beginning, exclude 10 of those formulations and just keep the candidate ones that we can actually experiment in the lab. In the formulation development, AI can support the excipient selection. So AI can predict for me what are the suitable excipients according to the structure of this and properties of this API. It can also predict for me what's the expected disintegration, dissolution, different properties, and it can help me in the selection of the formulation ratios. All this can be predicted for me using the supervised learning, I'm running out of time. Yeah. So using different AI models as well. We can use it in the process simulation to adjust the different parameters. I'll not go into details. I can predict drug release properties. I can predict even in vivo parameters. And for clinical trials that are risky and they are very costly, it can help me to select the patients, not to favor patients, of course, but to select the right group of patients, maybe to predict the endpoint of the trial, and maybe at some time in the middle, I can adjust my clinical trial protocol instead of ending up with a failure. Pharmacogenomics and personalized medicine by the genomic analysis as well. Just 1 minute, I'll just summarize the challenges that are faced, because I think I was very optimistic talking and showing that it's just a piece of cake, and it's not. There are some challenges, mainly with the data, or when I want to apply my model to realistic data or actual populations other than my data. I know I ran out of time. I'm very sorry. So I'm very happy to be here, and of course, I'll be happy if you get in touch anytime. Yes. I had a lot of information to go through, but I know that I was very limited.
You have beautiful information. So much respect for everyone's time. And I'm sure there's a lot of questions for you. So go ahead.
Thank you. That was a very beautiful talk. I'm kind of like the biotech founder in the UK. And my question is straight to you.
Yes.
Is your model ready? For example, I pick 2 popular drugs and put them on a combination pill, and I bring it to you. Can you do all these end results that are?
No, actually, I'm not working on a ready model. I'm an academician, so I'm working in an academic institution. So what we're doing, we are just designing models and training them for minor research activities. We don't have a model which is ready for a whole thing. But I don't know, maybe we can contact, we can communicate. If there is a model that we can design for your work, maybe we can work on something like that.
What you do, can you send it to for example, Charlie?
Yeah, definitely. Yeah, I know how to manage. Thanks.
Other questions?
Thank you.
Thank you.
Always you have a problem with the bias.
Yes, definitely.
Okay, if we conquer this bias, it is difficult, but the artificial intelligence, is a pipeline of antibiotics.
Yes.
We have only a few antibiotics.
Yes.
Because the investor is not interested, because it takes about 20 years till they get
Yes.
from the antibiotics. Not everybody is using that.
Yes.
Does this AI procedure reduce the cost for making research and developing the new drug?
Yes, it will definitely reduce, but of course, the regulatory challenge is very there. I didn't get enough time to talk about the responsible attitudes when using AI. We have to be very transparent, like how the model was trained, what is the data that we use. We have to justify this in order to get regulatory acceptance for that. So we have also to set on accountable outcomes. Like, for example, if this model ends up not working well, who is taking the responsibility? Is it the developer or the user himself? So everything should be justified, should be explained, and most importantly, the human oversight. Always, this AI is there to support humans. We cannot just let the AI lead. So always a human should be checking, should be ensuring that everything is happening in the right way. But definitely it is expected to help, to support humans, and it is expected to reduce the cost, but still regulatory, it's a challenge. I'll be there anyways, and I'll be happy to connect with everyone.
A lot of information for us, so thank you so much.
Thank you.
And next, before our break, we have our speaker that is Miss Imogen Husband. And hi, Imogen. She's a junior doctor from Birmingham, and her presentation is audit of perioperative care compliance with NCPOT guidelines in pediatric patients undergoing surgery for Crohn's disease at our institution. Welcome, Ms. London.
Hello. Hi, my name's Imogen. I'm one of the junior doctors training in surgery in England. And last year I was working at the Children's Hospital based in Bristol, where I did this audit, which was to look into our compliance with NCPOD guidelines, which I'll talk about in a bit more detail, about the compliance with our perioperative care for patients undergoing surgery for Crohn's disease. So I'll talk a bit about NCPOD and who they are, talk through our methods, the guidelines that they suggested we should be compliant with, our results, and then our next steps. So NCPOD stands for National Confidential Inquiry into Patient Outcomes and Death, which is not, doesn't really roll off the tongue, that's why we call it NCPOD. It was established in 1988, and it's a UK-wide organisation that released guidelines that should be followed by institutions providing surgical care. And it was created in response to some perioperative deaths, and there was investigations done into these, and they recognise basically that surgery is obviously a high risk time, both from surgery and anaesthetic, and so they release guidelines in compliance with this. And it's mandatory to follow their guidelines. So what they do is they don't introduce new treatments or anything. They look at the services that are provided in institutions undertaking surgery, and they discover the standard that is achieved, and they try to provide ways in which the standard could be improved. So the standards that they released that are relevant to my presentation are relating to patients undergoing surgery for Crohn's disease. So the guidelines that they created were targeted at patients over 16, and at the time I was working at the Children's Hospital, but we felt that these guidelines are still relevant to children with Crohn's disease because in all likelihood they will survive into their adult years, and it's important to provide good care for children as well as for adults. And so we audited our compliance. So I'll talk in a bit of detail about the specifics of each of the parts of the guidelines. This is an infographic that was produced alongside their recommendations. And they're, broadly speaking, the guidelines relate to the necessity to provide holistic care for patients with Crohn's disease. We should be optimising their medication at all stages of the pathway. We should consider surgery early as a potential treatment option rather than as a failure of medical treatment. We should perform surgery promptly once we've decided that surgery is necessary, and we should make sure that our communication in terms of handover to post-operative care providers is necessary. So in terms of methods, so we took a 5-year period from the beginning of 2020 to the end of 2024, and we audited every patient who had been admitted under the surgical team that had a diagnosis of Crohn's or inflammatory bowel disease. We included anybody who had resectional surgery, and we included anyone who had an elective or an emergency surgery admission. We excluded anybody with ulcerative colitis because the guidelines did not relate to patients with ulcerative colitis. And we excluded anyone who had minor surgery for Crohn's disease, so non-resectional. And we used a handy audit tool that is produced alongside the guidelines to audit our compliance. So in terms of our results, so the audit tool uses multiple questions to provide a kind of percentage of compliance with the recommendations that they suggest. So you can see they produce this infographic, and the guidelines are around the side, 1, 2, 3, 4 around the side, and the percentage compliance is kind of shown with the spider diagram, and you can see that we've got some variable compliance. Some areas are quite good and some areas not so good. And the target is 100% in each area. In terms of our patient population, there were 25 patients who had had resectional surgery for Crohn's disease in a 5-year period. So it's not a very common thing. And this is the only centre in the region in the southwest of England that does these surgeries. So it's not very common for children to need these operations. There were slightly more male than female patients in our population, and a third of the patients underwent emergency surgery. And the youngest patient was 10 years old. So I'll briefly go through our results. So in terms of access to holistic care, so this relates mostly to kind of other specialties within the hospital other than medics and surgeons. So access to pharmacy, access to dietitians, stone nurses, and the pain team were what was audited as part of this guideline. And also it was important to make sure that patients had access to psychological support. In these guidelines, which are created for adults, they also suggest support for wider health issues such as fertility and smoking cessation. And in our presentation to the gastroenterology team, we were criticised that these were not relevant, but actually I would argue that they are relevant, and we should be providing preventative measures to stop people from smoking before they even start, and we should be thinking about fertility early on. The guidelines also suggest we should be providing written information for patients, and we, at the time of auditing, we were not doing that. In terms of medication optimization, this was another area we scored quite well, but not 100%. Most patients did undergo a preoperative assessment with a gastroenterologist who considered things like biologics and steroids preoperatively. MDT timing was something that we thought a lot about. So there's currently no MDT at this institution that involves the surgical team discussing inflammatory bowel disease patients. And while all patients that went into surgery had a discussion between the surgeons and the gastroenterologists, most of them had not been discussed at a formal MDT with other members of the wider health team. So this was something that we focused on. Of the patients who did have an MDT discussion, we were quite good at documenting. And this is just to show that a third of our patients had emergency surgery, so this was not really applicable because it's not possible to have an MDT discussion prior. The audit tool didn't have a parameter for the appropriate referral for surgical consideration, but it was mentioned in the original audit that NCPOD produced, and I think it's relevant to talk about because in the original audit they suggested that nearly 20% of patients could have been referred earlier for surgery. And one of the points that the audit guidelines make is that we shouldn't be considering the need for surgery a failure of treatment. We should be considering it as a treatment option. Audit number 6. So this was about the preoperative anaesthetic consultant clinic. And in the paediatric patient population, most of the preoperative assessment was done with a questionnaire that was filled in either by the patient or by the parents. And they're mostly not seen preoperatively face-to-face, which is out of compliance with the guidelines. I have spoken to the anaesthetists about this, and they do suggest that in the paediatric population, the preoperative face-to-face clinic is probably not always necessary, and the questionnaire was probably fulfilling the requirements. We should be performing abdominal surgery within a month of deciding to operate. And we were quite good at doing this. Though most of the time when this didn't happen, it was because patients had GCSEs or A-levels. And in line with this, another parameter that wasn't assessed as part of their audit tool produced is that we should investigate any serious incidents, and specifically they're relating to if patients have been listed for elective surgery. If they come in while they're waiting and they have to have emergency surgery, then that's a serious incident, and we should take that into account. And the final parameter was that we should be handing over well to our colleagues who continue to take care of patients after they've had surgery, and we work quite good at this. So to summarise, we have quite good involvement of team members, so pharmacists, dietitians, stoma nurses, and the pain team in the perioperative period. We can make improvements with compliance in terms of anaesthetic assessments, and we also should try and make sure that the surgeons can attend the regular gastroenterology meeting so that patients with inflammatory bowel disease can be discussed and surgery can be considered early rather than as a failure of medical management. And we should also be providing some written information in terms of a patient passport. So in terms of the changes that we've made, so we've made this patient information leaflet, which is now going to be distributed to patients who are seen in the joint gastrosurgical clinic. And there's also a website called the Transition website, which is part of the Bristol Children's Hospital website. And the aim of this website is to help children transition well into adult care. And so we're currently in discussions with the gastroenterology team and the Transition website to try and optimise our inflammatory bowel disease area of this website. We really would love to prioritise a regular multidisciplinary team discussion that the surgeons can take part in to ensure that patients are referred early for surgery if necessary. In order to do this, the inflammatory bowel disease surgeons need to rethink their job plan. So this is quite a slow process, but it is now in the process. And we're also reviewing our preoperative anaesthetic assessments. And the plan after we've made these interventions will be to re-audit. Since producing the patient information leaflet, actually no further patients have needed surgery for inflammatory bowel disease. As you can see from only 25 patients in 5 years, it's not very common to need this. So the plan is to re-audit in 12 months and see how our interventions have made a change. Thank you very much. Any questions?
Thank you, Imogen. Any questions for Imogen?
Yeah. Should I use microphone or something?
So we can all hear you.
First of all, thank you so much. Fantastic report. Can I ask you, like, it's not connected with this article, but do you use like advanced endoscopy technologies to assess remission of chronic disease, for example, like endoscopy?
I would say as part of the surgical team, it's not so much.
Yeah.
I know that's normally done by gastroenterology, but I think that as far as I can see from my kind of limited experience, I think endoscopy is one of the ways of assessing Crohn's, but also small bowel MRI seems to be one of the ways as well to assess for disease activity, as well as capsule endoscopy, yes. Yeah.
Thank you.
Thank you very much, Imogen, for that beautiful. My question is, what makes surgery in Crohn's emergency? Did they, as a child, in the education group, did they develop acute abdomen?
Yes, yeah, mostly acute abdomen perforation. Normally in the patients that I read their notes for this audit, it was mostly they came in and they were very unwell and there was a trial of medical management so that during the time in hospital, mostly it wasn't that they came in and went straight into surgery, but they had a trial of medical management with steroids and antibiotics and then failed to kind of progress on conservative treatment and then needed to have surgery after that, rather than needing to come straight in, go straight for an operation.
As a second question, medical treatment, do you think one day that there may be a role for probiotics? Because I was at a conference 2 years ago, and I think there's this UK biotech that was trying to develop something for Crohn's. We were thinking that gut microbiome might be lateral with this. Do you think in the future it's going to happen?
Absolutely. I mean, I think it's a very interesting area of research that we're learning more and more about over recent years. And absolutely, I think it will be important in terms of both perioperative and kind of for medical management of Crohn's, definitely. Yeah. Thank you for your questions.
Also, any more questions for Imogen? All right, let's thank her. Please. I would like us to take a photo with the certificate holders first, and then we'll have to wait till 10:45, and then we'll be sharp at 10:45. I think we're way over before break after this very intense morning of loading our brains with beautiful information. But I'm gonna ask the participants please to come and so we can take a quick look. All participants.
Oh, really?
Yeah.
You want me to come for it already?
Yeah, like 3 years ago, but different.
Let's have the taller people on stage and some of us here. All of you, please, come on. We're gonna take it to the next one.
Wait, this is.
Only if it's personal.
Yeah, I know the agents.
We need people on stage, please, if people have someone who say, so we can do the layers.
Fantastic.
Thank you. Thank you all and see you at 10:45. Okay?
Attenzione a scendere.
Yeah, that's it.
Наши доклады поздно, все проблема.
Да,
Trying to be like a coffee.
Come back.
Hello everyone, welcome back. Welcome back everyone. Our next speaker, is she here? Sarina is here. Our next speaker is Ms. Sarina Muhammad Rashid from King Abdulaziz University Hospital. And her presentation title is Effectiveness of Nurse-Led Cardiac Rehabilitation Program in KAUH Coronary Care Unit. I guess we're gonna know what KAUH is. Please welcome Ms. Rashid.
Assalamu alaikum. A very good morning to all. MashaAllah, the weather here is more cooler than in Saudi Arabia. I'm really happy to feel that. Thank you, Ms. Catherine, for introducing me. Myself, Sabina Rashid, working as a nursing area manager for coronary care unit at King Abdulaziz University Hospital, Jeddah, Saudi Arabia. So today we are going to discuss about our nurse-led cardiac rehabilitation program success story through KAUH. As we know, CVD is a significant public health problem, same like in other parts of the world, in Middle East also. The studies shows in Kingdom of Saudi Arabia, the national data, the prevalence of self-reported CVD among those over the age of 50 is 1.6%, and it increases significantly with age. Goes with Al-Hijali 2023. Same time, when we talk about readmissions, because we are talking about readmissions and cardiac rehabilitation, we focus, it is considered widely as an indicator for the quality of care within our hospitals. So cardiac rehabilitation is a comprehensive multidisciplinary program that incorporates evaluation, medical evaluation, exercise training, risk factor adjustment, education, psychosocial support, and long-term follow-up. It's a key component of secondary prevention of cardiac diseases. So if you see, the researcher repeatedly demonstrate the cardiac rehabilitation program increases the patient quality of life, decrease recurrent episodes, and improve the functional capacities. So why we start this project or the study? In 2018, we had a high number of readmission rate in our coronary care unit. So when we do the root cause analysis, we found most of these patients coming back with another episode due to either they stop the medication or they are not following the diet or exercise, which make them again another attack of heart disease. So from there, we started the multidisciplinary team project, which is aligned with the British Association for Cardiovascular Prevention and Rehabilitation, which enhance patient adherence and reduce avoidable readmissions. So we have assigned a cardiac rehabilitation nurse. Our cardiac rehabilitation nurse is a nurse specialist, senior nurse specialist, who has given training in the field of cardiac rehabilitation. Also, we develop educational materials for 5 different languages, which is very common to our hospital, which is Arabic, English, Hindi, Urdu, and Bengali. Also, we provide staff education to make the staff more aware what is their importance and role in preventing the patient readmissions. Also, we have done the implementation of the program and monitor the program. So these are educational material when we are teaching the patients. So we ask them to sign as they have received this education, just to confirm that we are not missing any patients during the episode. This is an evidence of our educational materials, which is in 5 different languages. which is very helpful for the patients who can take them home and read again about it. How to manage the heart disease, how to manage their exercise after discharge, how to manage the diet, everything is well explained in this one. So role of cardiac rehabilitation nurse. So what she do generally, she is the one making sure the patients are eligible for this program. So she do the initial assessment for assessing all the patients who is eligible for cardiac rehabilitation enrollment. So conduct comprehensive assessment daily and review the plan of care with the other multidisciplinary teams. And then she developed a personalized cardiac rehabilitation plan for each individual. She coordinates among multidisciplinary team. For example, she discussed with the physician, she discussed with the dietitian, she discussed with the physiotherapist, and so on. And she also supervises the early mobilization and exercise for the patients, educate each patient on daily activities and lifestyle modification they need to do according to their status. Also, she provides emotional support and she do phone follow-ups, discharge to monitor complaints and identify any red flags after discharge. And she documents her activities in the mission medical record, and we see the improvement and evaluate accordingly. So our study objective from the beginning was to reduce the cardiac rehabilitation program as a method to reduce our readmission rate. Okay, as I said, it was a very high rate, so we want to decrease it by at least 50% was our initial objective. So the methodology we have done it is in the settings, cardiac center at KUH did the research design was retrospective descriptive study design, and the population we took all eligible patients which are admitted in our coronary care unit, and the sample size was 4,300 patients. And our objective was to decrease the 30-day readmission rate and improve multidisciplinary team participation in this program. So what are the inclusion criteria? Here you can see we have selected all coronary heart disease patients, which is in two types, acute coronary syndrome, which maybe some of them go for post-PCI, angioplasty. Hope everybody understands the terminology here. And some may be for multi-vessel, either for post-CABG or for CABG. Some patient may be discharged with medication management. This is one category. The next one was a heart failure. They were on post-implantation of cardiac devices or also on medication management was chosen. Also, we had patients post-heart valve repair or replacement, atrial fibrillation, and chest pain with normal coronary sometimes also was included in this criteria. The exclusion criteria, we excluded patients on arrhythmia for ablation, congenital anomalies, and patient with missing data. So around 383 patients were excluded from this study since due to these reasons. We had got ethical consideration from KAU Research Ethics Committee, and this is a reference number. So our expected reduce 30-day readmission rate, improve patient education through effective multidisciplinary team involvement, better adherence and self-management post-discharge, support of scaling nurse-led CR programs. So due to this high effectiveness of CR program, we could see the readmission rate become very less. Actually, the program when we started, we did not think we could get this much effect on this only the nurse-led program. So as I discussed, the methodology, it is a retrospective descriptive study, and all patients met eligibility criteria was included. And the samples were the readmission rate, patient categories, and multidisciplinary team documentations were collected. And we did analysis to descriptive statistics, percentage, and graphical summaries. Also, independent t-test and one-way ANOVA was used. So in the data analysis, the data was presented by numbers and percentage for all categorical variables. The average number of patients was compared with demographic characteristics using t-test and ANOVA, and statistical significance was set at P0.05 level, and it was analyzed by SPSS version 26. So the results are, as we could see, the 2018, our rate was 16.3. So we started the program by 2018 end, by 19 we already started it. So we could reduce in 19 by 10.3, then in 28.3, 21 like that. And alhamdulillah, so far until now, we could sustain the achievement. So the readmission rates were calculated, total number of discharged patients by number of patients readmitted within 30 days. So these patients are within the inclusion criteria. Also about the patient education by multidisciplinary team. In the beginning, we had difficulty because not all the multidisciplinary team was cooperative or collaborating with us. So we could do special education sessions for them, and it became 100% for all the multidisciplinary team, like CRN is the cardiac rehabilitation nurse, the physicians were involved, the nurses, dietitian, etc. was involved. So this is the result analysis we have done. This is for all the eligible patients we admitted in coronary care unit for the year 2019 to 2024. It is almost 6 years data, and the result shows. Actually, for the age group, we could see there is a significant difference in the age from 55 to 64, which was tested by ANOVA just to make sure it is not by chance. Also for the male and female, as usually, and we could see any cardiac diseases. If you go, we could see most of the cases was occurred in male patients, which also shows in our study also. When it comes to the Saudi or non-Saudi patients, which is need further study actually, because our area is an area which is our hospital is the only one tertiary care hospital we could access very easily. It could be one of the reasons we have a high rate of non-Saudi patients, but we are in the plan to do further analysis to see how and what may be the reason for this one. But in this study, we did not focus on this area. When it comes to post-PCI and post-CABG, it is also worldwide or internationally, we could see most of the patients admitted with coronary syndromes are going for PCI rather than post-CABG. So our studies also shows with the international studies currently available. And the male and female, most of the studies, it shows the male patients are more prone to have heart diseases, which shows in our study also. So in conclusion, nurse-led cardiac rehabilitation program enhances outcome for cardiac patients, strengthen multidisciplinary care collaboration, support adoption of CR protocol across institutions, add evidence to global CR literature. So what is our recommendation here? To expand the service to the outpatient service. Currently, we are doing only inpatient services. So now we have planned to move to outpatient services and to other area of the hospital. Now we are focusing on coronary care unit, as I said. Also do further studies on the effect of CR on patient physiological parameters. As we know, the physiological parameters are really affected by cardiac rehabilitation program, but in this study, we did not include it. So our next plan to measure the functional capacity, patient satisfaction, and quality of life, etc. Also to conduct multi-center studies on national and international levels if possible. So this is a recommendation in addition to this study. These are our references. Alhamdulillah, we could get the Ada Health Award for 2024 for this study in last year from Saudi government. It was a great achievement for our hospital. And this is our cardiac rehabilitation team at King Abdulaziz University Hospital, Jeddah. Thank you.
Thank you so much.
Do you have questions for me?
Thank you very much. Yes, so happy to see you here. And I have a comment and a question.
Yes, please.
Teaching at the University Hospital is accessible for a lot of people.
Yes.
But I have need also to highlight that Ministry of Health Hospital is covering 70% of the health system in the Kingdom of Saudi Arabia. 70%
Yes.
for the Ministry of Health. And the other things for the other private and other government sector. Mr. Lina, do you benchmark yourself with the same
Yeah, actually, we tried to benchmark in Saudi Arabia. We have no definite studies or benchmark even in MOH, but we are trying internationally. This 10 to 15 is acceptable for cardiac patients readmission rate. So currently, we are going with this rate. But I think we need to have a proper benchmarking in this field of cardiac ventilation and readmission rate, which could be done with the multi-center studies, inshallah, in the future.
Thank you.
Thank you, sir.
Any other questions for this seminar? I'm so grateful
Thank you.
for your information. Congratulations.
Thank you. Thank you very much.
And now, for another exciting exhibit today, we have Mr. Carlo Sala. And Mr. Sala is sales director of the EMEA and LATAM at San Francisco. His exhibitive presentation is going to be about Symbio, which is translating the microbiome science into global health solutions. Wow. Please welcome Mr. Sala.
And for the presentation. Done. So, first of all, thank you for inviting us and having the possibility to just present our company, but at the end, our studies, because we are a science-based company and we believe a lot in the science. So, we invest a lot, and this is part of our journey. So, I'm here to present Symbio. And Symbio is, in our opinion, one of the best solutions for probiotic use globally. It starts from literally years and years of research. And now we would like to leverage this research inside in the commercial field. But let me talk a bit more about Symbio. Why we have this solution, how we discover it. All start with a European project called Crown Life. And this project was trying to understand how the microbiota can change literally between north and south part of Europe, understanding the impact of the diet, understanding the impact of different diseases. And this is more, it's literally important to understand how the microbiota can modulate also the health. And it's part of the key of the role of Symbio. And based on that, we was also trying to understand how literally the impact of just one food can have literally an influence on the gut microflora. Starting from that, we go directly inside in this project that was taking literally several partners from different parts of Europe, from UK, from Ireland, from Spain, France, Italy, Sweden. And we was trying to touch different kind of countries just to understand this impact. And one of the rules of Symbiotech, that is the spin-off of University of Camerino, that is part of Sacco System, that is the second oldest university in Italy. Just to give you an idea. What they try to find is the impact literally about the diet. So they was trying to recruiting different kind of people inside in different centers. They was analyzing the short chain fatty acid. They was trying to understand the fecal bile acid inside the feces. They was trying to identify the strain that are inside in this sampling. They was trying to understand and characterize this. And once we have done this, we discover Symbio. Symbio is literally a blend of two strains. Both of them are coming from a human source, but I will talk to you about later. And this is a patented brand and a patented, let me say, mix. From the Crown Life project, so we discover from a centenarian male inside in a small town in the center of Italy, these two strains. And we discovered that these two strains can work synergically very well in different kind of diseases. And they was trying to adapt themselves inside in the human body. They was trying to adapt themselves and also modulate and try to have a big impact on that. So the first scope in the science, when you discover something, is try to understand if this can help literally inside the human body. And the first screening was literally an in vitro screening in which we was trying to understand if the impact of the, let me say, stomach acid that you can have, or the bile tolerance can, let me say, have an impact on Symbio or not. Second part was the adhesion of Symbio. It was able to enter inside in our microbiota and colonize. The third part is also the antimicrobial activities. And also linked to that, the percentage of adhesion about this strain, these two strains inside in our gut. And that is the last point, but it's more important, the persistency, so the presence of the strains, these two strains inside in our fecal samples. So we was trying to go directly in different kind of analysis. The antimicrobial activities was done on all the most, let me say, famous pathogens, just to understand if this two strain can have an impact on that. And yes, they have. And go directly, we was trying to understand the different application about Symbio. If was able literally to work in different kind of areas. And we start from the gut because it's the most simple. When you think about probiotics, you think about gut. But at the end, starting from gut, we was trying to touch different kind of areas. The gut application, one study, was done on 50 subjects. And we was trying to take this Symbio blend, composed, as I remember again, by one paracase, one rhamnosus, inside in this capsule, and was delivered to 50 patients for 12 weeks. What we saw was literally an impact inside in the microbiota. So Symbio was present and was able to, let me say, recolonize with Lactobacillus and Bifidobacterium species our gut microflora. So it's able to colonize and also make in favor, so regrow Lactobacillus and Bifidobacterium. When we are becoming older, the Bifidor are going down. Here we are trying to help with something, and Symbio was working very well on that. Beside that, was able, as several probiotics, to have an impact on the constipation, have an impact in the intestinal regularity, and literally have an impact also in the frequency when we are going literally to make our stuff. So based on that, another super impact was done on the short chain fatty acid that we will try to speak about that later. But the short chain fatty acid was and are linked literally to different kind of stuff. Let me take not just in the gut microbiota, but let me think also in the mood and stress. These are very, very linked. So how much you have short chain fatty acid, how much you are able literally to, let me say, resist better to the stress. Butyric acid, etc., are linked to that. And was having literally a big impact on the fecal mass. But this was another study, very well done. As you can see, 848 subjects, so very big number. And we was trying to deliver Symbio in different kind of condition, not just in capsule, but also in some food. Just to see if Symbio was able to adapt himself to different kind of delivery. And yes, was working very well in all the formats. And what we was trying also to see is the impact of Symbio on the gut-brain axis. That is another great topic linked to the microbiota. So how the strain can have an impact inside in the brain. And we was trying to have this study on 40 subjects. and was linked to the air flight crew. So we was taking this air flight crew, we was giving them Symbio to check the impact about this product, and was literally super nice to see how was having impact first of all in the immunity. So the IgA was increased. The IgA is a marker, is linked, to the immunity. So we was try to see this inside in saliva. So it's a very fast test. But at the end, we saw an increasing about the IgA. Based on that, we was checking by a questionnaire different kind of impact on Symbio, linked to tiredness, linked to stress, linked to all this stuff that now we are calling gut-brain, and was working very well. One of the scope of the Crown Life project was try to understand the impact about the diet inside in the, let me say, living longer. So we was trying to go also in this area with another project called Probiotics Senior to try to understand if Symbio was having an impact also in the health and strength aging. But let me think about more about what is this aging. When we are past 50, generally we are becoming, let me say, mature. But in the same way, we are starting to have some inflammation. This low-grade inflammation is generally called as inflammaging. So we start to have this inflammaging, we start to suffer about this secondary level of inflammation. And what happened? Happened that later, time by time, and with, let me say, some impact, you can start to have some diseases, some issues that can come. Why? Because our body is reacting, let me say, less than previously. We are reducing our biodiversity inside in the gut. And moreover, we can have starting this low-grade inflammation and can, let me say, attack the different other area like the muscle. And I can touch this like sarcopenia. I don't know if you are familiar for sure, but yeah, this is another, let me say, disease that you can have with this precursor of the inflammaging. And with Symbio, we was trying to understand the impact about these two strains inside in this area. So we saw that the strain was isolated by a centenarian inside in this retirement home in Fermo in the center of Italy. We saw that this was having literally a very good impact in terms of certain fatty acid, was able to increase the, let me say, the to decrease the oxidative stress, was able, in another sense, to recolonize. And this is another part. So making agar again, Lactobacillus bifido species. So helping the immunodefense. And we go directly in one study linked to this healthy aging. And we was taking 97 subjects over 65 and giving them 5 billion, so 5 10 to 9 cells gram to them for 6 months and checking the impact. And what we discover is literally linked to an improvement of this, so a reduction of this inflammation precursor, like the HSCRP. We have a good impact, as you can see in this graph. And we have also a reduction of the serum cytokines that are pro-inflammatory cytokines that you can check literally. And side effect, let me say. Also, we have an increasing of Akkermansia. I don't know if you have heard this super strange name, but Akkermansia now is quite well known that can have an impact inside in the human body. There are some professors like De Vos that are working a lot on these species. And we saw that Symbio was working very well on that. So, based on this and increasing on the bifidobacterium, we saw literally a strong impact about Symbio. This is another part that was studied inside in this Probiotics Senior project. And as you can see, literally the increasing in 6 months of the short chain fatty acid and an increasing of the butyric acid. Last but not least, as I said, the impact inside in the immunostimulation, plus all the reduction of these, let me say, precursor of the, let me say, more severe inflammation. And IGF-1 is one of the markers that we was checking. And moreover, with this questionnaire, we was also trying to see how was the impact about this administration on the malabsorption of, let me say, not well recovery about the food. We passed from the gut. We was going to the healthy aging, and we was trying also to see, linked to these immunostimulation properties about Symbio, to check the impact inside in the allergy. And we took literally subject adults from 18 to 41 years, and we was trying to check the impact about Symbio on the dust mite allergy. Why the dust mite? Because at the end, inside in our home, we can have dust. We can have these mite that can be present, and you don't know. You start sneezing, you start to have a different kind of impact, but maybe you are suffering about an allergy you don't know. So we was trying to understand the impact about Symbio, with this kind of, let me say, diseases. And the impact was very nice because at the end we saw that there was a reduction of the scratching the eye. There was a reduction of all the reactive symptoms that you can have when you have an allergy. And we took Symbio with just 15 billion. That was a normal, let me say, a boosted concentration, but quite nice. Sorry. Based on that, we was trying to understand also the impact on Symbio on another area again. Why? Because one of the pain in the muscle that you can have after the sport activities can be literally linked to some oxidative process. So you can have pain in the muscle due to that. All of us know that when the muscles are running, we are producing lactic acid. So sometimes you need something that can help to reduce this. And Symbio was able to act also on that. A study was done, the first one on 24 subjects, 1 billion a day. And we just saw the reduction of these on their own. And the impact again on Symbio that you can check easily here on the antioxidant activities. So with these antioxidant properties, the athletes, let me say in a simple way, was recovering better. Another study, the second one that we have done in the same field, this is done on cyclists. We was just checking also the impact, not just on the recovery, but also on the immunostimulation part. And we saw a reduction of cold, fever, despite, all the other parts that are linked to having less tiredness, having literally another part, sorry, most important was literally the pain that when you are, let me say, train yourself hard, you can have, and there was a reduction. All this was done by another study. Last but not least was literally linked to the impact post activities. You have always a lag phase in which you can have, let me say, a recurrence of fever, cold, or something like that, because your immune defense are going down. And with Symbio, we are able to restore this, and let me say, have an impact on that. So, journey that starts from the gut, healthy aging, we was going in the allergy, in the sport application, and we touched the last point, that is the woman health. It's the most trendy, let me say, area in which a probiotic can have an impact. And with Symbio, we start still also here a journey, starting from the lab. So going in and checking the activities of Symbio against all the, let me say, human health pathogens, starting from the Candida formally, and going that are yeast, and going inside in the E. coli and all the pathogens that can have an impact on that. And we saw an activity that was very nice against these pathogens. So we have literally production on antimicrobial activity, on antimicrobial, let me say, components. We have this co-aggregation with the pathogens, and you can check here the impact on the different candidates made by Symbio. That is this combination. This was translated literally on all. I mean, we was checking Symbio against also competitor and the mechanism of action about Symbio, just to check that. Sorry, Symbio was mainly able to inhibit and displace the pathogens and go against this. And we decided to go in two kind of format. The first one was vaginal suppositories. That was a medical device until 2 years ago in Italy. Now we was not able to have this anymore, but we was just working on another version. And in this version, we was able to see how the impact of this probiotic can, let me say, go against these pathogens. It can report back to the normality or at the end, let me say, go to normal level of the Nugent score. And subject are 35, 1 billion a day, and the treatment was 7 days, let me say, 5 days, and after one suppository, vaginal suppository a week. After this, since the regulation was changed and the probiotic was not allowed anymore in Europe to have this in the medical device, we was checking the impact of Symbio in an oral product. And based on that, we was giving 5 billion a day, checking after, let me say, a week of treatment, and in total in 15 days. What was the impact linked to the Amstel criteria, to the Nugent score, to the presence about all these pathogens. And the baseline was that Symbio was able to go inside in the gut, reach the vagina, try to colonize, and at the end was having literally an impact on the Amstel criteria, coming back to the normality, and on the Nugent score. So in literally, let me say, 2 weeks, we was able with an oral administration of probiotics to avoid any kind of antibiotics, but to work just with something natural that is a probiotics to return back to the normality. So this was the last part of this different application of Symbio that show how 2 strains of probiotics discovered in let me say in a project can have literally a big impact on different kind of areas that are very far from them, but these show how the gut is in the center of our well-being. How the gut can have an impact on different kind of, let me say, axis. And just to show how the probiotics now are growing, and are growing more and more, because at the end, we are always thinking to find some natural solution, and this natural solution can come from probiotics. Thank you.
So much for this really very enlightening presentation on Symbio. I'm sure we have a lot of questions for
Thank you very much. First, I'm taking some time probiotics. But my question, all the studies on microbiome, they show association, not causation. Most of the studies. The association can be also highlight different things. But there is no study with causation. We are free to visit these 3 and 4. Secondly, what is did you have in the field of malignancy any study about your product? Third, you should have brought some samples for us.
So for sample, we have several sample there. So if you would like, we can share it easily. The important stuff is that probiotics are now, in my opinion, trying to raise up the voice, let me say, because for now are just food supplement. But there are a lot of companies, and I can tell you because Ecosystem is also producer of API, so the pharma product. There are several companies that now are going in phase 1, phase 2, phase 3 with just probiotics, nothing more. This because at the end we believe that these components can have an impact equal to, let me say, chemical components. And it's shown that they work very well. I mean, in different areas. In Europe, to sell a probiotic, you cannot sell just Symbio. You need always to add something that can, let me say, help you to have the claim. So if I have to go to the woman health, I need to put cranberry, or I need to put, I don't know, demon nose to try to show that it's working in that area. But in theory, just probiotic can work. But this is a message that I cannot deliver to the consumer because at the end, the European, but also in globally, let me say, because it's just the US that can help and also Australia that can have these authorities to just show the study about the probiotics. But in the other area, you need always to combine. And in my opinion, it's a bit, let me say, a poor position about the probiotic that can do a lot. So I hope that in the near future, we can raise up the voice and we can show that probiotics can be literally, I'm not saying a medicine, but something like similar. Yeah.
We see 100 billion, 50 billion. Which one should we take actually? And which, and ask also for other billion. Lactobacilli or which other bacteria we should take?
Yeah, I can tell you that, I mean, Ecosystem, we have more than 150 years of experience. It's a family company. We are producing probiotics from years. And we have done probiotic with 95 strain in one capsule, probiotic with 100 billion, 5 billion, 1 billion, independently. But what, in my opinion, is important is literally study. And what we have done with Symbio was studies that can show that this kind of, let me say, concentration is able to do that. There are several products on the market that can have a lot of billion inside, but maybe also with less can work. And we don't know.
So I'm curious to rephrase what Dr. Muhammad is saying, because there are a lot of questions. He talked about collaboration and association. So that's the first of his questions. And second is the relationship to malignancies. Have you studied that with malignancy or not?
No, for malignancy, no. I need to recap a bit. But in terms of causation, etc., yes, you can have you can study the causation of some diseases, and with the probiotics, you can act on this. But you need, first of all, to understand also the mechanisms of action about the probiotics, how they can have an impact on that.
So, even as healthcare providers, we don't know. He just said we don't know today. We don't know everything. So as healthcare providers, we need a lot of education for the consumers.
And this is why, as Ecosystem, we are trying to participate to this kind of events, to try a bit to share knowledge. Because we believe now I'm not selling a concept, I'm trying to speak about probiotics. and try to sell the fact that the probiotic can help in different areas, but with the right dosage. Because I saw literally on the market tons of products with different billion, with different components. You asked before, is better lactobacillus or better bifido? When you are becoming older, you are reducing the bifido, so you need bifido. When you are younger, you have, let me say, higher bifido and less lactobacillus. So if I have to give you a proportion, maybe after 50, more bifido, before more lactobacillus. What now is growing is Akkermansia. Why? Because at the end we discovered that lactobacillus and bifido are just, let me say, 20% of our full microbiota. We have tons of other strains, Fecalibacterium prausnitzii, Akkermansia, Veillonella, are all present inside in our microbiota. And we never discovered these until 10 years ago. So it's still a growing, yeah.
But the half open, like from decent fibromyalgia, and it's something that's around a lot of women and men are being attacked by because of oxidative stress. Are there any studies on these autoimmune diseases specifically with symptoms?
We are working on that, I can tell you, because we saw this kind of potential of antioxidant activity, so we are working on that. Because we believe that Symbio can go more and more and deep on that. Now we start, let me say, from the top line. It was going down more in the, let me say, chronic diseases, because we believe that Symbio can work very well on that also.
Questions?
Thank you for that beautiful talk. My observation is the way you presented it. Symbio will go for allergies, Symbio will go for vaginal, mental health. Do you guys consider to compatibilize things in the context of the fact that you make a nasal preparation for allergy, you make a vaginal substitute for women's health, you make a oral capsule for each? Because really, I'm just looking at it, all you want in this room, one set of needs and health personnel. And if you tell me you're good at this, you're good at that, you're good at that, the next piece of the mix. But if you make specifics, if you're saying lactobacillus, video is the main thing, and you have a thought point, then you go this one vagina with different preparation. You go that one as a nasal spray for allergy. I think it makes it more, this is just an observation. It makes people get more of the concept, especially the healthcare, and then less comparison to the rest of the world. That's my first observation. The second thing is, are you looking at dental preparations? Because the likes of psoriasis, acne is a strong growth microbiome. So are you looking soon to go in that direction? Second, my last question is, you explain why it's not easy to get authorization in Europe. My next question is, America is looking at everything. So if you are looking at American market, for example, are your oral capsules given approval as a medical device, or will they stay as a food supplement? Or do you think, you made mention of the fact that a couple of companies are already doing phase 1, phase 2, phase 3 trials. So if you are getting as far as phase 3, that tells me you're open to get a license and a proper indication and a method of being to say as a medical device. So do you think that's going to happen anytime soon in terms of this? And the lady from Berlin that presented earlier was talking about Crohn's. Crohn's ulcerative is so clear. If you smash it, you get it right. There is a huge population there. There is a lot of people seeing that there's evidence showing that if you can modify their gut, maybe we don't need to start going towards the first one and the rest of it, because everything actually becomes sweet. So those are my thank you.
Thank you for your observation. I can go easily on the dosage format that you were saying at the beginning. What we need to guarantee is that the probiotics can stay alive. And this is the most important fact. I mean, I cannot sell a product that is not able to act. And the suppository, the vaginal suppository, is not allowed anymore in Europe. It was in other countries, but was working very well in that area. And so this is why we skip and we go through the oral intaking. to check if it was working also in that. For the nasal application, as I said, you need to guarantee that the products can stay alive. And this is why we are thinking on that. I'm not saying no. But at the end, we were showing that just with oral intaking was having an impact, Symbio, inside in the allergies. And there is already another product in the market that contains one of the strain of Symbio that was having a study on the seasonal allergy, just oral intaking. So we are thinking on how we can have an impact in different area with different dosage for, yes, but we need to guarantee the strain can stay alive because we don't want to sell something that is not working. I mean, the other point linked instead to the, let me say, phases in terms of application with probiotics and going pharma way. I can tell you that we are strongly active in the pharma field. We are producing already 2 pharma products, one that is sold in Italy and in Colombia and Ecuador. That is linked to the, let me say, chronic diarrhea and this kind of stuff. With already clinical evidence, it's a drug, literally. And another one that is working on a female care in association with an hormone. And what I saw is that in the last 5 years, we had literally a booming of companies that are pharma companies that are going inside in the Crohn disease stuff, or let me say, not just in the Crohn, but all the chronic diseases, because they are trying to find something different than a chemical molecule. And I can tell you that for sure in 5 years, we can have something, because there is a lot of work in the back. And skin. We are touching skin. Symbio is working on atopic dermatitis also. And there we have also some concept that we have think for the skin, because we saw a great action in the gut, let me say, skin axis. And we are working strongly with one super professor in Spain, Vicente Navarro, on different kind of application for probiotics in skin diseases. We was treating with probiotics the alopecia, the rosacea. We was trying to work in these diseases just with probiotic oral intaking. But we have done also concept in topical use. And we guarantee that probiotics are working also in topical because we check the shelf life of the product. So what we would like to sell is a product that works with probiotics, not just because we have other components inside. I hope that I touched on this point. Good. Yes, please.
I myself am from the United States.
Yeah.
Pelvic floor physical therapy clinic. So my question to you is, as a potential consumer of this product, is this third-party tested? Because when I recommend product to my patients, I want to make sure that it's at least NSF certified or third-party tested.
Yeah, let me say that this product was just checked by external parties, not by us. And we was, let me say, acquiring Symbiotech, that is the spin-off of University of Camerino, some years ago. So all this activity was done without any company filling in. That it's the most important part, because at the end, if I'm sponsoring let me say, a study, for sure, I would like that I can have results that can show that the product works. So these are results done by university and after purchasing that acquired by Sarcosystem. And what we are now trying to do is also work more in collaboration with distributors around the globe. Means we are giving them Symbio, they are making their own studies, we are not involved, just to show that the product is working without any interaction, let me say. And it's a process, so we are growing, but I believe that what we show here, it's quite interesting because we can literally touch areas in which several other products doesn't work, and is the scope of sharing information. Thank you.
One more question, I believe.
Yes. There is another one, I think. Sorry.
Dr. Salas, when you're going through your presentation, you brought up Peninsula, which I know is newly discovered. Someone that I work in private practice and I'm always looking at kidneys to testing and I'm always shopping for probiotics. Did your study show that it was an increase in Peninsula using this in you?
Yeah.
Yeah.
Yes.
I know that Peninsula works in the line with the gut, the brain, and also keeping the tight junctions.
Yeah, it's working very well on the, let me say, metabolic system. So you can find different studies now. One is the Accredited company that is linked to Willem de Vos, that is the discoverer of this species. There is pendulum also in the United States, but pendulum come later to this Accredited. The Accredited of pendulum was coming later than the Accredited of the Accredited company. And despite that, what they show is that this strain can work also in inactivated version, means a dead version, or also in live version. What we are trying to show here is that CPO is able to increase your own Accredited. And it's super in my opinion because you don't need to have any other supplement than just yourself. Yeah, is able to increase this one. So this is the scope. I mean, find a way in which two strains combined together in the right way can give you an impact on different kind of areas.
The probiotics that I use has the rhamnosus and has the Pantera.
No, these one are one paracasib, one rhamnosus.
But then again, I work for my company as proposed, so I do like
Research.
lifestyle, everything. So it is a synergy, the combination of everything. So technically, you don't have to take Accredited alone. You can take a different kind, right? Those two strains improves.
Correct. Yes, you can take something that can increase, or you can take also a combo. I mean, what we saw is that this is able to increase your own one, but there is someone now that are working more. There is a GLP-1 areas that is growing so much, is booming, and so all the guys are now working. Okay, Accredited can work on that, but the scope at the beginning was not just to reduce weight. The scope was to have just a good well-being, a good metabolism, let me say. And now we go more in the commercial, but Accredited work also in that area.
You guys do sell in America, right?
No, we are not still selling in America. I mean, we have a production site in United States. We are now trying to enter. We are already selling some brand, but Symbio is now looking on partner also in United States. Thank you. Yeah.
Actually, it's a very small question.
Yes, please.
My question is there any lack of warning regarding the use of these products, especially people for severe immunosuppression or anything?
No, we was trying also to work with Symbio inside, in the cancer therapies. So we are giving them the probiotics when you are making, the treatment for the cancer. And we saw that the strain is working quite well, not having any kind of impact inside in the immunosuppressed person, but it's literally helping them to increase the immunity capacity. So in my opinion, it's a product that is safe, is working very well. And we tried also to overdose without any kind of impact in the human body.
Thank you, Mr. Salah.
Thank you.
Graciously answered
Thank
all our questions.
Thank you.
Patients, thank you for all the presentation, and we can accept this certificate.
Thank you.
All right, on to our next speaker, and we have now. We have Dr. Fuji. We have Dr. Fuji, our Masashi Fuji.
His presentation's title is Comprehensive Approach to Strengthening Endoscopic Healthcare Infrastructure with a total stimulator. Welcome.
So my slide I have to change. So can I change the computer?
No, actually, everything will be like disconnected.
I'll disconnect. Okay. 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 So hello everyone, and I'm an endoscopist from Japan, and Japan is one of the fastest aging countries in the world, and many people die from cancer. I've worked in endoscopy for many years in this situation. Cancer can be cured with early detection and early treatment. Today, I will talk about how we can grow endoscopic medicine to help find cancer early and treat early. Okay. Many people around the world die from cancer. Colon cancer is stomach cancer cause many cancer deaths. Colorectal cancer has been increasing year by year, and in recent years, the rate of increase has become even higher. In many countries, responding to colorectal cancer has become a major challenge. If colorectal cancer is found before symptoms appear, more than 95% of patients can be cured. The only effective test examination for early detection is endoscopy. Early detection also helps reduce the cost of treatment. But there is one big problem when we try to increase endoscopic exams. We do not have enough endoscopists over the world. This problem is even bigger in countries outside Japan. As you can see here, Japan has about 28 endoscopists for every 100,000 people. In many other countries, the number is less than 1/10 and 100 of that. The shortage of endoscopists affects the 5-year survival rate of cancer. In Japan, the 5-year survival rate for stomach cancer is 75%, and for colorectal cancer is 76%. But in many other countries, the rate of often below 50%. The main reason for this difference is the stage at which cancer is found. In Japan, more cancers are found at stage 1. Other countries show a similar pattern, including Saudi Arabia and the UAE. Also found late stage, so the 5-year survival rate is very low. This chart shows the ranking of cancer deaths in Japan. Stomach cancer was the #1 cause of the cancer deaths in 1965. But by 2023, it had fallen to fourth place. Another report shows that the same trend. In the 1960s, the 5-year survival rate for stomach cancer was only 42%, but by the late 1980s, it had improved to 71%. This slide shows the treatment outcome by stage in different decades. Even today, there has been a little improvement in survival rate within each stage. And especially stage 4, 5-year survival rate is not improved. One important reason is that the endoscopy was introduced in routine cancer screening system in the 1970s. Because of this, many cancers were found earlier. This clearly shows that effectiveness of endoscopy, which is the only method that can truly achieve early detection and early treatment. From this, I believe we need a society where everyone can get endoscopic medicine. To do this, we must find a way to increase the number of endoscopists in more efficient way. So I developed an endoscopy training simulator that allows doctors can learn by themselves. We now have 3 models: the upper GI model, and the colonoscopy model, and the ERCP model. These are the Mikoto series endoscopy simulator designed to help doctors learn faster and to support the spread of endoscopic medicine. In the past, learning endoscopy skills took a long time, but with Mikoto, doctors can train in a much shorter time, from a few days to a few months. Mikoto Simulator series is the world's first simulator with a scoring system for each procedure. It checks patient pain, observation, and scope control. With this system, doctors can learn good endoscopy skills faster. Mikoto series has a learning content, so doctors can practice by themselves. It is designed to help them learn endoscopy skills in a systematic way, even without a trainer. Training only in the clinical settings has limits for learning endoscopy skills. But with this simulator, we can train many endoscopists at the same time. We have worked in more than 60 countries, and our work has been highly valued. This time, we developed a few new system for further support endoscopist training. We call this system Mikoto ID system. With this system, each device connects to the web. Users can check their scores and skill levels. There's also a trainer feature. Trainers can see how trainees improve, and by teaching more doctors, they can raise their own trainer score. The reason we added this feature is that it is important to evaluate trainers who teach endoscopists in a fair way. Today, we often use the term KOL, Key Opinion Leaders, but I would like to propose a new idea, Key Education Leader, KEL, a score that shows how many endoscopists a trainer has helped to develop. Finally, I would like to introduce the training we conducted with Mikoto and at the recent APDW, Asian Pacific Digestive Week 2025 in Singapore. we worked with Olympus. We had a self-training event with 10 Mikoto units. More than 150 people joined the training over the 3 days. And the total number of training sessions was over 1,300. We used this program for the training. When participants cleared each score, they moved to the next stage. During the 3 days, we could see their endoscopy skills improve. We believe that endoscopic medicine has the power to save lives and help humanity grow. We will continue to work to expand endoscopic medical infrastructure around the world. Thank you very much.
Thank you very much. Myself, I did more than 600 endoscopies with gastroscopy and colonoscopy at the University of Vienna Medical School when I was studying like that to take on a board certification. That time we were blindly observing. Now it is much easier. You see, and it is much easier and it's safe to as expected. But one of the difficulties in the endoscopy set is the cleaning of the endoscopy set. It is very important. And do you think in the future we'll be having disposable scope or not?
Thank you. Very good question. I think it is important that cleaning of the endoscopy system to spread the infrastructure of the medicine. But nowadays, the endoscopy system is makes a very high cost. So we should reduce the cost of the endoscopic medical care, and it is very difficult to make some disposable system. Some companies making and selling the disposable endoscopies, but it is limited use in the emergency part in the infectional part. So I think it is important how to clean and certificate the how cleanest in the of the endoscopic systems. Yeah, I think. And also, I think we often use pig or other animals to learning how to endoscopy and endoscopic procedures, but it is our Mikoto simulator series is a non-animal can no without animal with a how to procedure like a ESD or OM. It is important to the animal-free processes for the learning, I think.
Thank you. I think interesting presentation and good simulation. I think I found something interesting in the presentation. You were stating that Japan has the need for the greatest number of endoscopies compared to the rest of the world. Can you please tell me why this is so? What do you think are some of the factors that account for why in Japan there's a need for so many endoscopies as compared to the rest?
Yeah, thank you for your question. I think the several reasons it has. The first one is in Japan, the gastroscopy is not for only specialists, but also the other doctors can gastroscopy in their clinic. So the endoscopist is a very high volume in Japan. Gastroscopy is very easy for learning by simulation training, and in Japan, many universities using the simulation trainings. But in other countries, only specialists using an endoscopy. So it is I think it's a program. And also in UK and Sweden and the Netherlands and some states of USA is allowed the endoscopic nurse. The nurse can endoscopy, including colonoscopy and polypectomy themselves. It is more reduce the cost of the endoscopic procedure and screening systems. So we want to increase the number of endoscopists and endoscopic procedure person with such as education. And endoscopic, especially colonoscopy, is very difficult. So we should improve the insertion method and insertion instrument for the more easier with Olympus and the Fujifilm. So that.
Thank you so much. Any more questions? Thank you. Thank you so much. Our next exhibitor is Mr. Wael Soliman, EMEA Commercial and Professional Education Manager at ASP. His exhibitive presentation title is Shaping the Next Era of GI Processing. Please welcome Mr. Soliman.
Can we have the chance to run the presentation? Yeah, we'll just check. Right, so good morning, everyone. So thanks to the organizers for giving us this opportunity to present today. My name is Wael Soliman, and I represent ASP, or Advanced Sterilization Products Company. I work for professional education in the company across Europe, Middle East, and Africa region, and we are pleased to be with you today in this nice presentation and view together how we can look into shaping the future of reprocessing of the GI endoscopes. ASP is a pioneer company in solutions related to infection preventions. So what basically we do, we provide low temperature sterilization technology that can sterilize the heat sensitive equipment, and one of them is the endoscopes. But just before I start, a disclaimer that we provide a solution, we provide a technology. However, it remains to the healthcare setting to have the right setup to their patients and follow the manufacturer instructions for using any kind of medical devices within the healthcare system. About our company, we are innovating infection prevention solution. We actually, more than 30 years ago, we are a global leader in providing technology related to low temperature sterilization for the heat sensitive devices. And that came because of our mission that we want to protect the patients during their most critical moments. If you look into our presence worldwide, we are a US company and we work across the globe with more than 20,000 installations. That gives us more responsibility toward our patients and toward our healthcare professionals to be available, providing the best quality products and the technical support. And we are so proud to have millions of devices being processed in our equipment to provide contaminant-free instruments. Today, we are going to talk about something different, as we are putting our step into the endoscopy suite with the GI endoscopes. And I will shed a light in this presentation how we can view together the way we are processing one of the most important GI endoscopes we use, which is duodenoscopes. But before talking about duodenoscopes, let's have a quick refreshment about ERCP, the endoscopic retrograde cholangiobacteriography, which is the primary endoscopy procedure that we use it for treating the pancreatic diseases and biliary diseases that combine the upper GI endoscopy and the radiological imaging. This procedure could be diagnostic and could be therapeutic, as you know. And actually, you always use two types of imaging during this procedure. The image that's coming from the endoscope itself, and we combine it with the radiologic imaging when we use the guide wire to do more therapeutic treatment to the patients. How it all started. In 1968, the first ERCP procedure had been done as a diagnostic procedure, just injecting a contrast to the patient to detect the anomalies within the biliary area and pancreatic area, and then the patients would be referred to the surgeon for further treatments. Then, 1972, we have seen the first cannulation was performed within the ERCP procedure, transforming this procedure from being a diagnostic procedure to being more therapeutic procedure when we have the guiding wire involved. Today, the ERCP procedure is a primary endoscopic approach for treating the pancreatic and biliary diseases. And according to the National Institute for Cancer, we would expect that 3.3% of all new cancer cases are pancreatic cancer. And you can note the volume of this proceeding happening globally. I would say only in USA, they perform more than 650,000 procedures of ERCPs annually. That's why we need to give more attention around how we can reprocess this kind of endoscopes, because they are being used in more critical way nowadays. And to achieve your procedure beyond the diagnostic procedure and going further with the therapeutic part, probably you need to do a lot of interventions, sphincterotomy, stone removal, stent placements, taking biopsy, drainage, dilation, tissue sampling, a lot of procedures and interventions you can do within this procedure. And to do this, you need to have some accessories in your hand, like using the balloons while using the stent for doing some dilation to the biliary duct or pancreatic ducts, or using even the baskets for removing some stones with it. And that should be assured because this instrument should be sterile being used within this procedure. The most important for us as a company providing solutions to prevent infection is to look around. What is the risk associated with the ERCP procedures? All of them are there. Post-ERCP pancreatitis, cholangitis, hemorrhage, duodenal perforation, and the most important for us, and we're gonna deep dive into this, which is infections, different sorts of infections, more related to the nosocomial infections. What is nosocomial infections? As all of you know, we call them often the hospital-acquired infections or the healthcare-associated infections. Those infections happen to the patients when the patients come to the hospital to receive a treatment, and during his stay, he will acquire an infection. And these kind of infections became a burden on the healthcare facilities because you can imagine that these patients can stay longer within the hospital and can occupy another patient's bed, can occupy a bed within the ICU. And you can imagine that we need a qualified staff to treat those patients when they acquire those kind of infections. The length stays, and even we look into the financial burden where we see even how governments are financing this kind of treatment to the patients. And even when we look into the private sector, for example, we see even the hospital image and the reputation is really important when it comes to this topic and how it could be impacting the reputation in some way. And that sometimes insurances will not be able to cover this kind of infections. I would like to reflect as well related to the infections related to the duodenoscopes and ERCPs. A super bug tragedy happened in 2015 in the US after ERCP procedure, an outbreak coming from the CRE, one of the multi-drug resistance organisms. And in USA, they figured out that 7 patients were infected with this kind of microorganism. 2 of those patients have been died. And the thing when we look into analyzing these cases was 15 studies that revealed that the contamination rates, and you can see of the rectal cyst patient-ready duodenoscopes was more than 15%. So you can imagine that even the duodenoscope was patient-ready and it was already reprocessed, but we still can see a high contamination rate within these kind of endoscopes. And the question here, where we can categorize these duodenoscopes? Can we consider them as critical devices or semi-critical devices? Because as you know that there is a bit old scheme for classifying the medical devices according to the scientist who created this classification, Dr. Iris Spalding, more than 70 years back. So thinking to classify the instrument into 3 main categories, into critical devices, and those instruments can come into the main bloodstream, more invasive, come in touch with the internal body organs and the striate tissues within our bodies, and we consider them as the major surgical instruments, and they require sterilization. However, when you look into the semi-critical devices like flexible endoscopes, we see them under the category of the semi-critical devices because they come in touch with the mucous membranes when the scope, for example, the GI scope or colonoscope, that penetrate through the natural orifice within the body and come in touch with the mucous membrane. Will not invade these mucous membranes. However, we see often during this procedure, we need to do some therapeutic treatments. So this kind of classification became a bit an old scheme because if you look into the semi-critical devices, we would understand very well that high level disinfection would be enough. And we would agree theoretically that high level disinfection would be enough for the semi-critical instruments. like the GI endoscopes, and I will say still we call the duodenoscopes under the same critical category. However, can we consider the high-level disinfection is enough for reprocessing this kind of duodenoscopes? Theoretically, we assume yes. However, there's a lot of challenges on the way. When we look into the complexity, for example, of these duodenoscopes, we see often them consist of sensitive materials with multi-channels. And as we live in an imperfect world, we would expect that dealing with this kind of complexity and the channels while the nursing team trying to do cleaning and to do high-level disinfection to this kind of scope, it would be very challenging. So we assume that HLD would work if we are doing perfectly all the reprocessing steps without having any soils remains or contamination remain on the duodenoscopes itself. Look into another challenge related to the biofilm formation, and all we know this with the complexity of the medical devices. We could expect a lot of areas within the device will not be easily accessible for cleaning. And if we have imperfect or inadequate cleaning, we could expect hotspots for material accumulation, and then the biofilm will start to develop. Then it will be very resistant to remove those kind of biofilms. And this could be a source of infection for sure. Look into testing and monitoring. While we are doing high-level disinfection to those kind of scopes, we only validate probably a high-level disinfectant chemical and use it for processing those devices. However, how we look into adding a higher safety margin to the process, because generally, what is the difference between sterilization and high-level disinfection? Sterilization means destruction of all microbial life, including the bacterial spores, which cannot be killed by high-level disinfectants. So that's why we need to look into all of these circumstances. We need to look also about the cross-contamination that could be happening on the way. During the procedure, as I showed you, you are using some accessories. You are using biopsy forceps, you are using baskets, you are using catheters, you are using the guiding wire during this procedure, and we assume that those kind of accessories are sterile. And you can imagine if these endoscopes are not sterile, probably, and in most of the cases, we would expect a cross-contamination will be happening to the patient itself. Not only that, even during, I would say, storage for this kind of scopes, we need to consider a lot of factors in place. One of them, drying. We need to make sure that we have these kind of scopes away from any contamination, because we believe that water is still a source of contamination, and a lot of waterborne contaminants could be there. Sometimes, or I would say most of the time, that if we have a heavy load, microbial load on those kind of scopes, it will be very hard to the high-level disinfectants to take this microbial load down. And even to validate the process, if I would ask any user how you can validate the safety or the outcome of a high-level disinfectant endoscope, we look here that probably if you look into how the nurses are reprocessing the endoscopes within the department and how they are looking into the efficiency of that process, they only probably checking the concentration of the disinfectant that they use by a kind of test strips that they use for testing the concentration of the solution. However, when we look into sterilization with the higher safety margin, we have a lot of assurance because we have a lot of validation with this process. We use more indicators like biological indicators, for example. So the thing, if I would love to ask you a question, if you have an answer right now, you can tell me. If you have an answer later on during the day or after we finish the presentation, just think about that. Think about it. If you could sterilize the endoscopes, will you agree to switch from high-level disinfection to sterilization, considering the different reasons that I explained? Anyone would love to comment? Yes? I need to hear yes or no. Absolutely. That's amazing. Thank you so much for raising your voice for that. So, what is a solution that we would love to introduce for you today, leveraging on our experience as a global leader in low temperature sterilization? And actually, our equipments are already there in the market more than 30 years. For example, here in UAE, we have more than 150 sterilizers. In the Middle East and Africa region, we almost have around 1,000 sterilizer, and we work in a strong relation with the CSD departments, with the infection prevention teams within the hospitals. So we are already there, we are already available. But what we are introducing to you, a new sterilization cycle, which is FDA approved, and we received the CE mark last month for it. And we are really proud to provide you a new option for sterilizing compatible duodenoscopes. So here we are not talking anymore about high-level disinfection, but we are talking about the incremental safety margin that we can provide by sterilizing those kind of duodenoscopes. So this cycle will be running for 68 minutes. However, we can reprocess 2 duodenoscopes in one cycle. And the thing here that we'll get many wins out of this process. First, we will get the scope already terminally sterilized. I mean, like the scope will be cleaned, then it will be packaged in a container, then it will be sterilized in the sterilization cycle. And this equipment uses hydrogen peroxide for sterilization because we work under low temperature. We have a big experience in reprocessing other kinds of endoscopes like cystoscopes, like hysteroscopes, like bronchoscopes, the flexible ones. So, but now we are expanding our claims for a breakthrough innovation with sterilizing the duodenoscopes. So if you have this equipment within your hospital, we can only just upgrade the software to an extra cycle. So no need, if you have this equipment already within your CSD department, for example, we can just upgrade the equipment to a new cycle, or we can offer a new machine with a new cycle for reprocessing the hydrogen peroxide compatible duodenoscopes. We can reprocess up to 2 duodenoscopes in one cycle. And the claims that we got based on the FDA and the CE mark that we can reprocess the duodenoscopes up to 4 channels with 8 branched lumens. And we have been working on qualification listing with the different scope manufacturers. We have started with Fujifilm, and we have already some confirmations with some scope that can be processed. We also have been working with Pentax and in US, and we are extending this to the EMEA region and Middle East. And we are looking forward to have more collaborations with the different scope manufacturers within the GI endoscopy world. How we can achieve this? We are going to provide you with some good accessories that we can use. We use this aluminum tray where we place this duodenoscope inside, and we wrap it with the wrap sheet, and then we place it 2 scopes in one cycle. And we also provide you what we call it a BCD, or a process challenge device. We place inside a kind of a biological indicator where we have a bacterial spores. So this is a kind of a test that we do with the cycle to make sure that we are able to kill this type of bacteria, which is the most resistant bacteria, to provide the sterility assurance level. So here we are talking about new process where we provide the sterility assurance level, same way we sterilize any medical device, but now we are going to do this with duodenoscopes. So I wonder to open it for questions, and I hope this topic would be new and interesting. So I would love to open if you have any questions or any comments to share to this session.
Just one.
Thank you so much for your presentation. Just a quick question, before the processor, the stabilizer is in the processor on this device, there is manual pre-stabilization.
Manual cleaning?
Yeah.
Manual cleaning should be always there. If you are referring to manual cleaning, even followed with automated cleaning, should be still there. But yeah, exactly. That should be happening. There is no negotiation in cleaning the endoscopes and doing decontamination, removing all the soil, the same standard way you do your cleaning, manual followed by automated. And as you know, that automated will not even replace the manual cleaning. You should continue doing, because cleaning is the queen step for any kind of processing. Then there is another thing that we need to think together, how we can manipulate the good workflow of this device. Because it depends that, like, if you would love to go directly after the cleaning to sterilization, or you still need to do HLD in place, then you go for sterilization, for example. This could be something that we can talk around that. But just to straightforward to answer your question, we should continue doing cleaning. Okay.
What's the time of the sterilization from the scopes?
It's mentioned here, it's a 68 minutes, but that's for 2 scopes in one suck. But believe me, when you look into the outbreaks and look into the infections and contamination that could be happening in place, especially why duodenoscopes? Because we understand that duodenoscopes are different from the general GI endoscopes. You have the elevator wire mechanism, because it's a very critical procedure that we do, the ERCP procedure, and using the guide wire to go into a very critical channels within our body, in the pancreatic duct or in the biliary duct. And those kind of accessories, by the way, we assume that they are sterile. So the thing that from infection prevention view, it would be more feasible. And even we have seen these directions from the guidelines, even if the instrument is semi-critical instrument. But if you have the option, if you have the possibility to go for sterilization and adding the higher safety margin to the process, for sure, we need to go for this. So that's why we have been sterilizing already long time back the other types of flexible endoscopes, like as I mentioned, cystoscopes, hysteroscopes, the flexible one, nephroscopes, ureteroscopes, all of this kind of flexible endoscopes. We have been sterilizing this in our equipments many years back. But what we are expanding today, we are expanding our claims to the GI era, which is something new for us as well. So that's why we wanted to provide a contaminant-free duodenoscopes, understanding all the dilemma around these duodenoscopes. Yeah, thank you. So, any more questions or comments?
No, thanks. Just ask you, can you validate the infection outbreak?
Sorry?
You talk about, let's say there was an outbreak of CRP.
Yes, exactly. We can provide the references. For sure, there is a lot of data worldwide around duodenoscopes and around outbreaks. We have even recent publication that came in 2025 from Corey Ofsted in the US, and she was summarizing many outbreaks, mainly coming from duodenoscopes, but also talking around the flexible endoscopes. Because we often say, probably when we look into infection prevention, we assume that the problem is coming from the critical instruments being used within the OR, but we realize that there is also a big risk coming from the semi-critical instruments. For example, like I mentioned, the duodenoscopes, especially when we do intervention during the procedure. So that's why it is a lot of things that we can discuss here. But if you need some references, maybe during the break, I can prepare something to share for you 100%.
So especially if you talk about scope or other scopes, is the same machine but focused one or different machine?
Same our equipment, but in a different cycle. So if you can see here, we have this cycle called the flex cycle, and we use it only for the flexible endoscopes. Like I mentioned, the cystoscopes, hysteroscopes, lithotroscopes, so on.
The endoscope also we go in that.
No, these kind of duodenoscopes, because duodenoscopes are special and they are different. We have a more longer length for the lumens, and we have more channels inside involving these duodenoscopes. So that's why while we are innovating our new cycle, we should go through a lot of testing and a lot of validations toward the nature of this complexity of these duodenoscopes. Then it ended up with that cycle. And we are really proud that we are the first company in the globe to provide an innovation, FDA and CE markets and approved for reprocessing the duodenoscopes in collaboration with the medical, with the scope providers, that we should have for sure qualification list with the scope manufacturers 100%. You're welcome.
Thank you. Thank you so much, Mr. Mohan, for the presentation.
Thank you so much.
Okay, and last but not least, before our lunch break, we have Dr. Akram Mahmoud Sanat, gastroenterologist at Zulekha Hospital in Sharjah. And his presentation is magnetic compression anastomosis for treatment of post-cholecystectomy by duct injury related biliary strictures. Welcome, Dr. Shaman. Welcome, Dr. Shaman.
Thank you.
Okay.
Hi.
It's here.
Thank you. Good afternoon, dear esteemed colleagues, distinguished doctors, and honored guests. As you know, I find this conference is an opportunity to share our expertise. So I would like, and I am pleased today, to share the results of the novel non-surgical interventional technique, which depend on the compression, magnetic compression anastomosis for treatment of post-cholecystectomy bile duct injury, related biliary strictures as a case report. As we know, during our journey towards advancing health care, we're still facing surgical complications as a core challenge. So, as a gastroenterologist, from my side, I prefer non-surgical interventional methods for resolving these complications. This novel non-surgical interventional techniques is considered an innovative solutions for this injury.
I need to from how can I slide? Okay, thank you. I would like to clarify and explain what's magnetic compression anastomosis, MCA. This novel innovative techniques
is a minimally invasive. As I mentioned, it's non-surgical alternative for resolving structured, narrowed bile duct. The principle of these techniques is two specialized magnets are placed across the structures. One magnet above the structures and other magnets below the structures. Mechanism, the strong magnetic force compresses the tissue between the two magnets and causes controlled necrosis, fistula. Magnets should be less than the duodenoscope channel diameter. As we know, the channel diameter of duodenoscope is 4.2 millimeter. So the magnet should be suitable to pass through the channel. So should be less than 3 millimeter in diameter. I have to mention the inventor of this technique is Dr. Igoro Yamanoshi. He is Japanese doctor. Actually, he is the inventor of this technique. So sometimes the technique is sometimes referred to as Yamanoshi magnetic compressions and anastomosis, highlighting his pioneer role. This technique is invented to resolve the problem of post-cholecystectomy bile duct injury related strictures. When there is severe injury to the bile duct, which we call it transactions, the whole obstruction. Incidence of these complications during laparoscopy, cholecystectomy, or even open cholecystectomy is very rare, 0.1 to 0.3%, but still exists, as we see. A major cause of this injury is thermal injury, diathermy burns. Biliary injury may be recognized at the time of the laparoscopic surgery, and sometimes the injury is not recognized until after conclusion of the surgery. Repair of major biliary duct injury should be approached by a multidisciplinary team consisting of a surgeon, diagnostic radiologist, interventional gastroenterologist, and interventional radiologist. So actually, this treatment, not a solo performance. We have many classifications for determination of the type of injury. We have Strasbourg-Bismuth classification, for example. We have also other classifications, Hanover classifications. Common bile or common hepatic duct injury. Injuries to common bile or common hepatic duct are the most serious complications. It's a disaster complications, actually. So I'm not going to go in detail of biliary injuries. Just I would mention in a short, in cases of more significant damage to the duct, hepaticojejunostomy will be required sometimes, and this is a massive surgery. The delayed presentation of common bile duct or common hepatic duct injury includes complaints of vague abdominal pain, persistent nausea and vomiting, low-grade fever, and jaundice. If patients present late, these symptoms will appear. Initial treatment requires decompression of the liver with percutaneous stents. This initial first step to reduce the risk of cholangitis and define the injury, although some patients can be treated with dilation and stent insertion by ERCP. Most will require hepaticojejunostomy. We have many types according to Bismuth Strasbourg classifications. We have many types of injury. Now, I will explain in detail the magnetic compressions and anastomosis for treatment of post-cholecystectomy biliary strictures, MCA. MCA represents a new paradigm for benign biliary strictures, which occurs post-surgery, that are difficult to treat with conventional methods. It can be utilized to perform biliary anastomosis in post-cholecystectomy, treatment of bile duct injury without a major surgery. The aim of these techniques is to avoid surgery. Further long-term studies are required to establish its durability. Biliary strictures. Incidence I mentioned already. This is the case this is my patients. 32-year-old woman presented with jaundice 3 weeks following laparoscopic cholecystectomy for gallbladder stools. On abdominal ultrasound, the bile duct were prominent, with an aberrant cutoff at the level of the mid-common bile duct, suggesting strictures. Initial diagnostic. ERCP done during the endoscopic retrograde cholangiopancreatography. ERCP, the common bile duct, showed a complete cutoff at the mid-level and the occlusion couldn't be crossed. There was cutoff at the mid-common bile duct. We tried to pass the guide wire. It was impossible to pass through the stricture. This is ERCP, which reveal here there is clips put during cholecystectomy. There is cutoff here. There is cutoff and prominent common bile duct. So after ERCP failed to pass the strictures, we decided to do BTBD. One on percutaneous transhepatic cholangiogram, there was complete occlusion also of the mid-common bile duct. The stricture couldn't be crossed also. The diagnosis of complete common bile duct transection with 1.5 centimeter long bile duct stricture was made on both ERCP and BTBD. Both procedures, ERCP via endoscopic route, we couldn't pass the transections, the strictures. Also via percutaneous BTBD, percutaneous transhepatic cholangiogram, also we couldn't pass the strictures. Actually, there was occlusion. This is the images of magnets, miniature magnets. It should be less than the outer diameter of the magnet should be fit with the duodenoscope. We can use many types of magnets, ring magnets or rod magnets. This is after doing BTBD and putting magnet above the transactions, and this is the magnet. This is 3 millimeter magnet long and 10 millimeter in length by ERCP. This magnet set by BTBD. This catheter of BTBD, and this magnet set by ERCP by duodenoscope. So the magnet was placed at the proximal end of the obstruction, below obstructions. Other magnets set above the transactions. Now we have transaction, we have magnet above the transaction and magnet below the transactions. This attractive force of both magnets will compress the tissue between each other and cause controlled death, tissue death. This will lead to fistula. When there is fistula, we do daily monitoring by fluoroscopy and to see the approximation of both magnets. These clips put during cholecystectomy, during surgery. Now we have transection here. We couldn't pass these transections by ERCP by endoscopic route, and by PTPD. Both methods failed to cross this transection. So we put one magnet above the transection, other magnet below the transections. And we monitored by fluoroscopy every day. We monitored the approximation of these both magnets. We need these both magnets to attractive to each other and compress the tissue between them. When this compression occurred, this will lead to controlled tissue death, necrosis. This will make us able to pass the guide wire, then to pass the, as we will see, to pass the stent. Yes. These two magnets attracted to each other. In day 4, therefore, it's attracted to each other. Then after attracted, we tried to pass guide wire. Guide wire passed successfully. We insert multiple plastic stent. As we know, we use plastic stent in benign strictures, not metal stent. Also here, this is duodenoscope. We see this duodenoscope, this attractive magnets, these plastic stents, which offer a good drainage, biliary drainage. Also here, also we see this. Additional plastic stents were placed monthly via the endoscopic route for 6 stents. Every month we were putting more stents and more stents to cause dilatation of strictures till we reach to 6 plastic stents. As we see, plastic stents very good as known, very good for benign strictures. We use always plastic stents for benign strictures. For malignant strictures, we use SEMS, self-expandable metallic stent. But here in benign strictures, no rule for metallic stent. We insert 1, 2, 3, 4, 5, 6, sometimes more than 6 stents. These plastic stents will cause dilatation of the biliary duct, of the common bile duct. As we see, multiple plastic stents. Magnets retrieved later. The magnets retrieved during the next endoscopes were retrieved by Rath forceps, foreign body forceps, and the patient was followed up every 2 months with liver function tests, bilirubin, total bilirubin, liver function test. Cholangiogram was normal. This is a clip. This is a clip which put on post-cholecystectomy. And strictures almost resolved, no strictures. The patient was followed up every 2 months with liver function tests. Cholangiogram in the sixth month showed complete resolution of the biliary stricture. All 6 stents were removed. Conclusion. MCA, magnetic compression anastomosis, is a promising non-invasive technique. Non-invasive. Actually, it's innovative, non-surgical. Avoid the patient massive surgical. Hepatico-jejunostomy is a promising non-invasive technique for difficult benign biliary strictures. It performs biliary anastomosis to repair post-cholecystectomy bile duct injury, eliminating the need for major surgery. MCA represents a new paradigm for benign biliary strictures that are difficult to treat with conventional methods. Because some patients have coagulopathy problems, some patients have comorbidities, they can't obtain major surgery, high-risk surgery. some patients have comorbidities. So these techniques represents a new paradigm for benign biliary strictures that are difficult to treat with conventional methods, which means surgical. Long-term studies are needed to confirm its durability. And thank you. If there's any questions, please.
You have a question for Doctor?
Yeah.
In how many times occurs the history?
Some patients after 4 days, some patients take 7 days. There is no definite time, but we have
The patient stay in the hospital and you want your control. Well,
first and second day. Actually, ERCP considered daycare operation, daycare procedures, not major surgery. But the first 1, 2, 3 days, we can keep the patient in hospital. But I can't decide what time exactly the fistula will occur. Some patients after 4 days, some patients after 7 days. It's depend on the comorbidities of patients. If the patient is diabetic, if the patients have comorbidities, young patients different from elderly. This variant, not fixed. But these techniques, the aim of this procedure.
imagine you use for control is only radiology or
Only fluoroscopy only. We monitor the attraction of both magnets only by fluoroscopy. No need for interventional procedures during monitoring attractive.
Why you don't use the SEMS full regular?
Yeah, same self-explanatory metallic stents. It's good for only malignant.
But
because it's flexible. Same metallic stent, self-explanatory metallic stent. It's good for malignant, but no rule for, in general, no rule for benign. In benign strictures, we need dilatation. We need plastic stent. Plastic stent, not expandable, fixed size. When we insert more than multiple, this this axiomatic information. We use multiple MPS, multiple plastic stents for the benign biliary stricture. But what I would like to clarify here, this technique weighing sometimes if there is stricture in biliary by that post cholecystectomy, sometimes via or during ERCP, the guide wire will pass. But this technique only for transection. If there is transection due to diathermy injury or due to clips, this is very good as an alternative method an alternative to surgery, to major surgery. The idea of this procedures very simple. With magnets, one above the transection, other below the transections, it will attract to each other. This attraction, strong attractions, will cause necrosis, tissue necrosis. So will cause fistula. This fistula we can pass guide wire. After passing guide wire, we can pass a plastic stent. Plastic stent we can pass during endoscopic route, ERCP, during duodenoscope. Also, we can also set plastic stent during BTPD, same time. If we don't put the plastic stent via duodenoscope, via ERCP, we can try via BTPD. But what we need to pass the guide wire. And this is important issue. Yeah.
First of all, I want to thank you for the success of your treatment, and the patient will be. We're very happy for the patient also. So you avoided major surgery for her. Just, I know it's new technique, but on your experience or maybe on that I project, is there what's the kind of complications you have? You could have perforations, or maybe the two devices they will reach other it can be spontaneous. I don't know.
Thank you for your great questions. First of all, this is not my technique. This is actually Japanese technique. I mentioned the doctors of yeah. There is conditions. If the transaction before to do ERCP and PTBD, we do ultrasound, we do MRCP, we do sometimes CT scan. If the structures very long, the extension of structures very long, in this case we can't use this technique because despite the magnets very strong, this nodium earth magnets, despite the strength of these magnets, if the extension of structures very too long, through 3 centimeter, 4 centimeter, we can't use this technique actually. This technique used if the extension of structures is, for example, 1.5, around 1.5 centimeter. Sometimes the extension, if not diathermy, because sometimes the clips set wrongly, instead of putting on cystic duct, it put on common bile duct. The distance will be very short, 3-4 millimeter. But the problem, if the injury by biliary duct injury is related to diathermy injury, sometimes the distance is too long. The magnets couldn't attract. Actually, this new technique, but the Japanese scientists invented it in 1990. Actually, the first case was for a liver transplant patient. It's a new, but not a new, too much new. There is some studies, for example, for Dr. Malay Sharma, for example, he has also published study. Also, the Japanese doctors who invented also, he has study. There is many studies for this technique. It's safe technique, but for complications, actually, like any irritation of biliary strictures, perforation could be occurred, but perforation, I don't mean fistula. Fistula, definitely we have penetration. We need this hole, this necrosis. But sometimes, like any irritation of biliary, for example, bile leak, but at the same time, this technique for treat bile leak, also bile leak could be occurred, but rarely. Also, perforation, for example, during attempt to pass guide wire, sometimes instead of pass from proximal segment to distal segment of common bile duct, could be pass outside during the abdominal cavity. So this, like any complications of ERCP or PTBC.
Thank you so much for that.
Thank you.
We're running short on time.
So I just want to ask you, the plastic stents, are they absolutely, or what happens? You remove them for 10 years? That's my first question. And then this technique you described, is it laparoscopic? As in, you have magnet on top, magnet below. Is that laparoscopic or is it open surgery?
No, at all, not laparoscopic. Endoscopic. PTBD, percutaneous transhepatic, PTBD, cholangiogram, and endoscopic. No rule for laparoscopic. This procedure, this invented technique is to avoid laparoscopic, to avoid surgery. And actually, a correction of post-cholecystectomy biliary duct very difficult by laparoscopy. Most surgeons open surgery. I have patients since 4 months. The strictures was too long. We couldn't do this technique. We do open surgery.
And so, are they absorbable or do you need to remove them after
No, removing the stent, very simple issue. Not a big issue.
He's asking if it's removable after a certain period of time.
Yeah, removable, of course. Of course, plastic stent removable. Otherwise, it will occlude it. It will cause occlusion itself. As we know, plastic stents, longevity, the long time, what time we can let the plastic stent? 6 to 8 weeks, for example, 1/2 month, 6 weeks, medium, 6 weeks. Otherwise, plastic stent itself will occlude it. You can't let plastic stent permanently. This in contrast to metallic stent. Actually, metallic stent permanent. You can't let especially in malignant regions like cholangiocarcinoma or other. So plastic stent we can't let the plastic stent inside combine that for a long time, more than 6 weeks or 2 months, 6 to 8 weeks. We have to remove it definitely.
Thank you so much. And going to bigger roles. And please join us for a photo shoot, all of us outside. Please join us outside, please, for a photo shoot, and then we will go for lunch break, and we will be back at 2:05. 365. Yeah, the lunch is in the 365 restaurant. Okay, it's here at the Yes, but it's more important.
Still?
So it's impossible.
And how many patients do performs
actually this injuries fortunately very rare.
right, but it's very good for the patient.
You know, it's my first time. So because it was my own story. Yeah. Yeah. Look, there is a text here. Okay, so if I want to move from slide to slide, this one, you can this one slider. No, it's okay. I can use this one. Yeah, this one. Thank you. Thank you so much.
I just want to go back to my presentation for a photo.
Yeah, sure.
Copy you because I did not get a good one. Right? It's okay. They keep calling me for photos. Yeah. You have a good mobile. Just tell me where I should stay. Okay, like this. This one, it has to be there, right? Yeah. You can go to the side. Okay. Today I want the last one. This one. Good. Yeah, keep on this one. Take one layer. Thank you so much. Sorry to bother because they don't understand the situation here. Okay, I think before because my airdrop was only contact centers. Now it's okay, the previous one. Previous one. Got it. Thank you so much. Your name? Yeah. You're living in Saudi? Yeah. How long? Or like settled over there or you are in India? I'm in India also. I'm just working there. 20 years only. Okay.
But we wait until the. I can put it here. Is it working? Yes. Hi, everyone, and welcome back from the break. I'm introducing myself. I'm Dr. Miriam Sabir, PhD from the University of Montreal in Canada, and founder of Clean Bridge CRO based in Dubai and covering all GCC. So it's my pleasure to chair this next session featuring experts in pediatric health, nutrition, and development. So we will proceed with this speech with Dr. Muhammad Ubaida.
Yes, assalamu alaikum. Hello everybody. Welcome back after the break. I hope you are not sleepy after that lunch. I know that you are from different backgrounds. Today I will talk you about the journey of the process of a product development or a drug development, talking about what's going on for the products that you are using for your patients when you are dealing with certain drugs. How these drugs have been developed. Usually, all these medication has been started from the science called the drug discovery, where we have new chemical entities that will be further developed into different products and then can be used. But in many cases, this drug discovery science will end up by having certain molecules that might have some limitations in terms of solubility, in terms of toxicity, or sometimes in terms of stability. So many therapeutic molecules have been currently, they have limited use because of these problems. And here it comes the concept of nanotechnology, what has been introduced several years ago that can solve these problems, that can increase the solubility of medications, can increase the stability, or can reduce the toxicity of these medications. If you're talking about nanoparticles or nanotechnology, we have many types of nanoparticles that can be used in field of science called drug delivery or nanotechnology drug delivery. Many of them have been investigated with several molecules. The most commonly used types of nanoparticles, they are lipid type nanoparticles like liposomes, which has been introduced to the market several years ago. We have also another types of lipid nanoparticle like lipidoid, niazones, and micelles. Now, not only these nanoparticles will improve the characteristic of new molecules, but they also help in something called targeted delivery, where we can control where these therapeutic molecule can be distributed within the body, where we need this molecule to be accumulated in the diseased area only and avoid the healthy tissues. And, you know, for example, the most common side effects for chemotherapeutics is their toxicities, but the use of these nanoparticles can help in accumulating this molecule into the target disease. Also, with the emergence of nanotechnology, there has been also a new, with the different technologies like biotechnology and other omics technology, also have been developed something called personalized medicine. Now, it's not now the science of drug development is going beyond rather than just using drugs for treating disease towards what's something called personalized medicine. Now, the traditional use of medication that we are using, the same drug for different patients. Same drug can be used to treat different patients that have the same, let's say, the same condition. But indeed, different patients, they have different gene map, they have genetic map, different gene characteristics. So that's why we have different variability between medications in terms of efficacy, in terms of side effects. Now, the evolving of the biotechnological techniques and the omics and the gene mapping have allowed to something called personalized medicine, where we are using the genetic structure of the patient to treat the disease, rather than just giving a random drug for treating certain condition. Now, this can be utilized using something called the nucleic acid therapeutics, or what's called through the use of genomics sequencing, where we can use certain therapeutic nucleic acids to treat certain diseases based on the genetic mutation of the patient. Sometimes we have two patients with lung cancer, for example. They both have lung cancer, but they have different mutations. So we cannot just treat them as lung cancer patients. We need to look for what are their genetic structure, and then we can use the personalized medicine. Now, this can be achieved again using something called a nucleic acid therapeutics, where the nucleic acid can help study first the genetic structure of the patients, and then can be used to develop certain drugs to treat the disease. If you look for the types of nucleic acids, we have 5 major types of nucleic acids. We have something called antisense oligonucleotides that will result in a cleavage of the mRNA. Now, the way the protein will be synthesized is we have the DNA that will be translated into messenger RNA, and then messenger RNA will be translated into protein within the cells. Now, antisense oligonucleotide will result in breaking down the messenger RNA, preventing the expression of the protein or preventing the formation of the protein. We have the RNA inhibitory, which using the siRNA, also it will result in breakdown of the mRNA, preventing the protein expression. We have the aptamers, which prevent the binding of the protein to its receptors. We have also called mRNA translation. We can use mRNA to enhance the production of certain proteins. And we have something called the CRISPR, which is the gene editing that works on the DNA level, where we can modify the mutation at the DNA level. These types of medication have been started long time ago. 1972 was the first ever research about the use of nucleic acids by the different researchers. Antisense oligonucleotide have been proposed in 1978, going back to the 1990, where the concept of mRNA encoding has been introduced. In 2000, the very novel technology called RNA inhibitory has been discovered, and the scientists have won Nobel Prize for this discovery. Then we have the mRNA therapeutics that has been first ever introduced in 2019 in COVID-19 pandemic. And currently we have many medication already approved in the market that are based on nucleic acid therapy for personalized treatment. Now, the challenges for these drugs or these medications, that they are highly hydrophilic, they are quite large in size, and they have very limited stability. So that's why using this medication, it's not an easy forward or not straightforward process. It's very, very challenging. Now, to overcome these challenges for best use of these medications, they have different proposals for use of these medications. Some of them, some of the scientists have proposed using chemical modification to modify the structure of the nucleic acids. We can also use something called cell-penetrating peptides that enhance the penetration into the cells. And we can use also nanotechnology. We can use a drug delivery system in order to improve the stability of these nucleic acids and improve their penetration to the cells. Now, different types of carrier have been proposed for nucleic acids, like viral vectors, like liposomes, like other types of nanoparticles have been proposed and investigated. And currently, we have many approved drugs in the market that many of them, they are based on nanoparticles. They are nucleic acid-based drugs for targeting different disease. You can see also here, for example, we have some of them, they are based on siRNA or short interfering RNA. Some of them, they are based on messenger RNA, antisense oligonucleotides. And we have many, many others with under clinical trials. So we have many types of drugs that are currently being developed for different indications. Mainly, they are for the concept of personalized medicine, where instead of just treating the disease randomly using general medication, the same medication for different patients, we are detecting what are the genetic map of the patient and then design the type of medication based on the genetic requirement of the patients. One of the interesting technique that has been proposed extensively for personalized medicine is the siRNA or the RNA inhibitory. RNA inhibitory is one of the techniques that will result in a breakdown of the mRNA, preventing the expression of certain proteins within the cells. So, for example, if we have cancer, and this cancer characterized by overproduction of certain protein, then we can design siRNA to prevent the expression of this protein. This will limit the growth of the cancer cells, eventually lead to the death of these cells. So simply, these siRNA molecules will bind to something called the RNA inhibitory silencing complex, and then they will be complementary to a specific structure of mRNA. This will lead into the breakdown of the target mRNA and the prevention of the protein expression. So eventually, no protein will be expressed, and this is very interesting technique to treat different types of cancers and other types of disease. So simply, or theoretically, to treat diseases using this technique, we need to design siRNA and introduce this siRNA into the cells. And then this will bind to the RNA inhibitory silencing complex here, and then will prevent the expression of the protein. So this is theoretically, we can design any siRNA of interest because we already know the structure of the protein. We can work backward, and then we can determine the structure of the siRNA. Then we can synthesize the siRNA. But the challenge, how we can deliver this siRNA into the cells, how we can get this siRNA into the cells, because the story is not an easy. siRNA, they are highly hydrophilic molecule. They are highly negative. They are large in size, and they are unstable. So we cannot give them as an IV injection, for example, for treating of liver cancer, for example, or other types of diseases, because they will be within 50. If we give siRNA to the patient as an IV infusion, for example, or injection, it will be eliminated out of the bloodstream within maximum 15 minutes because it's highly susceptible to nuclease enzyme. So with this technology and in combination with the nanotechnology, many drugs have been translated into the market that are based on siRNA. The first drug that has been introduced to the market is a drug called Patisiran, or the Impatro, which has been introduced in 2018 for the treatment of certain neurodegenerative disorders. It's based on siRNA using liposomes, and this drug was the cornerstone for the development of COVID-19 vaccine 2 years later. We have also other types of drugs that also based on siRNA that can be effectively used, but they are using different techniques, like it's called conjugation or glucose conjugation. Patisiran or the Onpatro is the only drug in the market that's using siRNA loaded into nanoparticles called liposomes. Because of that, we have many research currently being done for investigating different options for the delivery of siRNA into the target cells for personalized medicine. Because the problem with liposomes, they are quite expensive. They have certain challenges in terms of stability. So in our lab, we decided to investigate, can we design different types of nanoparticles for delivery of siRNA or for nucleic acids to suppress or knock down certain protein. So we started by designing something called niosomes, or nanoparticles based on surfactant rather than based on liposomes. Now, the way these liposomes and niasomes, they are prepared in a technique called self-assembly, where when we expose these molecules or these components to aqueous media like water or buffer, they will rearrange themselves into like nanoparticles or a bilayer nanoparticles. And this will be used as a delivery system where a drug can be loaded inside or on the surface of these nanoparticles. So going back to the in patro, it's using these types of nanoparticles. They are using liposomes to deliver the siRNA into the target cells. And again, COVID-19 vaccine, they are using the same technique, but COVID-19 vaccine, they are using mRNA rather than siRNA. Now, going back to niasomes, they are types of nanoparticles that are referred to as a bilayer structure nanoparticles. They are called versatile because we can load any type of drug inside these nanoparticles, either within the membrane bilayer or within the aqueous side of these nanoparticles, and they can be prepared using different techniques. However, for liposomes and for other types of lipid nanoparticles, in order to prepare them for large scale for the patient, you need an industrial technique. You need an industrial method that can be used for the preparation. Most of these particles, they were prepared using a very traditional technique called thin film hydration method, where we have our components dissolved in organic phase like chloroform, and then you need to evaporate this solvent and then hydrate it to prepare large particles and do size reduction in order to prepare your particles. This technique usually takes hours to prepare the particles. So imagine if you want to prepare large-scale production, this will be very, very challenging for the preparation of these types of nanoparticles, and you have very limited control on these particles. Now, one of the newest techniques that has been proposed for the production of particles called microfluidic mixing, which is another technique used in the formulation of nanoparticles, where we just mix the components of the nanoparticles with the aqueous phase in a specifically designed microchip or microchannel in order to self-assemble these nanoparticles under very controlled conditions. In this technique, we can prepare the particles within seconds instead of spending hours for preparation of the nanoparticles, and this will result in improving the control over these nanoparticles, controlling the size, the distribution, the stability, and we have proper production at larger scale. Now, we are applying these types of technique for both technique for the preparation of nanoparticle in our laboratory. We have very well established microfluidic system to prepare the nanoparticles at very controlled conditions. We have several articles published for the production of these nanoparticles and what are the parameters that will control the production, which I believe is purely formulation aspects that anybody working on formulation needs to take these into consideration. Now, preparing these nanoparticles using microfluidic, we have to control several parameters, like how much we are mixing from both components, how much aqueous phase, how much lipid phase, what are the speed of mixing, what are the flow of mixing, what are the temperature, what are the concentrations. So all will control the characteristic of these nanoparticles, or this nanoparticle will be, the characteristic will be determined by these factors. Also the type of solvent that we are using, either we are using, for example, water, buffer, phosphate buffer, Hepa's buffer, tris buffer, different types of organic solvent, methanol, ethanol. So all this will need to be controlled for the production of these nanoparticles. Eventually, again, you can prepare these nanoparticles very nicely, very precisely, and you can control how much the size of the nanoparticle you need, how much the charge of these nanoparticles you want, and this can be used as a tool for the delivery of nucleic acids. So if you go back to the siRNA, these nanoparticles can help in translating these nucleic acids into the true product. They can increase the stability and can help in something called personalized medicine. Now, next, we decided to see how can we deliver siRNA using our prepared niasomes. So we need to prepare the niasomes for the delivery of siRNA, and we need to test, can we really prepare certain formulation to knock down or to prevent the expression of certain proteins using this technique. So we have, these are the settings that we have. We have prepared first different types of formulations for nanoparticles. And then we got certain types of cancer cells. It's type of lung cancer cells. But these lung cancer cells, they are genetically modified to produce a protein called GFP, or the green fluorescence protein. Now, these cells, they are not normal cells. They are cancer cells, but their genetic structure has been modified to produce a protein called GFP. It's non-functional protein, but it will give a fluorescence. And then we have designed certain siRNA to target this GFP, to knock down the GFP, to prevent the expression of GFP. We loaded this siRNA into our niosome nanoparticle, and then we treat the cells with this formulation. If the formulation was successful, then we would see a drop in the signal of the GFP. If the formulation was not successful, then the cell will continuously produce the GFP as usual, and the signal would be the same. So these are the cells under fluorescent microscope. They are producing the GFP, and then we managed to achieve significant knockdown or significant reduction in the expression of GFP using these types of cells. We have prepared several formulations. We measured the protein expression with a technique called Western blotting. So we have measured how much concentration of GFP we have achieved, and we can see here the level of the GFP has been significantly reduced compared to the controls or compared to the untreated cells. So by using these nisomes that have been prepared with microfluidic mixing, then we can specifically knock down the protein. Again, this is non-functional protein. It's not naturally existing. These are flow cytometry results where we can see the shift in the signal, so which can prove the knockdown of the protein from the cells. Next, we wanted to investigate this in vivo, because this was an in vitro experiment on the cells. We wanted to investigate, can we give these to animals, for example? So we got another type of cells called F16, which are skin melanoma cells, but they are genetically modified to produce a signal called luminescence. And we designed a siRNA to target this luminescence signal, and we have induced skin cancer in mice. Then we have related these siRNA into our nanoparticles, and we injected into the mice that are producing this luminescence signal. And we can see with the treated mice, the level of the signal has been significantly reduced compared to the untreated or to untreated mice, or the mice that has been treated with free siRNA, which means also these nanoparticles, they were effective and capable of knocking down the protein in vivo, in animal model. However, both GFP and luminescence, they are non-functional, so they are not naturally existing. And if we are intended to treat cancer or to treat certain disease, we need to look for certain protein that are highly expressed in the cells and trying to knock down this protein. So we are looking for different types of a protein, and one of the interesting proteins that are usually overexpressed in cancer is a group of proteins that are called protein. C-MYC proteins usually one of the proteins that are required for the proliferation of the cancer cells. So without the C-MYC protein, the level of growth of the cancer cells is very, very minimum. So we have got the breast cancer cells, and we determine the structure of C-MYC protein within this that are expressed in the cancer cells, and we designed a siRNA to knock down this C-MYC protein. We have prepared very nice particles, as you can see, very uniform particles, and we encapsulate the siRNA that can knock down the C-MYC protein into these nanoparticles, and we treat the cells. This is the structure of the siRNA. We can see we can also achieve very high reduction in the level of C-MYC using these nanoparticles that are encapsulating the siRNA. So as a proof of concept, we have proven that the use of these niosome nanoparticles, or these nanoparticles that are prepared using microfluidic mixing, can knock down real protein. Now, the idea with personalized medicine that nanoparticles, they are like an envelope, and the siRNA is like the message inside the envelope. So you can easily change the message with what you want using the same envelope. So we can use different types of nucleic acid within these nanoparticles to target any type of a protein of interest in treating of disease. Now, these naosomes or this microfluidic technique for the production of nanoparticles can be used also for other types of medications, not only for nucleic acids. We have investigating these for encapsulating of other water-soluble molecules like doxorubicin. We have also the atenolol, and we have proving it for other types like curcumin, like a natural, which is one of the natural products. If you look for one of the first ever introduced drug into the market that's based on nanotechnology, it's called Doxil, which is doxorubicin loaded into the liposomes. It's different than the normal doxorubicin, where the doxorubicin is encapsulated into these nanoparticles, the liposomal nanoparticles. It's been introduced into the market in 1994. The first ever drug has been introduced to the market using nanoparticles. However, this was prepared at that time, was prepared using the traditional method for preparation of the nanoparticles. 6-7 years later, after the introduction to the market, the company has received the black box warning from the FDA that there are variability in between different batches for these nanoparticles. Because again, these nanoparticles, they were prepared using the traditional method. So the control on the production process was very limited. So that's why the level of side effects and variability in responses between patients was very high. In our lab, we decided to prepare our own Doxil, our own nanoparticle encapsulating doxorubicin. So I prepared these nanoparticles using something called pH gradient nanoparticles to see, can we encapsulate doxorubicin within these nanoparticles to prepare niosomal doxorubicin. Now, this is based on, again, on microfluidic technique, where we have prepared the nanoparticles by mixing different types of lipids with aqueous phase. And these nanoparticles are prepared by using a technique called pH gradient. And what do you mean by pH gradient? We have prepared the particles that have an external pH of 7 and an internal pH of 4. So we have different pH within the same nanoparticles. So again, this is the structure of nanoparticle. Inside the nanoparticles, we have pH 4, and outside medium, it has pH of 7. Normal physiological pH. And the reason for that, when we add the doxorubicin to this nanoparticle, it will penetrate inside the nanoparticles. When it gets inside the nanoparticle, it will face the acidic environment, the pH of 4. It will get ionized and trapped inside. So it will not be released, or it will not be a leak outside of the nanoparticles. The nanoparticles were very small, very nice. We have investigated these nanoparticles on different types of cancer cells, and we can manage to achieve high reduction in the level of the IC50 of the doxorubicin when we are using these nanoparticles compared to the free nanoparticles. We investigated also these types of nanoparticles with other molecules like atenolol, for example. It's a hydrophilic molecule. And the problem with hydrophilic molecule, they are very difficult and very challenging to be encapsulated in nanoparticles. And we managed to achieve very nice formulation, very nice structure of these nanoparticles for hydrophilic, and again for hydrophobic molecules like curcumin. Curcumin is a natural product that has been known since long time. It's been investigated for the different indications like antioxidant, antibacterial, anti-cancer, and also we managed to produce very nice formulation using microfluidic technique for the encapsulation of curcumin for the different purposes. So this techniques for the production of nanoparticles, they allow for the encapsulation of any targeted drug, any intended drug. It can solve the problem of, or boost the problem of challenges that are faced the drug development, because many of the drugs that are currently under development, they are facing certain limitation in terms of, again, solubility, stability, whatever. So this technique can help in improving in overcoming the challenging of the production. Now, if you go back 4 or 5 years ago in the COVID-19 pandemic, imagine that we need to prepare COVID-19 for the entire world vaccine using the traditional method of preparation. It's definitely impossible. But these vaccines they are developed by microfluidic technique. That's why they have been developed very quickly. Within 6 or 8 months, we have plenty of stock of this COVID-19 because the experience is there. we have the nanoparticle, the carrier, and we just change the envelope, the message. We just change the messenger RNA within these nanoparticles, and then they would be able to prepare vaccine for the entire world in very short period of time. So this technique can be very helpful in improving the production of drugs, can be very helpful in the process of product development, that we can manufacture, purify, and then sterilize this preparation very easily, and then can be given to the patient in very short period of time. By this, I would be thankful for your listening. Thank you very much. I would like to acknowledge all my colleagues, my partners, and my collaborators in research in different areas of the world. And thank you very much for your listening.
Thank you.
Can use this one.
So thank you, Doctor. Kindly come forward for your certificate of photo. Thank you. So thank you, everyone. Our next keynote speaker is Mr. Tahani Al-Surayyi from Kuwait Institute for Scientific Research. Her presentation title is From Isolation to Implementation, Developing Effective Probiotic Formulation for Livestock. Please welcome Madam Al-Surayyi.
My name is Tahani Al-Surayyi from Kuwait Institute for Scientific Research. Actually, I'm going to talk a little bit about probiotics in another way, not like my colleague here when they're starting to talk about the medical side. First of all, we had lots of projects related to probiotics in the animal sectors. I had been worked in the probiotics since 2006, and I have a lot of experience regarding the isolation and characterization of the probiotics as a brief introduction to my presentation today. From isolation to implementation, developing effective probiotic formulation for livestock. Yeah. Today, I will give you a brief introduction about my presentation, and I will talk about the isolation of probiotic strains, the challenges and limitations, and also I will go for all these points. And some points I will explain more regarding what the aim that I am focusing on today. Nowadays, all people are looking for alternative way to get rid or to reduce the use of the antibiotics. That was our aim, or the first point let us starting for doing our projects related to the antimicrobial resistance or the probiotics. Actually, probiotics not only can be the sustainable way to replace the antibiotics, it also enhance the gut health, reduce the pathogenics, and also improve the nutrient uptake by the enzymes. Probiotics, as Fuller told us, it is the living microorganism which, when ingested by human or animals, can beneficially influence the health by changing the intestinal flora. So we are what we eat. Maybe all of us heard about these things, this statement before. We are thinking of what we are eating. We all eat either the product of the cattle or the meat itself or the poultries, the eggs. All these things we have to make sure that they are clear and it is safe for us. Actually, today maybe I will explain more about my main project, which is the dealing with the cattle now. I have been isolated more than 80 strains. These strains were screened for their probiotics. And after that, we decided to choose 6 from these strains to be used as more or extra experiment to produce one product for giving as a feed supplement for the cattle. The weather in Kuwait, as you know, is too harsh and the environment is too difficult for the animals to grow. And they always suffer from the crowded because they are hatching in a place crowded. And the type of the food is also a little bit expensive for the farmers. So we are always seeking for another way for the food and as well as for their health. We want to make sure that we have our own product that can raise the immunity system for these animals as well as the food type. So I will tell you why I said food types, because I enter also the probiotics in another way for the food supplement. These isolates have been characterized in more than one biochemical reaction. We are looking for their tolerance to the bile salt and the acidities, antimicrobial activities against the certain pathogens, and also their ability to adhesion to the epithelial cell, their enzyme production, immune modulation properties, and also genomic screening for virulence and resistance gene. The mechanism of probiotics in the cattle or the animals, they're either competitive in the exclusion of the pathogens, or they can produce antimicrobial substance. Another way, they can enhance the intestinal morphology by increasing the good or beneficial bacteria, also stimulate the local and systemic immunity. The study that I did now during this project was to evaluate the efficiency of using the combined strains to serve as a probiotic product and find the optimal isolates and the lactic acid probiotic combination and the compatibility test result between them. Based in vitro assessment and the growth characterization and safety for cows, the lactobacillus strains were selected: Pediococcus, Pentococcus, Lactobacillus fermentum, Ramos, Plantarum, Brevius, and Casa. Before we started to use these strains, we have to make sure that they are compatible to each other, because I'm going to use a multi-strains, so we have to make sure that they can be compatible to each other. And at the same time, they can get rid from the pathogens that we are focusing on, especially the Salmonella Escherichia coli. So by the antimicrobial test, we find that they can they can resist the presence of the pathogens. At the same time, they can produce antimicrobial against these pathogens, and at the same time they are compatible to each other. These 6 strains, they can be combined to each other, and it can be one multi-strains. So at the end, we will find out exactly the dose that we have to put, because we are in the study now, that the multi-strain probiotics can be good or more beneficial for using rather than the monostrains. And they can be competitive to each other with a high percentage. the method now in the middle, we are going to, or we are started now using this mixture of the multi-strains in the dairy farm by two way, directly feed with their food or by water supplement. But there are more than one techniques to apply these probiotics for the cattle. For the in vivo testing and validation, we have to find out the small scale trials to evaluate the gut colonization and the growth performance metrics, feed conservation, immune markers, mortality and morbidity test for the cattle during this test. So based on it, we can go for the large scale field studies reflecting the real condition that we face. And at the same time, we are recording all these data, all statistical analysis as has been recorded. For the scale manufacturing, the commercial scale manufacturing, there are more than one challenge, is that maintaining the cell viability through production, preventing the contamination, achieving the number of colony forming unit of bacterial count. Is it interfere with the antibiotics or feed additives, other feed additives for the cattle? And the management of the farm practice affecting the outcome. Also, we have to control the fermentation, standardize the drying systems, also the cold storage condition, and quality control for each batch has to be done. Also, when we want to implement it in the farm, we have to introduce the probiotic gradual. We shouldn't give it as a one time. We have to feed it gradually, monitor the animal behavior, performance metrics, health indicators, and also we have to train all the staff that's working in this field for the dosage. Now, why I told you we do it in both ways, either by direct feed to increase the immune system or to get rid from the antibiotics. Also, we are lack of this animal feed. So we were looking for the another things which is used, the silage. Silage is depend on the probiotics, or not only depend, it is additives for the silage production. Maybe you heard about silage. Silage is a way to make animal food during the like the residues we have in there is a lot of waste residues. And instead of throwing this residues, we can reuse it by adding this probiotics in the harsh condition, and we can use it when we need it for as a food for these cattle. In the cold area, they use it during the freezing time. For us, we will use it, I think, a whole year except maybe 2 or 3 months, because we lack the grass, the grassing that this is another project that have used this also multi-strains probiotics in order to make the silage. So we can do it in both. We make the silage and we increase the immune system of the animal. As I explained for you, the green residues plant in Kuwait as a waste, they dumped or they burn, we cannot use it, but also they cannot trust to use it directly for the cattle. It might not favorable by the animals. It might has a high content of the cellulase or fiber with low protein level, so the animal will eat something not healthy for them. But they usually, the farmers, they abroad the silage from other countries, and it is expensive. So why we do not use it from the residuals? And it was maybe 23 years ago, successful project that produced the silage in Kuwait, in Kisar also, from the waste. So we will use it again, but with our probiotics that we are isolated from locally farms. Also, I have been deal with the poultry, and I have a novel probiotic product from the multi-strains regarding the poultry animals, poultry feed. Not, yani, this is an older project, and I succeed in get this product. And it has it supports our poultry industry in Kuwait, and also it can be used for all the closer yani countries which is similar to our condition, our climate condition. Alhamdulillah, we succeed in this multi-strains with poultry. So we are looking now for the same things with the cattle. I'm in the middle of this project now. Thanks. Thank you.
Thank you so much. Kindly come forward for certificate and photo. Thank you. Thank you so much. So our next speaker is Madam Katarina Lijovic from Croatia. Her presentation title is The Impact of Stress on Gut Health. Please welcome Mr. Lijovic.
Microphones. Hi, everyone. For sure, you all had that moment when you are about to have an important job interview, or you're facing some exciting news, or you're about to give a public speech, and all of a sudden, everything changes in your gut. You feel it in your stomach, you feel it in your intestines. Maybe you have those stomach knots, maybe you feel nausea, or maybe you have this sudden urge to run to the toilet. And that is actually how quickly stress can act on our gut. Or I will give you another example. We all had now a beautiful lunch. Maybe we have chosen something from a healthy salad bar, but we don't feel that something is right with our digestion. We just feel that something isn't working well down there. That's also stress in action, because if we maybe have our lunch eating very quickly, or if we are going through some stressful period, our stress completely shuts down our digestion and gut functioning. Stress is something that we have to live with and cope with. Otherwise, the stress can really took over of our life, and it can definitely take over of our gut health. And today we will explore the mechanisms behind the impact of stress on gut health. There is a lot of research done on this topic, and there is so much that we could dive into today. But in this short talk, I will cover the most important and evidence-based mechanisms behind the impact of stress on gut health. There are different types of stress, of course, and my presentation is going to focus on that psychological, emotional, or psychosocial stress. So what is actually happening here? How does stress make our gut react this way? Why are we having this sudden urge to go to the toilet? Okay. To better understand what is happening, we have to talk about this bidirectional communication between our gut and our brain, and for sure you're all familiar with this one. It's our gut-brain axis. I like to call it a super speed highway, because everything that is happening in our gut is impacting our brain, and vice versa. Everything that is happening in our brain definitely affects our gut. There are numerous pathways involved in this communication, such as neural, endocrine, immune, and microbial. And the key players in this communication are vagus nerve, immune system, endocrine system, neurotransmitters, and of course, hypothalamus pituitary axis or HPA axis. So when we are feeling stress and when the stress first hits our body, we are entering in our sympathetic nervous system mode and HPA axis is being activated. So let's unpack what is actually happening over there. So when we are facing an acute stress, the first thing that happens is that hypothalamus releases corticotropin releasing hormone or CRH. And that is the hormone that's why we are having this sudden urge to run to the toilet because of this release of CRH. CRH also acts on our microglia and creating inflammation in our brain, which is then completely changing how our vagus nerve action works. And also CRH is acting on our immune cells in the gut, on the mast cells. There are so many receptors of CRH on our mast cells. So when CRH is being released, it binds to those mast cells. Mast cells being activated are then releasing inflammatory mediators that act on our tight junction barrier. They are degrading those proteins like zonulin that is holding our tight junctions together. So that opens up our gut barrier. That's one of the mechanisms of leaky gut syndrome or increased intestinal permeability. And what happens then is that all those toxins, lipopolysaccharides, and everything can just enter our circulation. That triggers immune response and inflammation. Interestingly, then that inflammation reinforces against CRH, and that creates this vicious stress loop. The feedback is created. When this stress loop continues for a prolonged period of time, we are facing with the visceral hypersensitivity, which is one of the hallmarks of IBS, that chronic pain, and also other dysfunctional gut disorders. Furthermore, if neuroimmune activation is just keep going in that vicious stress loop, we are facing with completely rewired gut-brain communication. But CRH is just the beginning of this story. When we are under chronic stress, then cortisol takes over the scene. So cortisol acts on our gut function and gut health in different ways, in numerous ways. One of the things that happens with chronic stress is that we are having our immune system completely dysregulated. Our immune troops are redirected. We have redistribution of natural killer cells, we have lowered activation of T and B cells, and we have increased pro-inflammatory cytokines. As we said, CRH opened initially that intestinal barrier. Cortisol is continuously degrading furthermore our barrier. It also contributes to low mucosal immunity, and also it acts on our gut microbiome itself. So cortisol doesn't just act on how our gut functions, it also acts on our gut microbiome itself by lowering beneficial bacteria. And in that circumstances, opportunistic or harmful bacteria have a chance to overtake the scene. They are thriving in this circumstance. Okay, again, me and technology, sorry. Here we can just briefly compare the actions between CRH and cortisol. So just to briefly say that CRH tends to act quickly, it increases motility, we have the sudden urge to go to the toilet, it opens up our intestinal barrier. With continuous neuroimmune crosstalk and communication, we are just sparking and inflaming our gut. And on the other side, cortisol is acting more slowly because it slows everything down. We have lower transit time, we have lower bowel movement, we can become chronically constipated when dealing with chronic stress. So in a sense, it's not a contradiction to say that stress can both speed things up and slow things down. It just depends whether we are facing acute stress or chronic stress, or who are the key players here. Is it CRH response or is it cortisol response? Now let's have a look what's happening in our gut microbiome itself. We already said that chronic stress is contributing to gut dysbiosis because it lowers down the beneficial bacteria and then harmful bacteria can take over. But what is truly interesting that even acute stress can shift our microbiome within hours. We also have to emphasize here that with reduced number of beneficial bacteria, we have simply reduced number of short-chain fatty acids, and these are the metabolites of fermentation process. When our beneficial bacteria is processing that fiber and that beautiful complex carbohydrates, then in that fermentation process, short-chain fatty acids are created, like butyrate. And our epithelial cells are thriving on that butyrate. When there is not enough of butyrate, because there is not enough of beneficial bacteria, our epithelial cells cannot thrive. They are feeding on butyrate. When they're not feeding on butyrate, they're not also consuming oxygen at the same time. With a reduced consumption on butyrate, that oxygen can kind of leak into our lumen. And that is also creating more of aerobic environment, which is not good for beneficial bacteria. Beneficial bacteria needs anaerobic environment. So that's also another thing that's just perpetuating this gut dysbiosis. And we can definitely say that stress is not just a trigger for gut dysfunctional disorders. We can really firmly say that it is a driver of these disorders. I've seen it time and again in my practice that simply diet and supplements are not enough to deal with functional gut disorders. Stress is really a key player here that has to be addressed. I'll give you an example of SIBO, because stress in SIBO, it is such a strong factor that really has to be managed, not just with low FODMAP diet that we can manage SIBO, but stress is a key factor here because stress will go from back, top, and bottom. So we have this valve between our small and large intestine, which becomes completely dysfunctional when we are under chronic stress, so that we can have recolonization of our bacteria. It's going from large to small intestine. And from the top to the bottom, under stress, we are having lowered stomach acid, which is critical for our digestion, and it is just critical for our gut microbiome itself. If we don't have enough of stomach acid, all those microbes can just simply go from bottom to down and colonize in our intestines as well. As I said, the key thing here is we really have to work on that stress and enter into this rest and digest mode and activate our parasympathetics. So here we can just compare what is happening when we are in that rest and digest mode or when we are activating parasympathetics versus what's happening when we are under constant stress in that fight or flight mode. We are constantly cycling throughout the day and throughout our lifetime between this stress or fight or flight response and rest and digest or when we are calm. So blood flow is completely redirected from our gut towards our brains and muscles to fight that stress. It is time to survive. It's not time to digest. It's not time to heal. It's not time to absorb nutrients. Everything is impaired when it comes to our gut functioning when we are under stress. And there is not time when we heal. When we heal is when we enter into parasympathetics. When we enter parasympathetics, we have perfect environment for digestion, for absorption, for secretion of digestive enzymes, for microbiome balance, and that's the time when we can heal and repair. And a key player or getaway into this parasympathetics is vagus nerve. And we also mentioned vagus nerve as a key factor in gut-brain axis. So vagus nerve is exerting those beautiful anti-inflammatory actions on our body, and it's also in charge to calm ourselves down. Numerous people who are having anxiety-like disorders, depressive-like disorders, and mood disorders are not having enough functional vagus nerve. Vagal tone is completely compromised. So what can we do to actually improve our vagal tone, to enter this parasympathetics? There are numerous effective strategies, such as deep rhythmic breathing, meditation, yoga, mindfulness, intense gargling of water, humming, loud singing, splashing cold water into our face and cold showers. All these techniques that are so simple and effective are improving the functions of our vagus nerve, and they're also calming our nervous system down. Besides vagus nerve activation, there are numerous strategies and tools that are offering this calmness, and that they're supporting our gut-brain axis and mental well-being. And there are numerous integrative frameworks here. Of course, diet and lifestyle are playing such a huge role in this. Diet has a tremendous effect on our mental health. It has been in studies for so long now, and so much so that nutritional psychiatry was formed a few decades ago as a new scientific branch that was actually studying this beautiful connection between our diet, dietary pattern, and specific nutrients on impact on our mental health. It was really amazing to see in those studies how, for example, simple probiotic supplementation was improving depressive and anxiety-like symptoms in psychiatry section. Nowadays, diet that is focused around those nutrients that are especially beneficial for our gut microbiome is called psychobiotic diets. That is diet rich in fermented foods, probiotics, and prebiotics, all those beautiful plant foods that can enhance our beneficial bacteria, that can balance our microbiome, and then have via gut-brain axis these beautiful health, beneficial effects on our mental well-being. In research, it's been from a long time ago that certain probiotics can improve our mood, cognition, memory, etc. And then scientists asked, can we do something about stress resilience and stress response? And actually, studies have confirmed that certain probiotic strains can actually modulate our HPA axis, they can modulate stress response, and we can be more resilient to stress. So let's look at some of those strains. One of the probiotics that was first in a research was Bifidobacterium longum 1714. Early clinical studies done on mice showed that the mice had reduced anxiety and reduced stress response, and similarly, human trials confirmed the same. After only 4 weeks of intervention, healthy human adults have shown lower cortisol output in response to stress, lower subjective perception of anxiety, and there was a lower resting neural activity. Furthermore, Lactobacillus rhamnosus JB1 reduced anxiety via GABA receptor modulation. So all those probiotics are acting on our neurotransmitters and also on HPA axis. Then there is Lactic-Acid-Bacillus rhamnosus HN001 that has shown efficacy in reducing postpartum anxiety in women when given in second trimester until 6 months after postpartum. A blend of Lactobacillus helveticus R0052 and Bifidobacterium longum R0175 managed to restore that tight junction that is initially impaired by stress, and it also reduced HPA axis activity. Interestingly enough, this duo in one research was only effective when combined with lifestyle strategies. So food and probiotics aren't everything. We have to also incorporate lifestyle strategies to really support both sides of the axis. So what are all those lifestyle strategies? One of the strategies are those that are activating our vagus nerve, that are calming our nervous system. Others are adequate sleep, circadian rhythm alignment, mindful eating, mindful practices, biofeedback, cognitive behavioral therapy. All those techniques together can reduce stress and cortisol output and calm our nervous system. Because proper functioning of this gut-brain axis ensures gastrointestinal homeostasis. So we really have to always think of addressing both sides of this axis, mental or brain part and gut part. Before we step into what could future studies focus on, let's just go through key takeaways here. So both stress and gut are deeply interconnected through this gut-brain axis. Both acute and chronic stress are inducing gut dysbiosis. They are both weakening our intestinal gut lining and barrier. And just simply put, chronic stress means that we will chronically face gut issues. And we really have to step into these parasympathetics to properly rest and digest and to have our gut functioning in optimal way. As for the future research, we definitely need more causal mechanisms linking stress, dysbiosis, other diseases as well. And even though the research is growing about those probiotics and specific strains, so-called psychobiotics, we need more research on them, identifying more of these psychobiotic strains that can actually reliably and efficiently modulate our HPA axis and stress response. And with multi-omics integration, we can see what is happening with neuroimaging in the brain, and also in the gut with all these functional tests that already exist. I would like to finish this talk with one of my favorite quotes by Dr. Joe Dispenza that I heard on one of his advanced workshops many years ago that really had such a huge influence on myself and in my work with clients. And that is, you can eat all the organic foods in the world, but if you're living in constant stress, you're still eating organic stress. And that beautifully summarizes what we were discussing now. We tend to think that diet and supplements are simply enough to heal the gut, but that's not true. Stress is a key player here that has to be addressed. We have to work on that stress. We have to enter this parasympathetic or rest and digest. And it's not just physiology, it's a therapy. Because calming our nervous system is foundational part of our gut health, mental well-being, and overall well-being. Thank you. And I just have one thing. We have 30 seconds. I would like to invite you all to participate in one small exercise to enter our parasympathetics. I want you all to close your eyes, to have this deep breath. Once you're in that deep breath, go and do a second one to completely inhale everything. Do it through your nose. And then exhale slowly, slowly, slowly through your mouth. Do it 2 or 3 more times. And I hope that you will now digest better your lunch. Thank you.
Thank you so much for the I didn't join us for. No, it's okay.
Okay.
Thank you. Our next speaker is Ms. Luciana Goodwell from United States. Her presentation title is The Gut-Brain Connection, Cultivating Emotional Well-being Through Microbiome Health. Please welcome Ms. Goodwell.
So, what are we using? By the way, Katerina, where is she? Girl, thank you for giving some foundation to my presentation. That was amazing. I love it. Okay, everyone. My name is Luciana, and I'm so excited to be here with everyone today. And today we're exploring something bold, very ancient, and cutting edge. And we're going to talk about how the brain shapes our emotional resilience and how it communicates with the gut, which it's not something new, but now we're getting more of understanding how in depth and how it is connected to so much more. So the title of my presentation is The Gut-Brain Connection, Cultivating Emotional Being Through Microbiome Health. And today's objectives bring both scientific and the human experience. So we're going to talk a little about the brain connection. I'll make it very brief, and because I want to bring more of the message, bring the awareness of how important it is for us to live a comprehensive living. Even going back to Katarina was saying is, there's so much more than just taking probiotics and taking supplements and nutrition for our gut health. And then we're going to explore how the microbiome is connected to everything. Literally connected, is foundational to our health, and how it shapes our mood, our energy, inflammation, our emotional resilience. We're also going to talk about mitochondrial health. It's something that is research is talking about it more now, how without energy, nothing is gonna, right? We're not gonna be going further. We're not gonna be able to wake up and live our lives. And that's how even the gut is, how it is connected to microbiome and how they shape each other. And we're also gonna talk about a little bit of a couple of probiotics that is connected to emotional resilience. But science alone, my friends, does not tell the story, okay? Our daily lives, our rhythms play such a profound role in what we do from the morning we wake up to the minute we go to bed. When you think about the way our ancestors lived, okay, they owned it. They talked about that illness happens when we're not connected to our mind, body, spirits. And when there's a disconnection from that is when our illness begins. And why I'm bringing this is because our microbiome, it is some of all our experiences. And the way they live, they talked about connection. They talked about purpose. They talked about community. They talked about, how do we connect each other? So because the microbiome is the sum of all our experiences, it's something that it's being talked about more because we are so disconnected from each other, disconnected from ourselves. So, before I get into the science, I just want to talk a little bit about how I got here today. So, at the age of 14, I moved to America. And shortly after that, I started developing a lot of health issues. And I started working with a integrated functional medicine doctor for about 9 years, which understanding how everything is connected, how our bodies is connected to everything, getting to the root cause of the problem, optimizing functions in the body. And then later on, I became a registered dietitian, worked in a hospital for 9 years. During COVID-19, I worked in kidney, heart transplant, and all kinds of GI dysfunctions. And having that integrative approach and then working in a hospital, one thing that brought to my attention is how healing is super deeply personal, right? So science, labs matters, but everything else matters as well, right? Purpose, connection within ourselves. So factors like how are we processing our emotions? There's so many little details, and I see this with private practice as I teach my clients, as Katarina was saying, wasn't just about the nutrition. It's not about the supplements. It's really helping people understand that there's more into healing than, taking supplements, or it is a comprehensive approach. This brings me to a problem, or a growing problem in our generation, right? The rising strain of emotional health, anxiety, and depression, right? How many are suffering? In America right now, it's about 40 million. Global, it was about over 300 million of people are suffering from anxiety and depression, which, again, it's bringing, right, the disconnection. Why is it that is causing and triggering the anxiety and depression, which there is a gut connection, and how stress is affecting our daily relationships, right? How we connect to people. Working during COVID-19, I saw how many people have been disconnected from ourselves. And obviously after COVID, things have gotten worse. So it's not just recognized as a problem it's something that's recognized now worldwide. Which brings us to a powerful truth that our emotions, our biology are super connected, right? Everything's connected to our hormones, our talking about stress. Everything we do on a daily basis is going to affect our hormones, it's going to affect how the gut communicates with the brain, it's going to affect our diversity of the microbiome, because the richness, the diversity, the more diverse, the more of the beneficial, the essential guys we have, the better resilience, the better emotional health. A little about gut-brain axis, right? So it's a continuous conversation shaped by chemistry, emotion, lived experiences. So our gut and our brain are communicating 24 hours a day, 7 days a week. 24 hours a day, 7 days a week. And again, it's based on what we're doing from the time that we wake up, from the time that we go to bed. And obviously, in that connection, we have the microbiome. And for those who may not know this, is that as much as human cells we have in our bodies, we have bacterial cells. So it's actually a 1-to-1 ratio. Back in the days, it used to be for every 10 bacterial DNAs, we had 1 human DNA. Now that ratio is 1-to-1. So that's how much trillions of species we have within us that is connected to the gut-brain axis, right? So if there's a disruption of the natural flora, like Katarina was saying, is the pathogens, the guys that we don't want growing are going to take over. The vagus nerve, we call it a superhighway. So the vagus nerve is the nerve, the longest nerve that connects the gut-brain axis. And we call it as the biggest emotional messenger and also connected to our intuition, right? So we're disconnected from ourselves. Are we really listening to, right, what our inner guiding is guiding us, right? How many times you look back and you're like, oh my God, I made this decision and you knew better, right? But why did we make that decision, right? That's the disconnection. Because if we're really connected to ourselves, we're listening to where we're being guided, because it is connected to our intuition, right? Because it's connected to our emotions. All our neurotransmitters, right? Serotonin, we already know that serotonin, dopamine, all produced in the gut. And is metabolized through the gut microbiome, right? So if you don't have those species that are responsible for the conversion, then how is it going to be communicating back and forth to the brain, right? So if there's a decrease, which Katarina talked about stress the main thing that disrupts that natural flora, then it makes sense why depression and anxiety is on the rise. And a little bit about the microbiome, just for educational purpose, right? It's foundations of overall health. Now science is showing that it's literally connected to everything in the body, right? So if that foundation is not doing well, then it's going to affect other systems, right? Because everything works in synergy. Everything is connected, right? We're connected to each other. We're connected to nature. And that's how the human body works. If the gut is not doing well, then the brain's not going to do well. And then it's going to start taking a toll on other organs in the body. It also makes it break down essential vitamins and amino acids, right? So we get essential amino acids from our food. And then the microbiome also has the capability of making those amino acids. So again, if there's a decrease in that natural flora, we're not really digesting our foods. I always tell my clients is, you can have the best diet in the world, but if you don't not taking care of the stress, if you're not creating health, because that's what functional medicine is the science of creating health. So how can I start creating more health for myself? Because then everything starts healing and coming together beautifully. The microbiome is also part of what's called the phase 3 detoxification. Okay, so phase 1, phase 2 happens in the liver, right? So if you come in contact with something, or you start breaking down things, the liver, phase 1, phase 2 happens in the liver, phase 3 happens in the gut, right? So when you go the bowel movement, it's going to start bringing all those toxins out, because the microbiome is so beautiful that it can bind to heavy metals. It can really, if it's how optimal it should be thriving the way it should, it's going to do the job and start cleaning everything out. It also influences the production and supports our hormones, because remember, it's connected to everything, including estrogen levels, right? So if our bodies, right, estrogen, too much estrogen, it's an inflammatory hormone. We don't want an estrogen too high, right? Because everything is a perfect balance. And what's beautiful is the microbiome has the ability to metabolize that, take it out. If the good guys are not there doing the work, then all the estrogen, a lot of these, not only chemicals, but estrogen, like it's dumped back into the system. The body does not have the ability to metabolize it. And it is the sum of all our experiences. A big, in research, they've been talking a lot about the mitochondrial connection, right? The gut-mitochondrial connection, energy, right? The mitochondria is our energy makers, right? It's how the microbes
the microbes, they produce metabolites that support the mitochondria. And again, if the microbes are not doing well, then how are we going to really produce energy? How is really the gut and the brain going to communicating if our body's not producing energy correctly, right? So when there's dysbiosis, a disruption of the natural flora, now you have an inflammatory response, right? Your immune system is reacting, shooting that inflammation to the body. I usually tell my clients, like, think about a fire that never goes down. That fire is constantly just shooting the inflammation back and forth to the brain, to the rest of the body. And how the gut and the mitochondria communicates is through butyrate, right? So Dr. Salas was talking about butyrate. I know Katarina talked about butyrate. It is the preferred short-chain fatty acids, not only to make energy, but also to how it communicates. It's a signaling fact that the butyrate causes. And here's another powerful, to bring to everyone's awareness, that science now is showing that depression, anxiety, bipolar, Alzheimer's, schizophrenia, a lot of, pretty much most of the mental health dysfunctions begins in the gut before it shows up in the brain. So when there's constant inflammation affecting, right? So here are the studies. And when there's inflammation, right, there's a blockage. There's no energy. So this impairs the communication pathways, right? So it was powerful because the Alzheimer ones just came out recently with that study. That after all, it doesn't start in the brain, it starts in the gut. And here's the turning point of the story that I wanted to bring to everybody. Because the microbiome reflects our whole experience right from the day we're born, right? Were you breastfed? What type of birth you had? Were you the kid that played around with dirt? If it is the sum of all our experiences, then we require an approach, right, that it brings back to our natural design. So humans can live and thrive, because so many people are not thriving, right? They're in survival mode. So it's beyond biology, it's beyond psychology, so it's towards wholeness. I truly believe the future of healthcare with the gut and the brain where we're heading it's about education, right? How can we educate our clients, even me coming from the hospital, right? I always want to take time to educate everybody, but in the hospital it's so limited. And if we really just think about how we can, be the example for each other, we can have others thrive. This research was powerful, this study. It was the data that I collected that we're currently living in a global recession, right? So we're living in a generation where emotional intelligence is so important in every area of our lives, right? Because when we actually process our emotions, and I'm going to talk about a study that's coming up now, when we actually process our emotions, it actually, because it is connected, our gut health is connected, our microbiome is connected to our emotions. When we actually take the time to understand our experiences, right, process everything, going through that healing, it actually improves our gut microbiome. So then, reconnection with the design and the rhythm is also part of that solution, in conjunction, right, with targeted probiotics, with the nutrition, is bringing health lifestyle is important. And in private practice, the last 2 years, I don't work in a hospital anymore. After I left the hospital, I've been very more of, I want to educate my clients what it is that they need to create health in all areas of their lives, right? So I take the time to talk about stillness, process your emotions, reflection, right? These are things that we weren't taught as kids. And as I've been through my healing journey in the last 5 years, and I see how profound it is to be in stillness, to understand why things are happening to us, and really how it affects our health. Right? So wholeness is not achieved, my friends, it's remembered. It's remembering who we are. And this is the powerful study I was just talking about, the gut feelings, how emotions shape the gut microbiome. So this study that they did, they collected DNase-2 tests, and it showed when women process their emotions, the diversity of the microbiome improved, but those that suppress their emotions, it lowered their production. So it was really interesting how emotional regulation supports gut balance, right? So that's why, again, it's a comprehensive approach to healing, right? Because when you think about synergy, how everything's connected in the body, how we are all connected to each other, but
we see such a big disconnection, then we start, having that aha moment thinking, okay, how can we really understand that to really truly help someone is bringing how our ancestors lived, right? How humans were designed to thrive. And I know that Katarina talked about a little bit about psychobiotics. There's definitely different, probiotics that I use in private practice, but I've been researching more about the psychobiotics, which it is, it helps with not only systemic inflammation, but it helps with strengthen the gut barrier, right? Because healing the microbiome is also healing the lining that helps the barrier in the gut communicate. Because when that lining is disrupted, that's when things are going back and forth between the brain and the body, and with the inflammation and stuff. So by helping that integrity of the lining, by helping improve the microbiome, taking consideration, right, a comprehensive approach, then everything, comes together beautiful. And also the psychobiotics helps vagus nerve signaling. One of them is the bifidum longum, 1714. Human trials right now supports reducing cortisol levels, right? Because what happens when you have high cortisol, that means the body's producing more inflammation, right? It enhances calm focus and cognitive performance. It supports emotional resilience via the gut-brain pathways. And these are a couple of the studies that I found, but I pretty much kept for today's presentation, I kept the two main one. The next one was about Helveticus R0052 with longum combined, which it is with lifestyle included, how it helps reduce anxiety, psychological distress, it improves cortisol balance, but also taking consideration, right, the comprehensive approach. So the duo together, this study was a randomized that they did that it really showed the improvement in the participants' lives. A couple of key nutrients for the gut brain and the mitochondrial connection. These are used in private practice because again, everything works in synergy, right? So if you're giving probiotics, because it's personalized, right? I do very personalized nutrition, and not only I work on optimizing my client's microbiome, but I understand, okay, how can I come in and improve, right, inflammatory responses? How can I come in and teach them that, how meditation is important, right? To not be just, going a million miles an hour, but like, hey, take time to go sit by the tree for 10 minutes and stuff. So in conjunction to that, CoQ10, as we know, is, important to make energy. It improves ATP production. Omega-3 fatty acids, huge, right? It helps with inflammation, lower inflammation in the gut. Magnesium is one of my favorites, right? There's different kinds, and just, today I'm just like going beef because I'm tiny. And then vitamin D3, K2, super important, right? Especially K2 with D3 together. During COVID season, I was blown away how many people were taking vitamin D3 high levels and did not have K2 in conjunction with it. And K2 is so important because it literally, K2 acts like a little carrier in the car, like a little car that brings vitamin D where it's needed in the body, because every organ in the body has a receptor for vitamin D. So K2 is super important. Okay, then the foods for the gut-brain connection, right? So what foods are going to break down into butyrate? That's going to help support the microbiome and the communication, right? So just some ideas, right? You have garlic, banana, asparagus, prebiotic foods. We all know that. We talked about fermented foods. I know some countries are different. Back at home, it's, fermented foods, buying like kefir, kimchi, sauerkraut, like it's extremely expensive. So, but it's so important because again, it's not just about taking probiotics. It's about what kind of foods are also going to help synergy work in conjunction. Okay, so returning to our design, right? How important it is for us to understand the connection, living with purpose, right? The reason why I brought this into my presentation is the last 2 years, I've met so many people that lack purpose. And purpose is shown in studies to reduce inflammation, right? So are we waking up and going to our jobs 9 to 5? Like, do we love what we do? And seeing that was profound, seeing that in the last 2 years, how many people they're like looking for something to fulfill. They're going right to their 9 to 5 job, but they're lacking purpose.
So again, when you think about a comprehensive approach, living in our natural design and how humans were made to thrive, connection increases vagal tone and microbial diversity, right? So communities, there's studies on community now, how community is important for us for overall health and emotional well-being. Nature, right? It has a serotonin, microbial, right? So grounding, stillness. So another thing that it's important is, right, we can talk about it all day long, but are we actually putting into practice, right? It's the embodiment, right? It's integrating, right? So when we teach our clients, when we teach about how to enhance your emotional well-being, how to improve the microbiome, right? It's teaching them. It's something that I've been doing, and I've seen the transformation with a lot of my clients, and how it not only improves their lives, but improves the lives of their surroundings. Because everything works in synergy, right? If I'm thriving, I'm doing well, I'm gonna be like, hey, I wanna feel like her. How are you feeling? And I know we're from different countries here, but I'm sure that we can all come together and understand that this is a problem that's happening worldwide with a lot of people. Right, so again, today for my presentation was bring more the awareness, right? Purpose and meaning of my the information that I wanted to bring. And I truly believe that the future, the integrated approach is how do we bring healthcare and bring more of that connection and purpose, right? Me and the girls were having a little talk during lunch, and we're talking about that, right? If we're teaching, if we're in the embodiment, and we're teaching others, right? It doesn't matter if you're a psychiatrist, a psychologist, like whatever area you are, right? It's how can we help someone, right? Based on the microbiome. How can we truly help someone thrive, right? Not just by bringing the science, by bringing these, something that sometimes just stillness, we think it might be something little that doesn't help, but it really does help in so many ways, and the research is showing, right? So for the future is how can we bring more of the integrated approach into healthcare? And I see a little bit more now because I'm connected to a lot of people back at home, and I have a couple of people in my space that are in functional psychologists, and they're bringing back, right, the gut-brain connection. They're talking about probiotics. They're talking about movement. They're talking about, sleeping better and stuff like that, because it is important for our emotional health and for us to improve our overall health and the health of our surroundings, right? Because if I'm feeling better, that means I'm going to connect better to my friend. I'm going to have a connection with my husband. It's right, because everything works in synergy, right? Spiritual, emotional processing, huge. I teach nutrition for these kids in my community for my church, and I'm actually teaching them how to process their emotions, like when things are coming up for them, and teaching them that it's okay to be upset, but understand that our experiences is not here to affect us, but it's here to teach us, it's part of our evolution. And because I'm teaching gut health nutrition, I'm teaching them that by doing this, it is going to improve their health. They may not see now because they're young, but they're going to see if there's something that small that I didn't have growing up that I wish I did. Now, most of us, I'm sure, can agree. It really is profound, because we start really changing the direction of where we're heading. And obviously collaboration, right? Collaborating with other healthcare providers, and then personalized nutrition, which is happening a lot. You see more, right, the genomics and functional medicine. And it really is, from when I started with functional medicine 20 years ago, where I am now, it's beautiful to see the transition, like to see that happen. So some key takeaways is to remember our design, right? That gut health, emotional health, or gut-brain connection is not just about, what we're taking. It's about what we do from the minute we wake up to the minute we go to bed, right? To live in alignment, like our ancestors talked about. When we live in alignment to who we are, to live in the truth of who we are, it really drives your body and everything as a whole to come together, so you can truly thrive in your life. You go from surviving to thriving, right? And then your microbiome does reflect your lived experiences. So having that awareness that our microbiome is some of our experiences
that help us understand that what we do on a daily basis is extremely important. And thank you guys for just for inviting to be here. I hope that, I brought some awareness to what it is, how important it is for us living with purpose and clarity and meaning.
Thank you so much, Luciana. Please come forward for Thank you. No, okay. Yeah, so our next speaker is Dr. Federica Ricoveno from Italy. Her presentation title is The Influence of Probiotic as Immunomodulatory Therapy and Autoimmune Disease. Please welcome Dr. Ricoveno.
Good afternoon, everyone. It's a pleasure to meet you. I'm Dr. Federica Ricoveno, recently graduated, and today I'll present the results of my clinical study investigating the immunomodulatory effects of probiotics in autoimmune thyroiditis. Autoimmune thyroiditis represent a group of chronic inflammatory disorders characterized by aberrant activation of the immune system, resulting in the loss of self-tolerance. Consequently, self-antigens expressed on thyroid tissue are misrecognized as non-self and become targets of the immune response. The most common forms are Hashimoto's thyroiditis and Graves' disease. Hashimoto's has a global prevalence of 10-12%, and it is characterized by an infiltration of T lymphocytes alongside B lymphocytes producing antibodies against the thyroid peroxidase and thyroglobulin. This inflammatory process leads to the destruction of the thyroid gland, and this condition manifests with the symptoms of hypothyroidism, including weight gain, cold intolerance, fatigue, depression, and bradycardia. Locally, painless sperm goiter is often present. In contrast, Graves' disease has a lower prevalence, and it is characterized by the presence of antibodies against the thyrotropin receptors stimulating, and this condition leads to hyperfunction of the gland, manifesting with the symptoms of hyperthyroidism, including weight loss, heat intolerance, nervousness, agitation, exophthalmos, and tachycardia. Besides, both disorders exhibit a marked female predominance. The etiology is multifactorial and involves a complex interaction between genetic factors and environmental factors. On one hand, there is a genetic predisposition. Indeed, the polymorphisms in genes such as FOXP3 or CD25 disrupt the function of regulatory T lymphocytes. Alterations in CD40 promote B cell activation and the production of autoantibodies, while mutations in CTLA4, which normally inhibit T cell activation, may lead to the loss of immune tolerance. On the other hand, environmental factors play an important role. In this context, the intestinal microbiota is essential for immune responses. Furthermore, selenium deficiencies or excess iodine, smoking, or certain infections can act as triggers. Under normal conditions, named eubiosis, the Firmicutes and Bacteroides phyla predominate, especially Faecalibacterium, producing short-chain fatty acids like butyrate These derived from the fermentation of complex polysaccharides. These metabolites maintain intestinal barrier function, regulate inflammation and intestinal permeability, and modulate the thyroid hormone metabolism. This connection, known as the gut-thyroid axis, highlights how the gut microbiota can influence thyroid activity through two main mechanisms. First, the gut microbiota is essential for the absorption of key micronutrients like iodine, selenium, iron, and zinc, indispensable for the synthesis and conversion of the thyroid hormones. And second, short-chain fatty acids can influence the thyroid hormone metabolism by regulating the liver deiodinase activity, intrahepatic circulation, and tissue sensitivity to the hormone T3. In particular, butyrate inhibits histone deacetylases. This shifts the balance toward histone acetyltransferases, which add acetyl groups. As a result, the chromatin becomes more open and transcriptionally active. This process facilitates the expression of beneficial genes, like genes that strengthen the intestinal barrier function and tight junctions, and genes with antioxidant and anti-apoptotic functions. The Firmicutes bacteroides ratio is recognized as a general marker of gut health. So when this balance is perturbed, like Graves' disease, where FB ratio is lower than healthy controls, and in which bacteroides predominate, or in Hashimoto's thyroiditis, where FB ratio is elevated, and in which pro-inflammatory bacteria like Prevotella are increased, while anti-inflammatory bacteria like Faecalibacterium are decreased. This gut dysbiosis can lead to the leaky gut phenomenon. This increased permeability facilitates the translocation of bacterial endotoxins and the release of pro-inflammatory cytokines, which negatively affects thyroid tissue. Given this potential correlation between gut dysbiosis and autoimmune thyroiditis, probiotics may represent a promising adjunctive therapies. Such an approach could complement conventional therapies by targeting the underlying imbalance of the gut microbiota and its immunomodulatory effects. Certain probiotic strains may modulate this balance, this report, potentially attenuating autoimmune responses. Furthermore, probiotics may enhance the availability of micronutrients like selenium, zinc, iodine, and iron, and concurrently the supplementation of antioxidants like vitamin C and selenium may mitigate the oxidative stress, key pathogenic factor in autoimmune thyroiditis. Thus, the rationale of this clinical study is the growing scientific and clinical interest in identifying adjunctive strategies in the treatment of autoimmune thyroiditis, conditions characterized by increasing prevalence and significant impact on public health. In this context, probiotics arouse a particular interest as, unlike conventional therapies, they act directly on the pathogenesis of autoimmunity. This study aims to evaluate changes in levels of specific autoantibodies and thyroid function by analyzing thyroid hormone values. And it aims to assess the potential of a standardized probiotic treatment in combination with vitamin C and selenium, and to explore the link between intestinal dysbiosis and the pathogenesis of autoimmune thyroiditis through the response to probiotic treatment. This study, this perspective and cross-sectional study was conducted in the endocrinology of patient clinic of St. Apostol Andrew Hospital in Galați, in Romania, over 6 months. Inclusion criteria were patients with the age of 18 years, Hashimoto's thyroiditis in spontaneous hypothyroidism or euthyroidism, Graves' disease under treatment with conventional antithyroid drugs. Exclusion criteria were age under 18 years, dementia, chronic renal failure, recent immunosuppressive treatment, and probiotic treatment for other conditions. This study enrolled 48 patients who received the treatment with omnibiotic stress repair, a mixture containing 9 human-derived bacterial strains of Lactobacillus and Bifidobacterium. The study sample was divided into 4 subgroups according to the diagnosis, so 30 patients with Hashimoto's thyroiditis and 18 with Graves' disease, and according to that combination of probiotics with either vitamin C or selenium. Autoantibody and hormonal parameters were measured at the beginning of the study and after 6 months of treatment, specifically antithyroperoxidase antibodies, which are considered the best serological marker for the diagnosis of Hashimoto's thyroiditis being present in the vast majority of patients, and antithyroglobulin antibodies, although they are less relevant for the diagnosis of the disease, were measured. In addition, TRAB, or thyrotropin receptors stimulating antibodies, which play a crucial role in the pathogenesis of Graves' disease, were also evaluated. Regarding thyroid hormones, TSH, or thyroid stimulating hormone, which is the first line for assessing thyroid function, and FT4, or free thyroxine, which is the unbound and active fraction of the thyroxine, were evaluated. The statistical analysis was performed in SPSS software with a 95% significant level using the ANOVA test. But demographic parameters were also evaluated. Among the results, it was observed that the gender distribution was predominantly female, with males present only in the Hashimoto's groups. The mean age was approximately 49 years, plus or minus 13, and most patients came from urban areas. After 6 months of treatment, in both the groups of patients with Hashimoto's thyroiditis, so those treated with probiotics and vitamin C, and those treated with probiotics and selenium, a decrease in mean TSH value and an increase in mean FT4 value were observed. Regarding antithyroperoxidase antibodies, after 6 months of treatment, a slight decrease was observed, while the decrease in antithyroglobulin antibodies reached the statistical significance in the group treated with probiotics and vitamin C. Regarding Graves' disease, after 6 months of treatment, at the beginning of the study, all patients presented a TSH value below the reference range, but after 6 months of treatment, approximately 90% of participants reached normal values. As for FT4, values remained within the reference range. Regarding TRAB levels, after 6 months of treatment, the decrease in TRAB levels reached the statistical significance in the group treated with probiotics and vitamin C. The study demonstrated that autoimmune thyroiditis are more frequent in women around the age of 50 years, and especially from urban areas. In Graves' disease, the treatment with the probiotics and vitamin C for 6 months clearly reduced the autoimmune activities, and it is demonstrated by the decrease in TRAb and FT4 levels and the increase in TSH level. These results are in line with previous studies using Bifidobacterium longum with a conventional methimazole therapy. While in Hashimoto's thyroiditis, it was observed a significant reduction in anti-thyroglobulin antibodies under treatment with the probiotics and vitamin C, while the other parameters remained unchanged. This outcome is consistent with the limited literature available, where mixtures of lactobacillus and bifidobacterium or lactiplantabacillus plantarum used in the first available study showed no significant effects on autoantibodies and thyroid hormones. It is well established that gut microbiota varies between individuals, and it is influenced by the specific disease affecting each patient. So a major limitation of this study is that the composition of gut microbiota was not analyzed due to the time and the advanced methodology required. So for future research, it is recommended to analyze the gut microbiota of each patient using 16S rRNA gene sequencing in order to administer probiotics tailored to address specific bacterial deficiencies. Another limitation is that the study lasts only 6 months, which may be too short to observe benefits under treatment with the probiotics, especially in Hashimoto's thyroiditis, where the Th1-driven damaged tissue and autoantibodies change slowly. And in Hashimoto's thyroiditis, the modest effects observed may be linked to the Th1 dominant immune responses and the consequent direct thyroid tissue damage. So, starting probiotic treatment in the early stages of the disease could maximize immunomodulatory benefits. In conclusion, probiotics, in particular when combined with vitamin C, demonstrated potential immunomodulatory effects in autoimmune thyroiditis. These benefits were more evident in patients with Graves' disease, suggesting that the gut microbiota may influence disease activity and thyroid function. Our findings support the therapeutic potential of microbiota-targeted intervention and emphasize the need for further research into personalized adjunctive strategies for improving immunoregulation in autoimmune thyroiditis. Thank you for your attention.
People ready? I'm having time, so I can pronounce all this name. That was amazing. It's so awesome how the combination of probiotics and vitamin C just shows how supplements work, the synergy, right? How everything is connected and how they position each other too.
Yes, it was used for this synergic activity. So probiotics with antioxidants, in particular with vitamin C, because it has a systemic activity, while selenium acts in particular on thyroid gland. So probably it is the reason for this difference of results between probiotics and vitamin C and probiotics with selenium. Thank you. Thank you so much.
You were mentioning just the presentation. And just a question, do you recommend any specific strains of probiotics that you think is not due to or patients low the
Sorry.
Any specific strains of probiotics?
We have used only biotic stress repair, which is a mixture of 9 probiotic strains of lactobacillus and bifidobacterium. But in order to have a major result, it was recommended to analyze the composition of gut flora in each patient in order to address probiotic to bacterial deficiencies.
Okay, so it was targeted selection of strains based on their gut flora.
Yes, it is recommended to administer selected probiotic in order to balance the deficiencies of bacteria in the composition of microbiota. Thank you. Yes.
Certification. The next speaker is Dr. Rhonda Amiji Hassas. I said it right? And integrated gut health expert. Yes. What is it my levels? And her title of presentation is Gut Inflammation, the Whole Body Health, how healing gut is everything.
Sorry, to move the slide, I can put here. Okay. Because I want to read. So this one.
This one forward, this one backward.
Okay, thank you. Thank you. Stop. Okay. Good afternoon, everyone. I am so grateful to be here today among colleagues who believe, as I do, that the gut is at the root of our entire health. When we talk, about gut inflammation, we are talking about something that quietly affects immunity, hormone, skin, mood, and even how we age. And like many of you, my interests start with my own health story. Because when your gut breaks down, you will feel it everywhere, from head to toe. I want you to think about your gut as the soil of a plant. If the soil is inflamed or dry, the leaves wilt, no matter how much you water them. The same goes for our body. So in my 20s, I thought I was the healthy one. I exercised, ate well, worked long hours as a dentist in my clinic, but behind the scene, I was really exhausted after every meal. My stomach was bloated, my brain foggy, and I felt anxious for no reason. You know that feeling when you eat something healthy, yet your body feels like it's fighting you. That was me for years. A colleague once told me, maybe it's gluten, maybe it's dairy. I laughed and said, it cannot be that simple. But after endless tests, the biopsy confirmed celiac disease. So, overnight, my world changed, reading every label, avoiding restaurant, trying to understand cross-contamination, and still I didn't feel completely better. That is when I realized it wasn't only about gluten. Something deeper was wrong. My gut was inflamed, my hormone out of balance, and my nervous system was exhausted. That's what led me to study integrative nutrition and gut health And I get certified as an integrative nutrition gut health coach, autoimmune disease, hormone balance, and weight management. I became obsessed with understanding how the gut affects everything: energy, mood, hormones, even how we think. So I shifted from being a dentist who fixes smile to a gut health coach who help people feel like smiling again. So what is gut inflammation really? But before, I want to tell you that healing my gut didn't just remove my symptom. It gave me back clarity, calm, and confidence in my body. That is why I believe gut inflammation is the silent root behind so many chronic complaints, and the best part, it is reversible. So when most people hear inflammation, they imagine red swollen joint or fever. But inside the gut, inflammation often look like microscopic irritation and an over-alert immune system defending a barrier that is one cell thick. So I want you now to imagine your gut lining as a fine silk curtain. It protects what's inside from leaking out. Now imagine poking small hole in that curtain every day with stress, processed food, antibiotic, or gluten. Eventually, light start leaking through. That is your immune system reacting to things that shouldn't be there. So now, chronic stress, it tightens blood flow to the gut. Processed food and sugar, they feed the bad bacteria. Antibiotic, they wipe out the good one. Gluten, especially for sensitive people or those with autoimmune conditions, will damage the villi in the small intestine. The scary part is this inflammation doesn't make noise. You don't feel pain in your gut wall, but you might feel fatigue, skin flare, or brain fog. The gut talks to us in indirect way. For example, you eat a sandwich, 30 minutes later, you feel tired and moody. It's not the carbs, it's your immune system reacting to what slips through that leaky line. So as we see here, stress, gluten, antibiotic, sugar weaken the gut wall. When the gut barrier is damaged, it's not tightened anymore. Bacterial fragments leak into the bloodstream, leading to inflammation. So poor diet plus stress, it's leaky gut. Leaky gut, it's inflammation throughout the body. So how gut inflammation spread beyond the gut? The gut isn't an isolated tube. It is a command center constantly messaging the body through three highways: immune, nervous, and metabolic. So immune highway, when the gut barrier leaks, immune cells release cytokine, which is a chemical messenger that travels in the blood and create inflammation far away. That is why joint pain, skin rashes, or thyroid antibody can begin with an inflamed intestine. A patient with eczema whose rash clears once we treat the gut. It's not about the skin cream, it's about going deeper and healing the gut. Your nervous pathway, this gut-brain axis, gut-brain connection. You know, the vagus nerve is like what's up between your gut and your brain. 90% of its message go upward. When the gut sends Denshi message, the brain will respond with anxiety, low mood, or poor sleep. You know, women told me when my digestion is calm, my anxiety disappears. That is the vagus nerve in action. Now the metabolic pathway. Bacterial toxin like LPS or lipopolysaccharide leak into the blood, creating low-grade inflammation called metabolic endotoxemia, presence of endotoxin in the blood from the outer membrane of gram-negative bacteria, a silent driver of fatigue, insulin resistance, and brain fog. It's, like having tiny spark or particle in the bloodstream that never turn off. Your cells stay in defense mode instead of healing mode. So gut inflammation doesn't stay in the gut. It's a whole body story. Every mood swing, skin flare, or autoimmune flare could be a message from the gut asking for care. So science is finally catching up with what integrative medicine has known for years. Everything starts in the gut. I will highlight some research. So Divans and Zhou, 2023, they found that when the gut barrier is damaged, tiny bacterial fragment will leak into the bloodstream and trigger inflammation in distant organs. Late 2024, this study focused on lipopolysaccharide, toxin from bacteria that circulate in the blood and turn on immune cells, leading to chronic fatigue and insulin resistance. Leo 2023, they showed that butyrate, a short-chain fatty acid from fiber, acts like a natural anti-inflammatory. It strengthens the intestinal wall. Last, zinc 2023 and Mitchell Fusa 2023, together, this study showed that probiotic and balanced microbiome literally calm the immune system and reduce inflammation throughout the body. So as we see, different study, one clear message: when we repair the gut barrier, the body knows how to heal itself. It's not magic, it's biology working the way it meant to be. The gut isn't just where food is digested. It is also where our emotion is digested. It is also where inflammation is decided. So now, gut inflammation, like we said, manifests everywhere. It's not just bloating and pain. Most people think a gut problem means bloating or reflux or gas. But in reality, the gut speaks through the entire body, sometimes without a single digestive symptom. We will see the impact on our immune system. When the gut barrier leaks, immune confusion begins. The body starts attacking itself. That is how autoimmune issues like Hashimoto, rheumatoid arthritis, or psoriasis often start with a silent inflamed gut. On your metabolic system, gut inflammation raises cortisol, insulin, and inflammatory cytokine, leading to weight gain, fatigue, and insulin resistance. Many women think it's hormone or menopause, but the gut may be the trigger. On your nervous system, inflammation in the gut sends distress signal through the vagus nerve, changing how we feel. That is why gut issues often show up as anxiety, brain fog, or low motivation, not just cramp. On your skin, the skin is a mirror of the gut. Acne, eczema, rosacea, they are external reflection of internal inflammation. I always tell my client, your skin is your gut crying for help just from outside. So the gut is like the conductor of an orchestra. When it's inflamed, every instrument, hormone, mood, skin, metabolism plays out of tune. But when it calms, the whole body finds harmony again. So now, how do we actually heal gut inflammation? You know, over time, I found that every successful case follow the same rhythm, a simple, powerful sequence I call the 4R approach: remove, replace, re-inoculate, and rebalance. So we start by removing the irritant, processed food, sugar, gluten, dairy, and chronic stress. Imagine you are trying to let a wound to heal. You have to stop scratching this wound, yeah? For my autoimmune client, just removing gluten and dairy often reduce bloating, brain fog, and fatigue within weeks. Now we have to replace. We have to replace what is missing: enzyme, bile support, mineral, and hydration. Many people cannot digest well because they are low in stomach acid or digestive enzyme. Then we bring back the good bacteria. We re-inoculate through prebiotic fiber, and fermented food like kefir or sauerkraut. It's like replanting the soil of your inner garden. Sorry. Now, finally, we have to rebalance. Finally, we rebalance the gut-brain axis by managing sleep, stress, breathing, and movement, because no supplement can heal a gut that is under constant stress. Even 5 minutes of deep breathing daily can shift your vagus nerve and help digestion. Healing the gut is not a quick fix. It is a re-education between the body and the mind. When we remove, replace, re-inoculate, and rebalance, the gut learns how to feel safe again, and healing begins naturally. So we remove what hard, we replace what is missing, and we re-inoculate the gut and rebalance the body. So let me share with you a story from my clinic, a real example of how healing the gut transformed the whole body. A 47-year-old woman came in complaining of chronic bloating, constipation, and joint pain. So, she had done every test. Nothing major, just mild inflammation. She was frustrated because she was told it is normal for your age. But she didn't feel normal. She felt exhausted, heavy, and disconnected from her body. We applied the 4R approach. She removed gluten and processed food, add digestive support, probiotic, and daily movement. Most important, we worked on her stress and sleep. She started journaling and doing light walk after meal. After 8 weeks, her digestion normalized, energy returned, and her joint pain was gone. Even her CRP, her inflammation marker dropped significantly. This is what I love about gut healing. When the gut calms down, the body follows. You are not just changing the gut, you are changing the body entire conversation with itself. So what can we take home from all of this? So whether you are a clinician, a researcher, or health coach, these are the principles that truly change outcome. First of all, always consider gut inflammation. When a patient presents with unexplained fatigue, anxiety, or joint pain, think gut first, not last, because chronic symptoms often begin in the gut. Spotting inflammation is the first step. Second, basic labs are powerful. CRP, fecal calprotectin may look simple, but they reveal hidden inflammation before it becomes disease. Healing is a journey. Gut repair takes time. You have to be patient and consistent. There is no supplement or single probiotic that fixes everything. It's a process, step by step and signal by signal. For the microbiome thrive on diversity. So eat the rainbow, rotate fiber, vary probiotic, your microbe love variety. 5, when the gut calms down, the body stops shouting. Energy, hormone, immunity improve. This is my favorite line because it's true in every patient. When the inflammation quiet, the whole body find peace. The gut is the beginning of every healing story and the end of most suffering one when we care for it in a right way. So healing the gut is about restoring harmony. When I look back at my own journey with celiac disease, I don't see it anymore as a curse. I see it as my greatest teacher. It taught me that inflammation isn't the enemy. It is the body way of saying, please listen, something inside needs peace. We often try to silence symptoms, but what if they are simply the body language of imbalance? The gut is like the soil of a tree. Need care, or the whole tree will suffer. Let's stop chasing quick fixes and start restoring connection between our gut and our body, between our body and our mind. When we heal the gut, we heal the person. And that is not philosophy. It is biology in harmony. So healing starts in the gut, and the gut listens to how we live. It is the most important truth I have learned through my own healing and through hundreds of clients. Our gut health reflects our lifestyle, how we eat, how we sleep, how we move, and how we manage stress. All influence gut balance and therefore overall well-being. Many illnesses originate from gut imbalance or inflammation. True healing began within by nurturing the gut through mindful living and healthy daily habits. So every meal, every breath, every moment of calm, they are message to our gut saying you are safe. And that is where healing began. And as Hippocrates said it all over 2000 of years, all disease began in the gut. And I will add health start in the gut. Today we finally have the science to prove it. Thank you for listening and for being part of this global movement toward true healing. Thank you so much. Thank you.
Hey, our next presenter is Dr. Keynote. Is the secret? Keynote. He's from Seoul National University Hospital, Republic of Korea. Part of the proposal is impact of sets specific cutoffs on reducing this advanced fibrosis in women with MASLD. What does this stand for?
Metabolic dysfunctions associated liver disease. I'll tell again. Yeah.
That's the
I'm pleased to present our study titled Impact of the Sex-Specific Cutoffs on Reducing Risk of Advanced Fibrosis in Women with Metabolic Dysfunction Associated Steatotic Liver Disease. Non-invasive fibrosis indices such as NFS, FIFOR, FRE-NAR, and BART are widely used to assess fibrosis risk in mesothelioma. However, all of these scores were originally developed in NAFLD codes, non-alcoholic fatty liver disease codes, and they do not account for biologic differences between men and women. Women generally have lower AST and ALT, different body composition, and metabolic characteristics. Because the traditional thresholds do not adjust for these differences, female patients are more likely to be misclassified, particularly in the rule-out ranges. This sex-based bias can lead to higher false negative rates in women, meaning more cases of advanced fibrosis may be missed. Therefore, we hypothesize that sex-specific dual cutoffs may improve diagnostic accuracy and reduce missed advanced fibrosis. Our study had three main objectives: to evaluate whether sex-specific dual cutoffs improve the diagnostic performance of fibrosis indices in mesothelioma, to compare false negative rates and AURS values between conventional cutoff and sex-specific cutoffs, to assess how the diagnostic accuracy differs between men and women. We conducted a retrospective analysis of biopsy-confirmed mesothelioma patients at Brahma Medical Center. For each patient, we calculated NFS, FIFOR, APRI, AR, and BART score. When you apply two approaches, conventional cutoffs and sex-specific dual cutoffs derived using the Uden index separately for males and females. Our primary outcomes were female false negative rate for advanced fibrosis and AURC values, reflecting diagnostic discrimination capability. These slides summarize our overall workflow. From screened patients, we concluded biopsy confirmed mesothelioma cases, stratified them by sex, calculated all fibrosis indices, and applied both conventional and sex-specific cutoffs to evaluate performances. We used the standard and widely accepted conventional cutoff like this, and for sex-specific thresholds, we calculate the dual cutoffs independently for men and women, based on optimal sensitivity and specificity using the Uden index. Figure 1 shows the change in false negative rates. The key finding is that the sex-specific cutoffs substantially reduced false negatives, especially in women. In females, the improvement was most pronounced for NFS and AAR. In male, the greatest reductions were observed in NFS and FIFOR. Figure 2 compares AURC values. Overall, AURC improved in both sexes when sex-specific dual cutoffs were used. The most notable gains were seen in NFS and FIFOR, particularly among female patients. This demonstrates that adjusting for sex does not simply shift thresholds, but truly enhances the discriminative ability of these indices. To summarize the major findings, sex-specific dual cutoff significantly reduced false negative rates, especially in women. NFS and AAR demonstrate the largest improvement in females. In male, NFS and FIFOR showed meaningful gains. AURS values increased across most indices when sex-specific thresholds were applied. These findings have important. These findings have important These findings have important clinical implications. Conventional cutoffs may miss advanced fibrosis in women, leading to delayed diagnosis and missed treatment opportunities. Sex-specific calibration enhances diagnostic equity and accuracy. This approach can improve early identification of high-risk patients and may be especially valuable in screening programs and primary care settings. Implementing the sex-aware threshold could improve the real-world decision-making and patient outcomes. In conclusion, sex-specific urine cutoffs significantly improve the accuracy of non-invasive fibrosis indices in MESLD. They reduce false negative and provide better diagnostic discrimination. Our results highlight the need to incorporate sex-aware threshold into routine critical clinical practice. Future multi-center studies are needed to validate these findings across the broader population. Thank you very much for attention.
Speaker? No. Speaker is Miss Louise Clinton. It's Emma. And she's from the Arab Emirates, and the title of the presentation is Improving Outcomes by Empowering Nurses to Lead with Change.
Yes, the arrows. Yes, forward. Forward.
Thank you. Hi, good afternoon. I'll try not to put you all to sleep. You've had a lot of listening that you've had to do. Taking quite a diversion away from gut health, which I've found fascinating, by the way, and I'm going to look into that much more. I'm the nurse manager of acute stroke unit in Abu Dhabi, and we have quality improvement projects all the time, every year, and I've just been helping my staff to embrace and look at transformational leadership and how their ownership of our unit's quality improvement can really enhance patient care, but their own well-being and their own sense of satisfaction as well. So I'm going to give you a bit of an introduction of why we chose the particular improvement strategies that we did, background behind it, the frameworks and models we used, what the interventions were, the results, a discussion about those results and what the implications are and what's next for us. So, bit of an introduction. Healthcare organisations critically appraise and implement cost-effective strategies that address nursing staff engagement, encourage behaviour that improves patient satisfaction and outcomes. So, for safe, person-centred care, we need to combine all of those three things. So, patient satisfaction, nurse satisfaction, and clinical outcomes. If the nurses aren't happy, if the staff aren't happy, if they're not supported, if they're not resourced, then they're not going to provide the best care for our patients. Our patients are not going to be satisfied, and the clinical outcomes will suffer as a result. So this is about looking at the whole unit and what I could do to enhance the nurses' ability to provide patient care, best patient care. So as a manager, it's my responsibility ultimately in health organisations, the patient's safety and well-being and positive outcomes are at the forefront of our minds. It's the ultimate game. But when you're a manager, you're not delivering that direct patient care. So your responsibility is to make sure that your staff, the nurses, and the other any other health manager needs to make sure that the staff have the resources, that they are adequately motivated, that you have the right skills mix, that you have the right amount of education and motivation so that they can deliver the best care possible. And that's why you need to look at it holistically. So this is about person-centered care, and I need to make that differentiation between patient-centered care and person-centered care. So patient-centered care is about looking at evidence, the best evidence for the care that we provide. So it's evidence-based practice, and what the patients feel that their personal needs are, and individualise that care to them. When you're looking at person-centered care, it includes the nurses as well. So it's not just about the patients, it's about ensuring that the nurses are provided with what they need so that that patient outcome can be optimised. So background, I manage a 25-bed acute stroke unit within a large 590-bed hospital in Abu Dhabi. Our primary value is putting patients first through advanced personalised care. And so the aim of our projects, we all have our unit-specific projects. The aim of the projects is to empower and inspire nurses to provide optimal evidence-based care that enhance the patient experience and optimises clinical outcomes. So the frameworks that we use, there are 3 frameworks involved here. Transformational leadership was at the forefront. I'm not going to give you a whole lecture on what that is. It's up there, but what we did is we used this framework so that we could promote nurse engagement. So transformation basically means change. So you, as a leader, you're involved in changing, but not just changing the outcomes in the unit, but in empowering the nurses to initiate those changes themselves. So promoting their engagement, enhancing satisfaction through various different means, improving patient outcomes, it empowers teams to align with the shared goals. So it makes you a team that all have the same goals in mind and all working together to achieve those goals. And using this model fosters a culture of continuous improvement and ownership of those improvement strategies. So what did we do? Well, we started for transformational leadership. In practice, what I did was initiate some positive communication, sending the staff to communication and patient engagement education activities on teaching them how to communicate with each other as well as patients. Initiating that myself, like you can't be an effective manager. I cannot expect the staff to behave in a particular way if I'm not willing to do that myself. One small little thing that we did was that I initiated every morning I would come in and say, whatever day it was, happy Monday, happy Tuesday, so that we had this culture of celebrating each day. It's a good day. We're going to start the day with a positive attitude. And it took off, and everybody says that now, and we all come on and we all initiate this positive attitude. And you might think that that's something small, but it is effective. A negative attitude is just as effective as a positive one. So if you can model the behaviour you want to see in your staff, if you want them to go into the patients to improve patient satisfaction and outcomes with a positive attitude, then you need to start with yourself and show them that you're going to be positive. Fair and equal treatment of people might seem basic, but you've got to make sure you treat everyone the same way and be flexible as a manager. So the other framework we used is the shared governance framework, where we have a daily team system, our quality improvement system, which is based on, I mean, Dr. Mohammed, first thing this morning was talking about the Toyota, or the Japanese car system. And Toyota initiated this daily team system, which is a daily management system, and we use that so that any quality improvement activities can be initiated from the housekeeping staff to the CEO, the porters, anybody can initiate an improvement idea. It's about bottom-up governance. It's about having unit-based councils and then taking those ideas and building on them and initiating them and running with them and sustaining them so that you're giving power to the frontline staff. And encouraging collaboration and teamwork, very important. So what do? We have the DTS board with staff suggestions, collaborative decision-making, and teams, created teams for each clinical outcome that we wanted to improve. And the other framework of the model was the PDSA model, which is what we use, which is iterative. It's continuous, and we look at it and we adjust it in real time, and then do use ongoing measurements to make improvements and to change. So what did we do? We put ideas into plans, trialed interventions, disseminated information, tracked the data, and then adjusted the plan accordingly. And these were the interventions that we used. It was simple, but it was effective. So the domain for patient satisfaction, we did the engagement and communication training. We reduced things like, and this was a lot of this was initiated by the staff, reducing the sound of the call bell volume, which patients had complained was too loud in the middle of the night. So we reduced something like that. And it might seem very small, but if you're a patient in hospital and you're being woken up regularly by the bells and alarms going off, it is very unsettling and very uncomfortable. Daily positive communication initiative, which I told you about, a flexible rota practices, which allowed them to have 2 requests a month, which might seem simple, but that wasn't happening before. And I newly came on as manager a year ago, so I initiated that. A fair rotation of charge nurses. It's a long story, but the most senior charge nurse was always getting in charge of shift, and if they were below her, they were being staff nurse, and it wasn't a fair rotation, and so they wanted to do that, and it worked. Multidisciplinary team involved in morning huddles that we talked about our initiatives with, and they were all involved. So we used allied health, physios, OTs, SLPs, everybody was involved in our activities. Creation of teams for nurse-sensitive quality indicators such as falls, pressure injury, so the team would take ownership of those clinical outcomes, daily status exchange huddles, and nurse leader clinical rounding. And these were the results. So our NDMQI RN satisfaction survey, so after initiating this, our 2024 results, the supportive manager went from 4.42 to 5.3. The sense of autonomy, because they were involved in each teams themselves and they ran with their ideas, improved. Not a lot, but it certainly went up. Job enjoyment definitely improved, and decision making, their ability to make decisions. So all of that contributed to RN satisfaction, which then resulted in great clinical outcomes. So this is the Press-Ganey patient satisfaction. Our benchmark is 85%, and that's the overall for nurses. So your overall, I don't know if you're familiar with Press-Ganey, but you can have an overall for nurse satisfaction, overall physician satisfaction, overall unit satisfaction. So we went from 85, which was that. And so it wasn't too low, but we wanted it to be the best. We wanted to excel. So we went from 85 to 90, 91. It came back down to 88, and the latest one's at 89. So we are now sustaining, and which is really good. That's what you want. The hardest part about quality improvement, the hardest part about change is sustainability. It's keeping that change going, keeping that improvement going. And then our hand hygiene techniques. So the one thing that we chose, doing hand hygiene at the bedside, we know from countless studies that hand hygiene is one of the things that, whilst it has the greatest improvement in outcomes as so far as preventing cross-infection, it is notoriously not adhered to. There's really bad compliance universally. So we wanted the actual technique to be done properly, not just they do it when they come in and out. It was the technique where we were failing. And the benchmark for that was 90%, and we were at, I can hardly see it, so it's very small, 80% is what that says. So we did drop in the first quarter of 2025, and I'll explain about that in a moment, but then we went up to an 87%, and the last one was 90 that we had, which I haven't updated this yet because I just received the results yesterday, 92% compliance, and this is the whole multidisciplinary team. We also, now these were incidental findings, so also we looked at, whilst it wasn't an aim of our project, we looked at falls per 1,000 patient days and hospital-acquired pressure injuries. Now you'll notice in each of those, we had a 2.3, and then we had a spike for the pressure injuries, and then we came down, and last quarter 3 was 0. And you can see quarter 1 again, 2025, there's a big spike and down, and then we're below the benchmark there. And these are incidental findings. And also you notice with the hand hygiene, the compliance dropped in that quarter. What happened in that quarter? We expanded our bed numbers and we didn't expand our nursing numbers. So we went from 15 beds to 25 beds without any increase in nurses. And that then translated, we were working chronically understaffed for that quarter, and that's the result. So without planning it, we could show evidence, clear evidence that ratios, that nursing numbers directly correlates with clinical outcomes and patient safety. So once we got towards the end of that quarter, we got extra staff. I got 3 extra nurses. Our staffing numbers improved and our clinical outcomes improved. So those were incidental findings that we weren't initially going to look at, but I draw this attention to people's attention, particularly our senior leadership, to say, look at this. And the cost of those things as well, the financial cost of pressure injuries and falls, especially falls with injury, is huge. So these results were very encouraging. So what does it mean? It means that relationships between workforce support, leadership, and patient safety outcomes is obvious. You can draw these relationships. Patient satisfaction results, staff levels that increased, improved outcomes, and getting staff buy-in and ownership is what really counts in making these improvements. The limitations, it was only in a single unit, so it's hard to know the generalizability. The project design didn't allow for statistical testing. It was a quality improvement project. It wasn't a controlled trial or any kind of, it wasn't proper research. But it was difficult to know what other non-measured variables could have impacted on these results. But the long-term sustainability is critical. And that's really important, this ongoing improvement, reinforcement to keep these positive outcomes. And leadership development, shared governance, and structured quality improvement frameworks is what drives sustainable transformation, sustainable change. And there's a link between adequate staffing levels, supportive leadership, and patient safety. Aligning workforce capacity with patient acuity, will prevent adverse events, and evidence-based staff-driven approaches can be scaled across other clinical areas to strengthen safety culture. You want a culture of quality improvement and safety. So what's next? Well, we're building on what we've done. We need to sustain the change, so keep that up, monitor these clinical outcomes, and monitor the satisfaction of both staff and patients to keep it going. These are my references, and that's all. Thank you very much.
Thank you so much. Thank you. You know, you said something that I was talking about my presentation being advising for change, right? And how I drove the transformation. I literally got goosebumps when you were talking about it because something so minor caused such, you know, even if it was a small change, me working in a hospital for 9 years, how important it is for all of us to have support because we're all connected, right? So,
Yeah, we are. Thank you.
Thank you.
I loved your presentation. Thank you.
وكمكمتها تخيلي خالة وصحية الصباح.
Did I say it correctly? Nice. From Algeria. And her title, her poster is Biliary Spectrum in IBD, a hospital-based case series.
Good afternoon, distinguished chairs, respected colleagues and dear participants. My name is Dr. Samsar Nawal. I am gastroenterologist at the university, at the gastroenterology department of the Oran University Hospital. It's in the west of Algeria. I am honored to present our study about hepatobiliary spectrum in IBD, a hospital-based case series. So, inflammatory bowel disease is a systemic disorder with multiple extraintestinal manifestations. And hepatobiliary manifestations and abnormalities are frequent. Their incidence is about 11 to 29% according to some studies. The cause of these abnormalities, of hepatobiliary abnormalities, may be independence of the underlying IBD activity. The aim of our study was to identify and to describe hepatobiliary abnormalities in patients with inflammatory bowel disease, Crohn's disease, or ulcerative colitis. So we conducted a retrospective descriptive study from May 2021 to August 2025. We included patients followed in our departments for inflammatory bowel disease. So these patients, they were either under anti-TNF therapy or they were awaiting for initiation, in whom hepatobiliary abnormalities were diagnosed. So as a result, we analyzed 77 patients. Most of them, more than the 2/3 of them, sorry. So more than the 2/3 of them were already under anti-TNF therapy. They were on infliximab and adalimumab. Less than the third were waiting for initiation anti-TNF therapy. In this situation, these patients were on immunosuppressive therapy or on the steroid or on mesalazine. The mean age was 47 with male predominance. And as a result, we had in our study 80% of patients who presented hepatobiliary abnormalities. and the different types of hepatobiliary manifestations as shown on this figure. The spectrum was very heterogeneous. The main finding was viral hepatitis. We had acute hepatitis C, acute hepatitis A. We had also hepatitis B, followed by primary sclerosing cholangitis, hepatic steatosis. We had also gallbladder stone, and we had also rare hepatobiliary manifestation such as hepatic granulomatosis in a patient who had Crohn's disease and in whom we diagnosed sarcoidosis, hepatic sarcoidosis. And we had very rare hepatobiliary manifestations, which was hepatic abscess in patients with Crohn's disease. We diagnosed these manifestations in before initiation of anti-TNF therapy in 3 patients, and in 10 patients, we diagnosed these hepatobiliary manifestations during the course of IBD. It was based on clinical and or biological abnormalities. In one case, we diagnosed primary sclerosing cholangitis preceding ulcerative colitis. So in this study, the hepatobiliary manifestations occurred in 80% of patients. This rate consistent with published data reporting prevalence of up to 29%. Liver involvement was mainly related to viral hepatitis, followed by PSC and cirrhosis and steatosis, reflecting the well-recognized etiological heterogeneity in IBD. Most abnormalities emerged during disease evolution, underscoring the need for regular hepatic monitoring, while their detection during pre-therapeutic assessment supports the systematic evaluation of liver function before initiating any immunosuppressive or any biological therapy as recommended by scientific societies. The identification of PSC preceding IBD in one case further highlights the strong association, it's a frequent association with IBD in almost 50% of cases, and the importance of colonoscopy. Overall, our findings confirm that hepatobiliary surveillance must be rigorous and multidimensional in order to allow early detection, in order to guide therapeutic adjustments, and ultimately to improve the prognosis of patients with IBD. So, in conclusion, hepatobiliary manifestations in IBD may be under-recognized and masked by digestive symptoms. Their systematic assessments should not overlook common causes of liver disease in order to ensure timely management and to improve the prognosis of IBD and the patient's outcome. So these were my main references, and at the end, I wanted to share a view of our city, Oran. It's in the west of Algeria, and our university hospital where this study was conducted. And I thank you for your attention.
Thank you, Dr. Noel. Does anybody have any questions? So then our last speaker for the day is.
You were the last speaker? No.
we have our speaker for today, Ms. Assad. From Saudi Arabia, and the title of her presentation is Perceived Value and Barriers of Nursing Specialty, Certifications Among Clinical Nurses in Saudi Arabia, a cross-sectional study.
Good afternoon. My name is Alawiya Sada. Hello. My name is Alawiya. I am from Saudi Arabia, and I'm working as a clinical resource nurse, clinical instructor. So my research about perceived value and barriers of nursing specialty certifications among clinical nurses in Saudi Arabia, cross-sectional study. The outline is introduction, review, review of literature, methodology, result, conclusion, recommendation, and references. Nursing certification is essential, and it validates the nurse's knowledge and improving patient care. It also supports career development. The nurses may face many challenges like lack of time to study, financial barriers, which may affect their ability to get the certification. Despite those obstacles, many nurses, they are trying to get these certifications to advance their career development. As we said, nursing specialty certification. Okay. Some of these challenges is institutional support and financial help. They don't have mentors, which can decrease the number of nurses who can get these certifications. Addressing these barriers through support and education can lead to better patient outcome and nurse development. So our aim is to develop the value, determine the value of certification among clinical nurses, identify the barriers. Our study is descriptive, cross-sectional study. We used PVCT, which is perceived value of certification, adapted from another research. And we added one more question about the barrier. And the setting was in KSUMC, which is King Saud University Medical City. It's the umbrella of three hospitals, which is King Khalid University Hospital, King Abdulaziz University Hospital, and Dental University Hospital. King Khalid University Hospital is about 1,000 plus beds. King Abdulaziz is 600, and DUH, which is the dental, it's only 40 beds for 1-day surgery. Our sample was 2,350 eligible nurses. We took 334, which has more 2 years and above experience. Okay. We analyzed data using SPSS and with the one-way ANOVA and this standard deviation, mean and the frequency. So our result was we have 94% of the nurses who verified that the certification had enhanced their knowledge, and about 84 who said that find certification more they find that the certification more they can make it more, they make them more competent, and 50% they believe that it gives them more autonomy, and more than 50% they said that give them more chance to get hired. The barriers were the highest one is lack of institutional reward, which was 72%, followed by cost of exam. So the conclusion, the study emphasized the perceived value and barriers of certification among nurses in KSUMC, while nurses' knowledge acknowledge the benefit of certification, such as improve competence and personal achievement. They face significant obstacles, including financial constraints and insufficient institutional support. So the recommendation were to link the certification to career advancement, promotions, and salary, flexible schedule, which we already did in the hospital, increased salary for the certified nurses, and recognize achievements. We already established one now policy for any staff who will get certifications. They will have reimbursement of the certification, so they will be more encouraged to do it. And we arrange with the managers to fix their schedules in case they will take the certification. They will have 2 weeks paid annual leave for them to prepare. And that's it. Thank you.
Thank you, Ms. Aloya Assad.
Assad.
You just told your name. Can you please come forward to your certificate and photo? Does anyone have any questions? Okay, that concludes our day one. Oh, let's have the same time. So we kindly ask that everybody arrive around 8:30, 8:40 for tomorrow, whoever's attending. When we're starting around 9:30, 9. Guys, it was a pleasure. I know that when I spoke, if anyone had questions or share anything, because I know it was, we were alternating. Okay.
Big city.